using Siglec-1 and PD-L1 surface markers on circulating monocytes. I have no conflicts of interest. So, as you already know better than me, idiopathic inflammatory myopathies are rare, heterogeneous, systemic autoimmune diseases. They are characterized by a broad spectrum of clinical manifestations involving multiple organ systems, but primarily the muscles. And they are divided into several subtypes according to clinical presentation, histopathological findings, and laboratory findings. And these namely are dermatomyositis, antisynthetase syndrome, immune-mediated necrotizing myopathy, inclusion body myositis, and polymyositis. However, the challenge to clinicians is that there is no single standard for the diagnosis of myositis, and the diagnosis is often relied on a rubric of tests for the diagnosis to be placed. But as early as we can put a diagnosis of myositis, the earlier we can treat, and therefore we can prevent irreversible damage. So we are in search of biomarkers that can classify patients as early as possible in order to monitor their, to classify them and monitor their disease activity as best as possible. So among these are interferons, which are a family of cytokines divided into type 1, 2, and 3, as you can see. They each signal through designated receptors and activate downstream JAK-STAT signaling, leading to the transcription of ISGs, which then have effects in innate immunity. In the context of many autoimmune diseases, we see these pathways being hyperactivated with no pathogen being involved, and this is true for the case of myositis as well. So, for example, dermatomyositis has an elevated, very heightened type 1 interferon activity, especially in patients with anti-MDA5 positive autoantibodies. Antisynthetase syndrome and IBM have mainly type 2 interferon activation, whereas in the context of necrotizing myopathy, we don't really see interferons having a role, or the literature anyway is scarce. Now, when we try to assess interferon activity, as we saw on Monday as well, we have several drawbacks in the available assays. First of all, when we try to directly quantify the cytokines, there we have problems with assay sensitivity, specificity, the transient expression of cytokines. And when we try to assess these ISGs, the so-called interferon signature, there we have problems with high cost and high turnover time of the assays, as well as the assays not being available to most institutions and requiring specialized centers. So these have necessitated investigating alternative markers for monitoring interferon activity in myositis patients, and we propose here Siglec-1 as a marker of type 1 interferon and PD-L1 as a marker of type 2 interferon, which are both surface markers on monocytes, and we can perform a very simple flow cytometry assay and have results within 2 hours of venipuncture. So on one hand, we have Siglec-1. This is a CLO adhesin found on monocytes and is involved in pathogen internalization. It's among the strongest type 1 interferon-inducible genes, and has been proposed as a very good candidate biomarker for interferon activity, and it has been shown in some pediatric cases of other autoimmune diseases, as well as in some studies in myositis. So on that front, we performed a pilot study in order to quantify Siglec-1 expression on circulating monocytes of myositis patients. So we enrolled 40 consecutive treatment-naive patients under the suspicion of a myositis diagnosis, as well as 10 healthy controls, and we performed flow cytometry for Siglec-1 quantification on monocytes, and also qPCR for the quantification of type 1 and 2 ISGs. As you can see, patients had elevated Siglec-1 expression on their classical, mainly, and intermediate monocytes compared to healthy controls, but not in non-classical monocytes, which is expected as the first to mainly participate in pro-inflammatory actions. And these findings for the classical and intermediate monocytes were statistically significant. Next, when we performed qPCR for selected type 1 and 2 ISGs, we performed for some more that I did not include for the sake of time, but mainly patients had elevated activation of MX1, ISD15, Siglec-1, the transcript, IP6, CPP1, and some others, and especially Siglec-1, ISD15, and IP6, as well as MX1, which is not included, had a very positive correlation with the Siglec-1 expression on classical monocytes. So on the other hand, we have PD-L1, which is a transmembrane protein expressed on APCs, and you might best know it in the context of cancers. But physiologically, it functions to promote self-tolerance, and it is induced by interferon gamma signaling. And a very recent study published in February of 2026 by Lam and colleagues in pediatric inflammatory disease patients confirmed that PD-L1 transcript levels reflected the interferon gamma signature, while also the PD-L1 protein levels on monocytes only marked interferon activity, highlighting that PD-L1 can serve as a biomarker for interferon gamma activity, so type 2 interferon activity. So having all this in mind, we want to establish monocyte, Siglec-1, and PD-L1 as protein biomarkers of type 1 and 2 interferon activity in myositis patients for the first time. To do so, we have set the following objectives. First, we want to characterize circulating monocytes, Siglec-1, and PD-L1 surface markers via flow cytometry. Next, we are going to profile type 1 and 2 interferon pathway activity by sorting monocyte subsets and performing qPCR for selected type 1 and 2 ISGs, as well as performing targeted proteomics on the sorted monocytes. And next, we're going to integrate the generated data with clinical features such as myositis subtype, disease activity, organ involvement, and autoantibody status. And all these are going to lead to the verification of Siglec-1 and PD-L1 as very good type 1 and 2 interferon markers in myositis. For these, we expect to include about 90 consecutive treatment-naive patients that are present to us for the detection of autoantibodies, the MSAs and MAAs, and upon the written informed consent and from a single venipuncture, we're going to perform the autoantibody detection, as well as all the methodology I just described, which is flow cytometry, the sorting of subsets of monocytes, qPCR, and targeted proteomics. These patients are going to be followed up by expert clinicians in order to collect the aforementioned clinical data to perform correlation and statistical analysis. In terms of budget, we are practically ready to begin, even from tomorrow, and we expect this project to last for 12 months. The budget will be distributed to consumables for the experiments, as you can see in the pie chart. And we believe that the project is very feasible. We have an established patient recruitment pipeline. We have over 130 patients so far in 2026. We have the available infrastructure and the know-how to perform the experiments. Our preliminary data have already confirmed the one part of the project, that Siglec-1 is a good marker of interferon, type 1 interferon activity in myositis patients. We have a multidisciplinary team, some of which is here today, to collect all the clinical information and also perform the experiments. And we have the important ethics committee approval in place, which won't take time off our scheduled timeline. Now, this has a great impact since Siglec-1 and PD-L1 via this flow cytometry-based assay we can have a quick snapshot of the patient's interferon activity at any given time point via just 100 microliters of blood. And this can pave the way for tailored therapeutic approaches, especially as we live in the era of interferon pathway inhibitors that can help, and this assay can help us stratify patients as early as possible and also allow the follow-up of patients upon treatment. Thank you very much.
Thank you, Sylvia. Now we open this project for questions from the audience or the jury. You have the microphone. Maybe if you want to make some questions, just please come to the microphone closest to you.
Thank you for your talk. Could you tell us how this would offer an advantage over using type 1, type 2 interferon gene scores?
So how this is more helpful over gene scores? Okay. So first of all, it's a much quicker assay. It can be performed alongside, let's say, leukocyte counting when we do this assay. It's easier, so that does not. So meaning we don't have to do RNA extraction, cDNA, qPCR to perform the interferon score. In terms of interferon score, we also have problems on which ISGs to include, especially because some ISGs are affected by both type 1 and 2 interferon pathways. And lastly, it's very cost-effective compared to the qPCR, and it can be applied to many institutions, not just specialized centers. Thank you.
Yes, thanks for the talk. Just one more question. Flow cytometry has a quite high variability between patients. With just 90 patients and stratifying by subgroups, you will have very small numbers per group. How are you going to handle that, like from a statistical point? You might suffer like for statistical power, basically.
That's correct, but with this budget we can only include about 90 patients in order to perform this experiment, but we can also continue this project and include more patients, or maybe open this project to more institutions, have a collaboration, and maybe we can compare from multi-center studies these findings.
Ingrid Lundberg, Sweden. I may have missed it, but how many new patients do you see in your institution per year?
So last year we had about 400 patients that came to us for autoantibody detection, and about 2/3 were positive for autoantibodies. We don't follow up all of the patients, but most of them we do, so we can have their clinical variables. So we're only going to include patients that we can have clinical variables for.
And how many of them were new diagnoses?
Most of them are, because we cut them early on during the when clinicians try to find what they have to figure out what the patient has. So they refer to us for autoantibody detection early on, which is among the first steps, at the same time as you might do the biopsy or the clinical assessment or any other like MRI or these things. So they're mostly new patients.
Thank you.
Thank you.
Lager from France. Do you know if there is any other signaling or cytokine that could induce PD-L1 or Siglec-1, just to be sure that it's the real reflect of interferon involvement?
Okay, so first of all, PD-L1 and Siglec-1 do not have any crosstalk in terms of their interferon, but I'm going to explain what I mean. So this study by Lam and colleagues in pediatric inflammatory disease patient performed an assay where they induced all peripheral leukocytes with interferon alpha, beta, and gamma, and they saw that when they induced with alpha, we had Siglec-1, and when they induced with gamma, we had PD-L1. So there is no crosstalk there. But definitely, I don't think there's any other cytokine that can induce this. Probably there is, but maybe not on monocytes. Sorry? You have to verify because you have cross-reaction with probably like TNF. Yes, we have, yes, definitely. But that's why we're going to perform the verification with qPCR to see if the signal we're getting on by flow cytometry is a true signal reflected by the ISG expression. And also, we're going to perform some targeted proteomics for selected cytokines, chemokines and stuff.
The last quick question.
Yes, this is a very relevant question. So in children, at least, we see sometimes that Siglec-1 does not correlate with interfere with ISG at all. Okay. There is a huge discrepancy. Okay. How are you going to handle these cases, for example?
Okay, so we haven't seen this. We had a very positive correlation on these, the Siglec-1 on flow cytometry and the Siglec-1 transcript on total leukocytes. How we're going to handle this? Probably look at the literature and trying to find ways to explain this, and probably needing more upstream pathway deconvolution.
Okay, thank you. Thank you, Sylvia. Thank you very much. So now, Dylan, please. We are going to hear about the project Dylan Velas. Please, you can come. Idiopathic inflammatory myopathies, morbidity and mortality risk factors from the French National Health Data System and the French National Rare Disease Registry. So Dylan comes from France.
Good morning, everyone. My name is Dylan Velas, and I am pleased to present to you, thanks to Myositis UK, my PhD project entitled Mortality and Healthcare Consumption in Idiopathic Inflammatory Myopathies. Myopathies were initially described in two subgroups, polymyositis, which are muscle-specific autoimmune diseases leading to muscular impairment, and on the other hand, dermatomyositis, which are systemic autoimmune diseases that can be life-threatening due to extramuscular involvement, such as interstitial lung disease, cardiovascular complications, and association to cancer. Revised into 4 subgroups, 2 muscle-specific autoimmune diseases, the IMNM and the IBM, and 2 systemic autoimmune diseases, the dermatomyositis and the Antisynthetase syndrome. We know that myositis are chronic diseases and that the control of the inflammation in muscle is reached at a high cost. The treatment often includes corticosteroids at high dose, often associated to disease-modifying antirheumatic drugs. However, what is not known to date is what is the survival and the causes of death according to the new subgroups of myositis, and what is the extent of the treatment toxicity regarding the activity duration of the disease. To assess those questions, we used 2 complementary and independent data sets. The first one coming from the Drómos study, which is a nationwide case control cohort that previously linked the BNDMR, the French National Rare Disease Registry, to the SNDS, the French National Health Data System. The first one gathers the data from the rare disease networks in France, from which we used the identification of myositis subtypes conducted by expert clinicians with diagnosis and first sign dates. The SNDS contains all the inpatient and outpatient healthcare claim data with death certificates, and it also includes disease-free French people that were used as control after matching to myositis patients. However, the Drómos study dataset doesn't contain neither clinical data nor paraclinical exam results. That is why we had to build a replication cohort, a monocentric one, coming from the French National Expert Center for Myositis that was not part of the BNDMR at the time, and from which we were able to record the MSA status, the interstitial lung disease, and the cancer status. Finally, we included adults with a myositis diagnosis identified for one of the 4 subgroups of myositis by alpha codes or clinician diagnosis. We excluded the overlapping myositis, and the controls available in the Drómos study dataset were matched on the sex, the city of birth, and the year of birth to cases on a 1:3 ratio. To study the survival, we used a classical Kaplan-Meier estimate in both databases, and in order to estimate to quantify the impact of the covariate on the survival, we used univariate and adjusted Cox models, first on the pre-diagnosis Charson comorbidity index, which is a score that synthesizes the comorbidity of a given patient, available in the Drómos study dataset, and on clinical features such as ILD and cancer status from our replication cohort. Finally, to study the causes of death, only available in the Drómos study dataset, we used an univariate fine and gray model to take into account for competing risks. We are going to start by the Drómos study dataset, from which we included 1,888 cases of myositis, which were matched to 3 times more controls. And the Charson comorbidity index prior to diagnosis of myositis was significantly higher in cases of myositis compared to controls. And the mean age at diagnosis and sex ratio were consistent with the literature. This is the Kaplan-Meier curve of the overall myositis survival from the Drómos study dataset, with an increased risk of mortality. Indeed, the adjusted hazard ratio is estimated to 3.35. When we stratified this analysis on the subgroup level, we showed that every subgroup of myositis, and not only the systemic diseases ones, were associated to an increase of mortality even after adjustment. The higher hazard ratio is for the dermatomyositis, which is estimated to 5.11, and the lowest one is for inclusion body myositis, which is estimated to 2.46. Regarding the causes of death, we saw that the cancer was the main cause of death of patients affected by myositis, and that the distribution was significantly different from our control population. But when we compared the increase of risk of death from a given cause of death, it was the respiratory causes that led to the most increased risk of mortality in myositis patients compared to controls. Finally, what we can conclude from this first analysis is that there is a decreased survival for every myositis subgroup, and that the mortality is mainly increased for respiratory causes. Now we are going to switch to the replication cohort analysis results, from which we included 839 patients. The demographic data were similar to what we have from the Drómos dataset, and we included 30.4% of ILD and 14.9% of cancer. As we didn't have any control population, we compared here the subgroups to each other, and the dermatomyositis was the only subgroup associated to a decreased survival with an adjusted hazard ratio on age at diagnosis and sex of 2.11. On the other hand, the inclusion body myositis was the only subgroup associated to a decreased risk of mortality with a hazard ratio of 0.37 after adjustment. When we put in perspective those results from what we have from the Drómos dataset, we see that this is consistent with the dermatomyositis, leading to a significantly increased risk of mortality. When we studied the covariates available in the monocentric replication cohort, we found back that the ILD was indeed associated to cancer, to decreased survival, but was also the cancer. And finally, when we were able to study the MSA impact on survival, we concluded that the anti-MDA5 was the only serotype associated to a decreased survival with an adjusted hazard ratio of 3.03 compared to every other MSA, when on the other hand, the anti-GO1 was associated to an increased survival only in the Antisynthetase syndrome population. So we can conclude from the analysis of this replication cohort that it helped us to improve the accuracy of our previous results, and that the MSAs are indeed associated to different survival profiles. Now I would like to go a bit forward to the next step of the project, which is the estimation of the disease activity duration. We are conducting this by estimating an activity score based on the corticosteroids and DMARDs use. Indeed, we expect the controls in blue to stay at the same level of medication load a long time, when the patient affected by diagnosis, by myositis, would have a high medication load at diagnosis that would progressively decrease to join the same medication load that the one of controls when the activity of the disease would be over. Finally, this study is based on a 2,727 patients nationwide cohort that already has showed decreased survival for every myositis subgroup, and again, not only for the systemic diseases. And for the first time, we were allowed to study the impact of every myositis serotypes on the survival. Now we are starting to study the disease duration and the treatment toxicity. Indeed, we discussed here of many times of the new drugs that are currently in development, and we know that it is crucial to improve the knowledge that we have to more precise the population of our patients in order to give the good treatment to the good patient. I would like to thank the Professor Anne-Sophie Janot and Eva Lendabach for their supervision, and also, obviously, to thank again the Myositis UK charity to give me the opportunity to talk this morning. Thank you.
Thank you, Dylan. And now we open for discussion. Please come to the microphone with your questions. Ingrid.
Yes, thank you. If I understood you right, you are going to, in the first study, you are going to use a national registry. So my question is, how do you identify your myositis from this registry, and how have you validated the diagnosis in the national registry?
So these are data that come from expert center in France that are linked in rare disease networks. And there is a software that is developed, that has been developed previously, that is dispatched in this center, and these are the expert clinicians that just adjudicate the diagnosis. And we, on the other hand, we kind of validated that by the study of the medication, the hospitalization, the paraclinical exam, because from the Drómos study, maybe I can go back to, from the Drómos study, we don't have any clinical data, but we have every consumption of even MRI, NMG with a dedicated code, which are precise one, and that is how we assure that we were only including confirmed myositis patients. I didn't show the flowchart, but we screened the population, of course, at the beginning.
So if you are only using expert centers, is there a risk of selection for more severe cases?
I don't think, because in France there is this way to refer every patient in a given area to the referral center in the dedicated area. There is one coordinating center which coordinates the many other expert and competent center dispatched on the French territory. And so there is almost exclusively two kind of follow-up, maybe a regular one in a traditional center, and maybe once a year an evaluation in the expert center.
Thank you.
You're welcome.
You can go, Hector.
Hector Chinoy, Manchester. Nice slides. Slightly unclear, what do you need the funding for?
Yeah, of course. I didn't present that. We would like to join the monocentric replication cohort to the SNDS, which is not done. Which is not did today, because the replication cohort was not part of the nationwide cohort when it was launched, when it was previously linked to the healthcare claim database, and that would allow us to study the treatment and the healthcare consumption of the patients followed up in the replication cohort at the national, extra center level, and also to get the death certificates that we don't have yet for those patients, and to study the causes of death.
So what's the funds going to be spent on? Is it an application fee or the fee for the death certificates?
Sorry, could you...
The funds, are they to be spent on applying for the death certificates, or...
Not applying, because it's already available, but the patients are not linked to their given death certificates.
So I think the question is, how are you going to spend the budget that you are going to for this grant?
Yeah, it's something that costs money, unfortunately, to do this kind of analysis, because we are not conducting the analysis. In France, there is a dedicated organism that conducts the analysis and that gives back the data, and that is not something that is free, unfortunately.
Okay.
Okay, so if there is no other question, thank you very much, Dylan. Julia? So now we will hear from Julia Quiring, also from France, on the project Metabolic Dysfunctions Induced by Anti-HMG coagulant antibodies in Immune-Mediated Necrotizing Myopathy. So please, Julia.
Hello, everyone. Thank you for being here today. I would like to thank the Myositis UK and Gcom 2026 Local Organizing Committee for selecting my projects. My name is Julia Quiring. I'm a third-year PhD student at the Research Center for Biomedicine in Strasbourg. I have no disclosures. So immune-mediated necrotizing myopathies, also called IMNM, represent a specific, with anti-HMG antibodies, represent a specific subtype of inflammatory myopathies characterized by the presence of these antibodies in the patient's serum and by myofiber necrosis. Although the presence of anti-HMG antibodies is highly specific for this condition, the mechanisms by which they contribute to myofiber necrosis remained unknown. So to date, there are two other types of non-autoimmune necrotizing myopathies that can induce myofiber necrosis. There are toxic myopathies induced by statins, drug that lowers cholesterol by inhibiting HMG-CR, which is the key enzyme in the cholesterol pathway that converts HMG-CoA into mevalonate. Statin-induced myopathies are characterized by the presence by myofiber necrosis and by the accumulation of lipid droplets in the myofibers. Similarly, there are genetic myopathies that are due to a loss-of-function mutation in HMG-CR. So recently, Professor Andrew Mammen's team has shown that myositis autoantibodies, including anti-HMGCR autoantibodies, are internalized within the myofibers in the same subcellular compartment as their autoantigen. So in this context, it has been hypothesized that in IMNM, anti-HMGCR antibodies could have a myopathic effect by inhibiting their antigenic targets. However, no direct evidence for this hypothesis had been reported to date. So during my PhD, we have confirmed that anti-HMGCR from IMNM patients are internalized in the myofibers in the same subcellular compartment as their autoantigen. We have demonstrated that anti-HMGCR antibodies in vitro can inhibit the enzymatic activity of HMGCR similarly to pravastatin, and that biopsies from anti-HMGCR IMNM patients are characterized by the accumulation of lipid droplets in the myofibers. Moreover, we have demonstrated a myopathic effect of anti-HMGCR antibodies internalization in vitro using a model of human myotubes electroporated with anti-HMGCR antibodies purified from patients, control IgG and pravastatin. Red oil and hematoxylineosine staining showed that anti-HMGCR, but not control IgG, induce an accumulation of lipid droplets and a necrosis that is similar to that induced by pravastatin. So these results demonstrate that anti-HMGCR antibodies are internalized in the myofibers, that they inhibit HMGCR activity, leading to the accumulation of lipid droplets in the myofibers and ultimately leading to myofiber necrosis. So together, these results, the aim of these is to then evaluate the prognostic and therapeutic relevance of lipidomic imbalance in anti-HMGCR IMNM patients. The first task will be the assessment of HMGCR activity in the muscle of anti-HMGCR IMNM patients and its prognostic value. And for that matter, we will use a cohort of deltoid muscles issued from 76 inflammatory myopathy patients, including 18 anti-HMGCR and 10 controls with no neuromuscular diseases. For each patient, they're all prospectively sampled and untreated at time of the biopsy with no statins nor immunomodulatory drugs. Importantly, for each patient, we have a comprehensive data set of 90 clinical and biological variables that have been collected at baseline and throughout the 3-year follow-up. So because the direct measurement of HMGCR activity in tissues faces several technical barriers that haven't been resolved to date, we will measure the stable metabolites upstream and downstream of the enzyme as an alternative and use their ratio as proxy to measure HMGCR activity. We will then study the association between this activity of HMGCR in the muscle and the severity of the disease at baseline, as well as the outcomes of the patients during follow-up, both that are available in our 90 variable datasets. This will allow us to observe the level of HMGCR activity in the muscle of anti-HMGCR IMNM patients and its prognostic value. To provide a comprehensive landscape of the impact of HMGCR inhibition on the cholesterol-related pathway in the muscle of IMNM patients, we will then use and analyze those deltoid muscles through ultra-performance liquid chromatography mass spectrometry in order to, well, this method will allow us to identify the metabolites that are specifically dysregulated in the muscle of anti-HMGCR IMNM patients through a comprehensive and unbiased assessment of lipidomic dysregulation in the muscle that have been previously observed at the histological level. The second task will be the validation in vitro of those lipidomic dysregulation that we have just observed in the muscle of anti-HMGCR IMNM patients in task 1. And we will confirm the role of the internalization of anti-HMGCR antibodies on those lipidomic dysregulation using our model of human myotubes electroporated with anti-HMGCR antibodies. We will then look at the metabolites that were most strongly dysregulated in the muscle of patient in task 1 and analyze them through targeted mass spectrometry, which is highly sensitive and specific for hundreds of lipid species. So these results will help us validate in vitro that lipidomic dysregulation in the muscle of anti-HMG-CR/MNM patients are due to the internalization of antibodies. We will then unravel the pathogenic consequences of those accumulation of metabolites or deficiencies of metabolites by overloading human myotubes with accumulated metabolites on one hand, and on the other hand, on using strategies that would block the biodisponibility of deficient metabolites. We will then look at the metabolic consequences, which are the lipid accumulation through red oil staining and the necrosis with hematoxylin eosin staining, dystrophin immunostaining, and creatine kinase level in the cell supernatants. These will allow us to unravel the myopathic effect of each of the lipidomic dysregulations observed in the muscle of anti-HMG-CR/IMNM patients. Finally, we will explore the therapeutic potential of correcting those metabolic imbalances in vitro using our in vitro model of anti-HMG-CR/IMNM and trying strategies that would allow the clearance of accumulated metabolites on one hand, and on the other hand, the supplementation of metabolite deficiencies. We will then look at the reversions of the myopathogenic consequences, which are lipid accumulations and necrosis, as said before, to test and assess of the potential of correcting lipidomic imbalance in anti-HMG-CR/MNM patients. So there are several perspectives for this project. The first one is the identification of novel diagnostic and prognostic biomarkers, and also new therapeutic paradigm by testing those strategies first in experimental anti-HMG-CR/MNM model that have been already established at the Strasbourg Myositis team laboratory, and then in humans by combining immunomodulatory treatments with those correction of metabolic imbalances. The feasibility of this project is excellent, with prior data that have been published, technical and scientific expertise in anti-HMG-CR/MNM that I have gained throughout my PhD, a strong background at the Strasbourg Myositis team with preclinical models established and validated, pipelines for the generation and analysis of the experiments that are fully established, prospective cohort of non-treated patients, and the feasibility of lipidomic analysis in the muscle that has been validated on test samples. This project also benefits from local and international collaborations. And finally, the SPIT funding would cover entirely the cost for lipidomic analysis, whereas other experiments and salary have already been secured by dedicated funding. I would like to thank you all for your attention. I would like to thank the Strasbourg Myositis team, all of our collaborators, and a special thank you to my PhD supervisors, Professor Alain Meyer and Dr. Margherita Giannini, for their ongoing support. I'd be happy to take any questions.
Thank you. Thank you, Julia. So, are there any questions? Christopher?
Yes, thanks for the talk. Mass spec lipidomics usually do not detect cholesterol very well unless we have a very specific setup for the mass spec. How are you going to handle that? Do we have like a specific cholesterol-based lipidomics pipeline, or how are you going to do that?
For the mass spec, the first thing we want to see are most lipidomics. For the cholesterol, we want to do another strategy. We use another strategy. It's a fluorimetric assay that has already been tested on muscle samples, and we can look at total cholesterol, free cholesterol, and ester cholesterol with that kit and quantify those.
Okay, but for the mass spec, what kind of changes are you expecting if you're looking not at cholesterol but all the other lipids, basically?
I didn't hear you, sorry.
Oh, so sorry. What changes are you expecting for the mass spec lipidomics if it's not cholesterol? Like, what's going to happen with all the other lipids?
Oh, for other than cholesterol with mass spec, what we want to see, is that right? We want to look at the lipids that are, first thing we want to look at are fatty acids. That's a technique that has already been used by Professor Monica Guma in San Diego University. So first looking at the fatty acids, are they accumulated in the muscles of patients or not? And then we will look at sphingophospholipid and other types of lipids.
Okay, thank you.
Thank you.
Hi.
Hello.
Angeles Galindo from Sweden. I have a question regarding your longitudinal biopsies collected from the patients. Yeah. Because you wrote or you explained they do not have any treatment in the longitudinal. Is that correct?
We do not have a...
The patients are not treated.
No, they're not treated. When they have the biopsy, they don't have immunomodulatory.
At baseline.
At baseline.
During the longitudinal, they do have treatment.
Yeah. During the follow-up, they're treated, yeah.
How do you explain that probably you will have some variations from treatment, for example, corticoids?
The impact of that on our analysis? Well, we will analyze muscles, but just that we've obtained at baseline, so we won't see the impact of treatments on those muscles because they're not treated, if that was the question.
No, I was just thinking that probably in longitudinal samples, patients that are treated might can't find your results because of the treatment itself.
Yeah. Well,
so I think the question is more related to the use of corticosteroids and how that can maybe impact the lipid profile of the...
Oh, yeah. Maybe it can impact it, and that's why we want to try to test other, spare steroids using metabolites that won't affect those biopsies.
Jens?
Jens Schmidt-Rütersdorf, nice talk, thank you. Can you allude a little bit regarding oil red staining in myositis? What's the fraction, what is known in your samples and the published literature in myositis in HMGCR compared to other subsets of myositis? It's part of the routine protocol, oil red staining, so if your hypothesis is correct, this should have been popped up several times.
So are you asking, with those technique redo staining, what we've observed in our patients compared to other inflammatory myopathy patients?
What is known in the literature or in your lab findings in HMGCR positive patients when you do oil red staining, which is a routine workup for muscle histology?
Yeah. Okay, thank you. So what we know in the literature is that we knew that statin-induced myopathies induce this accumulation of lipid droplets with redo staining. In other myopathies, nothing was shown about that. So it's the first time that we've shown those lipid droplets. I haven't read in the literature before that it's a routine, but I haven't read it in the literature before.
Last question.
Maybe I just moved that. Okay, thank you for this beautiful presentation. I'm very interested because we have many patients actually for whom we have no real efficient therapeutic approach. My question is about the fact that have you think about looking for the very well and deep defined lipid blood analysis in comparison? Like, I know there is something that is going on muscle fiber, but will you compare that with the lipid profile in blood?
In the serum? So the first part will be the analysis in the muscle because that's where it happens. But after all those analyses, we want to look at the same patterns, but in the blood. Yeah.
And the very last one.
I was just going to address what Jen's question about the oil redo stain. It can be subtle, and I don't think it's been quantitated in a lot of myositis, like what's a big lipid droplet, what's more. So yeah, we see this in HMGCR, but it hasn't been quantitated very well, and it can be very subjective. It's like what makes a fiber have increased lipid, or how many lipid droplets do you have to see, and what's the size of the lipid droplets? More quantitation has to be. When you see it, but it's subjective.
Okay, thank you. So thank you very much.
Thank you.
Thank you. Nathan, please. And so now we are going to hear about the project on long-read analysis of GWAS-lossy in inclusion body myositis by Nathan Rutledge from the UK.
Hi, I'm Nathan Rutledge. I'm a second-year PhD student. I'd like to first thank you for allowing me to come here and present, and thank you to Myositis UK for giving me this opportunity to potentially do this project. So I have no conflicts of interest. So as many of you are probably already aware, inclusion body myositis is part of the idiopathic myopathy group, and it's got a prevalence of about 3 to 10 per 100,000. It's the most common acquired inflammatory myopathy in the ages over 50, and it's a slight predominance in males. But the important thing for this project is that there's no been real response to any immunotherapy or treatments, and when it comes to clinical trials, most of them have failed, although there are some ongoing. So one of the reasons for this could be its complex etiology. So like other myopathies and myositis, you have the autoimmune features that you see, the infiltration of the T cells, but you also have these kind of degenerative features like rim vacuoles, protein aggregates, things you'd see in things like Alzheimer's in the brain, you see in the muscle in IBM. Now, one of the hypotheses for this could be the autophagy or the proteasomal degradation pathways being dysregulated at some point, and there's some rare variant analysis that kind of backs up this idea. So we finally performed the first large-scale IBM GWAS with over 1,000 cases after quality control, and we can now have a good actual look at the genetic architecture of IBM. Now, you might notice some hits that have come up before, so P2PN22 has been well associated with other GWASs when it comes to myositis. Of course, the HLA region has come up many times, and SH2B3 as well, but there are some new ones, such as AHRR, OMDL3, and RMI2. So to follow on from this, we then looked at some RNA-seq muscle data. So at UCL, we did a collaboration with Italy. We had 25 RNA-seq data from skeletal muscle from IBM, as well as the published finished data set I was allowed to look into. And on the left, you can see just kind of the volcano plot, the distribution of the RNA-seq. And then on the right here, the more interesting one, it's a bit small, you can see in orange are all those GWAS genes from the loci in terms of the RNA expression. And you see that most of them actually aren't really that differentially expressed. And in the top, you see the top 50 dysregulated genes when it comes to the RNA-seq data. Now, there are a few up here that you can see, so it's P2PN22, SOC1, but there is one that actually appears in both data sets, and that's IKZF3. So what is IKZF3? It's a transcription factor. Its main role is in B-cell regulation. It's also got a role in T-cell apoptosis, and there's been research into the fact that it's got a role in CD4+ T-cell differentiation, and also one of its targets is CXCR3, which is a cytokine involved in telling T-cells to migrate to inflammation. So for IBM, it's a really interesting gene to come up when it comes to genetic analysis here. And if you zoom in on the locus plot, you can basically see here is this kind of purple SNP. It's like your lead SNP, and if all the other ones in different colors are telling you how likely you are to inherit a SNP when it comes to them. You'll see that most of these SNPs are not actually in IKZF3. They're further before IKZF3. And if you put this into some kind of, it's called RegulomDB, to look at predicting, do they have an effect on gene regulation? Are they in enhancer, transcription start sites? It's predicted that this might be the case. Now, when it comes to GWAS and looking further, this is kind of one of the endpoints you come to. There are other techniques you can look at, like high-chip-seq, you can look at further. But one really good way to look at this is to further on and do long-read sequencing. Now, how will the long-read sequencing again expand what we had done in the GWAS? Well, you could look at structural variants. You can look at duplications, deletions, inversions that you just don't see in a GWAS or in short-read sequencing. Also, if any expansions suddenly appear, we can also see them come up. You can also look at long-range regulatory elements that come up, because you have a longer fragments to look at and see how that's affecting. And then finally, you can also look at methylation. This is all done in one analysis that comes up. So you can look at epigenetic factors that might give us an indication what we're seeing from those RNA-seq results and the GWAS results. So what is the plan here? Well, we're going to investigate those GWAS loci using something called adaptive long-read sequencing. And the idea of adaptive long-read sequencing is that you don't look at the whole genome because there's a lot that you don't want to look at. You're able to focus on a few bits that are of interest, and you get a better read coverage because of that. So we'll take 10 IBM skeletal muscle samples and 4 controls. We'll then extract high molecular weight DNA, and then use it, we'll prepare with the library preparation with the Oxford Nanopore ligation sequencing kit. And then we'll also have a bed file prepared with all those GWAS loci and any other loci of interest that will then be implemented into the Oxford Nanopore Promethean sequencer. From that, we can then align and base call using MiniNote or Dorado to get our long-read sequencing data. And you can also use software like Sniffles and Dorado to look at methylation and structural variants, and then that can be then visualized using IGV. So how is this going to work in terms of timeline and costing? Well, the big thing we have at UCL is we actually have a long-read sequence service. It's very robust. They've done this pipeline many, many times before. We have good work with them, which means we know we're going to get good, robust results coming from this. And I've worked with them to come up with a plan. We could do this within 6 months. Once we have the samples that we need, we could do a short pilot to make sure everything is working well with 1 or 2 samples, and then that can be expanded to all 10 of them. And then when it comes to costing, we've also worked out that this would fit underneath the £18,000 budget. If it's £18,000 use, it's a bit over, but I'm sure my PhD supervisors could maybe contribute a bit to that, and there's a bit left in my PhD budget that I couldn't use if it was just doing the long read on its own. So what is the potential impact? We can hopefully further understand that genetic makeup we've just seen from the GWAS. Why are things like IKZF3, PTP2 being overexpressed? And why are those other GWAS genes not really showing any difference despite the genetic difference we're clearly seeing from the GWAS? Hopefully, it's only a small pilot, it's only about 10 samples, but it can give some indication that are there any new pathways or targets for us to look at that have not come up before, that we can then look into new trials. But also, I think most importantly, there's not been much long-read sequencing done on IBM to begin with. So any data that is provided here is good pilot data that can be used in any follow-up studies and can be used by anyone if they want to look at it further on. So I'd like to thank everyone involved, the International Myositis Society for having me, Myositis UK, it's not on there, of course my supervisor, Pedro, and the Neurogenetics Lab at the ION, where I am working mostly. Thank you.
Thank you, David. Thank you. Now we open for discussion. Questions? Yeah, okay.
Hi, wonderful talk. I have 2 questions, actually. One is, are you expecting to see something mutual among patients? Because there will be a lot of intra-individual variability as well. And how do you plan to look at the functional, like, to conform it on a functional level, what you see on the structural variant?
Confirming on a functional level, I mean, at the moment, I'm already also doing some muscle experiments when it comes to looking at protein expression as well. That is one of the problems, is the individuality. It is only a small sample set, but the hope is that once something comes up, that's what can be expanded on. The good thing is, because of the MyoGen consortium to begin with, a lot of the samples that could be available for the UK Biobank have already been in the GWAS, so we actually do know, we can quickly check if what's come up beforehand.
Christopher?
Yes, thanks for the talk. So IKZF is a marker for regulatory T cells. If you're comparing IBM to healthy controls, there are more regulatory T cells in the tissue, and the marker is going to go up simply because there are more T cells, I suppose. How can you disentangle that, or what exactly is the target you're looking for when it comes to the cell type? Do you think this is a muscle-specific effect, or you're actually looking at the immune compartment?
So that is a good question, and one of the, I guess, the problems of you doing this in the muscle rather than doing it in a cell-specific is you don't quite get that answer. But if I was to do it cell-specific again, it would be a lot more expensive to do it that way. I've mentioned IKZF3 because it's the most interesting target to come out. But the good thing about doing this, and especially the adaptive sequencing, is you don't have to just look at IKZF3. I mean, that is something I want to look at further in a separate project in different ways. But I think this is a great way of expanding the whole GWAS and the whole genetic architecture to begin with.
Okay, but the point I'm trying to make is if you're looking at any other muscle disease that has regulatory T cells in the tissue, that gene is going to go up, you know, because it's a marker for the regulatory T cells. So do you think that it's actually a genomic difference that's on the regulatory T cells, or is it something related to IBM muscle tissue itself?
That I'm not pretty sure. So it would make sense for it to be regulatory T cells, that would make sense. But I mean, from the RNA-seq data, again, that's bulk muscle. So it could be that it's included in there that not fully sure on. But I want to look at the muscle release to begin with. And if anything comes up, that would be then the next step, would be then to specifically look at the T cells.
All right, thank you.
Hector?
Nathan, as you know, PTPN22 is considered to be a pan-autoimmune gene. Yeah. It's been associated with many other autoimmune diseases. So what about IKFZ3? Is that been associated with other autoimmune diseases? What's in the literature?
Not from what I've seen. I mean, I've not seen it come up in any other genetic study, at least. So that's why I found it quite interesting that it came up like that. I think there has been some work when it comes to B-cell and other diseases as well. But at least in terms of genetic difference, I've not seen it come up before.
And has there been a signal for the gene in other IIM subtypes in prior GWAS studies?
Not that I've seen, no.
So it's just in IBM?
That's why I was quite interested in that, because that ORM-DL3 loci, which is what it was on, had not come up before in any of the other ones.
Okay, thank you, and thank you, Nathan. Thank you very much. Maho, please. So now we are going to hear about the project investigating pathogenic mechanism of antigen-specific B cells in anti-GWAS antibody-positive idiopathic inflammatory myopathies by Maho Nakazawa from Sweden.
Yes. Thank you for the introduction, and good morning, everyone. So first of all, thank you for this opportunity. And my name is Maho Nakazawa. I'm a postdoc at the Karolinska Institute. I don't have a COI. So I'd like to start this slide. I am a rheumatologist from Japan. And since when I was a medical student, the pathogenesis of autoimmune diseases has been an area of intense interest to me, especially myositis. And after I finished my PhD and training in rheumatology at Tokyo, Japan, I joined the myositis research group, Karolinska Institute, Sweden. And today I have the great opportunity to stand here at my first Gcom. So I have focused on the anti-GWAS antibody-positive myositis. As you know, anti-GWAS antibodies are the most common autoantibodies among myositis, and it causes severe and refractory lung diseases requiring aggressive immunosuppressive therapy. The pathogenic mechanism has not been fully revealed yet, but some previous studies suggest B-cell involvement in pathogenesis of GWAS-positive myositis. So, for example, anti-GWAS antibodies titer is related to disease activity. And after rituximab treatment, the titer of the anti-GWAS antibodies is decreased after the rituximab treatment, and it's correlated with changes in disease activity. And recently, the efficacy of CD19 CAR T cells in GWAS-positive myositis is reported, which highlights the potential of B-cell targeted therapy for GWAS myositis. So this data shows B-cell involvement in pathogenesis of GWAS-positive myositis. And its target is histidine transfer RNA synthesis, HISRS, which has 3 domains, and one located at the N-terminus is called the web domain. And sera from this anti-GWAS antibody-positive myositis patients showed high reactivity for this web domain. And interestingly, this web domain is reported to be highly expressed in the lungs. And germinal center-like structures were found in the lungs. So these previous reports support importance of antigen-specific B cells in the inflammatory site in GWAS-positive myositis. So I aimed to reveal the immunological characteristics and pathogenic involvement of antigen-specific B cells in anti-GWAS antibody-positive myositis in this study. So I have divided this project plan into 4 parts. The first part is identification of antigen-specific B cells. And second part is immunological profiling of antigen-specific B cells by immunophenotyping and single-cell RNA sequencing. And third part is B-cell receptor analysis of antigen-specific B cells. And fourth part is generation of anti-GWAS monoclonal antibodies. So in the first part, I will identify antigen-specific B cells. So we have collected peripheral B cells from anti-GWAS antibody positive patient and also healthy donors. And we are also collecting muscle biopsy and bulk fluid cells from the patient. So first, to capture his antigen-specific B cells, I prepare fluorescently labeled HisRS antigen, and we also prepare negative control using a random protein in the same way as a HisRS antigen to remove non-specifically binding B cells. So the antigen-specific B cells will be defined as B cells that are double positive for two different colors antigen after removal of non-specifically binding B cells. Then we will perform the detailed immunophenotyping analysis of antigen-specific B cells in peripheral blood and also tissue-infiltrated B cells using spectral flow cytometry, with which we can obtain the deep immunophenotyping data, but at the same time, we can sort the target cells. So antigen-specific B cell will be single-sorted and single-cell RNA sequencing will be performed. And we will combine immunophenotyping analysis data and gene expression analysis for the detailed immunological profiles of antigen-specific B cells to explore the therapeutic target. As I mentioned before, we collect the tissue samples as well. And we are also interested in differences in phenotypes and transcriptomes of antigen-specific B cells between tissues and peripheral blood, and also between before treatment and after treatment, since anti-GWAS antibody positive myositis is known to frequently relapse and to have severe tissue damages requiring intensive immunosuppressive treatment. So antigen-specific B cells may persist even after treatment, or antigen-specific B cells in tissue might have different profiles from those in the peripheral blood. So the immunological profiles of antigen-specific B cells that infiltrate tissues or remain after treatment are important for identifying therapeutic targets, potentially leading to novel treatment strategy for the relapsing or refractory cases. And then we also would like to identify the gene sequence of BCR variable regions of antigen-specific B cells and evaluate their chronality. As all of you know, each B cells have a different BCR. So to look the clone's CDR3 region is sequenced, and chronality assessment is important for identification of HisRS specific expansion, because it helps us if the B cells population comes from one dominant clone, which suggests strong antigen-driven expansion. And lastly, based on the BCR sequence data obtained from third part, we will clone and generate anti-GWAS monoclonal antibodies. These monoclonal GWAS antibodies will be applied to functional experiment in the next step. So through this study plan, so first we would like to identify antigen-specific B cells from the antigen-positive patient and investigate deep immunological profiles and assess BCR clonality and generate monoclonal GWAS antibodies in order to elucidate the pathogenesis of GWAS-positive myositis and search for new therapeutic targets. And monitoring antigen-specific B cells in patient might be useful to assess the disease activity of the patient. And moreover, this method is also potentially applicable to other antigen-specific antibody-positive myositis patient or other autoimmune diseases, which is of great social significance. So this study will be performed at CMM, Center for Molecular Medicine, in Karolinska Institute, and I would like to thank all people involving this study, and especially Ingrid and Caroline. And I also would like to thank the JSPS, Japan Society for the Promotion of Science. I have obtained the support of the postdoc scholarship, and all the patients with myositis. And thank you for your attention.
Thank you, Maho. So now we open also this project for discussion, for questions from the audience. So Lucy Weatherboard.
If it's right, thank you for a beautiful talk. Lucy Wedderburn from London. If it is correct that the antibodies themselves are pathogenic inside the muscle cell, we've heard a lot about that at this meeting, is it possible that the antigen-specific B cells will be the same transcriptionally as other inflammatory B cells? What do you think?
Sorry, the question is the antigens.
The question is, could you get a true negative from this data that all the B cells are inflammatory, so you won't see much difference between the antigen-specific and the rest of the B cells?
Sorry, I'm sorry. I don't know the echo, but
yeah, it's a terrible echo. She's right.
Yeah, there's a lot of echo here.
I'm just asking you about the transcriptional profile of the B cells, which happen to be antigen-specific compared to the bystander B cells. What do you think? What is your hypothesis? Why would they be transcriptionally different? She's right.
Thank you. Thank you for the question. So, thank you for the question. I'm not sure yet, but I believe anti-Drómos antibodies is pathogenic because some previous studies shows indirectly. So, for instance, as I shown in this introduction slide, Drómos antibodies is correlated with disease activity, and also, for instance, mice model, mice immunized Drómos DNA shows the production of anti-Drómos antibody, and also the developed myositis. And also in our group, it's 20 almost 20 years ago, but polyclonal IgG from Drómos patients shows the interferon alpha production. But with the RNAs, adding the RNAs in the simulation assay, there's no production of interferon alpha. So, which means, Drómos antibodies it can be pathogenic, but maybe it's with some, for instance, immunocomplex is maybe important.
Yeah. Yes. Thank you.
Heriditary.
Thank you. You said that one of the aims is to determine the clonality of the cells. For this, you need a certain number of the cells. So, do you have an idea what will be the yield from the lung in this sense, or from the muscle tissue, whether you can get enough cells to estimate the clonality?
Thank you for the question. Yes, maybe from the peripheral blood, the number of antigen-specific B cells is not so many, I expect, but at the same time, we will sort and analyze the memory, non-HISRS, non-antigen, but memory B cells at the same time. So we can we will analyze the other non-specific memory B cells as well, and then we can assess the clonality. I mean, yes.
Okay, thank you. So if there is no further question, thank you very much, Maho. And now please, Philippa, come to the stage. And now the last presentation is on the project, a comparative study of ultrasound-guided versus open surgical muscle biopsy for the diagnosis of idiopathic inflammatory myopathies by Philippa Costa from Portugal.
Good morning, everyone. My name is Filipa Costa. I'm a last-year rheumatology resident here in Lisbon at Hospital de Santa Maria. And I want to start by thanking everyone at Myositis UK, IMUs, and the Gcom organizing committee for the opportunity to present our project here today. So I'm very glad to present this project on the comparison of ultrasound-guided versus open muscle biopsy for the diagnosis of idiopathic inflammatory myopathies. I have no conflicts of interest to declare, and I will start by focusing on the clinical problem that was on the center of this idea. So as you all know, idiopathic inflammatory myopathies are rare diseases, and nowadays serological markers are getting more specific, and the methods for their detections are getting more and more sensitive. However, muscle biopsy is still very important for diagnosis and research purpose. But the patients that undergo muscle biopsies usually undergo open muscle biopsies. And this is a method that might be associated with more frequent complications. It depends on the series that we found, but up to 5% of cases. And this is mainly pain, post-operative pain, infection risk, scarring, and longer recovery times. And it might as well be associated with higher costs, depending on local practices. It might require an operating room. As you might have noticed, ultrasound-guided muscle biopsies have been described, and they show, they allow real-time visualization of the targeted muscles, and this is something that has been done before. So people screen the muscles to select the muscle to biopsy, but the advantage here is to use the ultrasound throughout the entire procedure, so we can assess neurovascular structures and avoid them when necessary, and select the most affected area within a muscle. The muscle might not be homogeneously affected, and we might have patchy areas where the inflammation is more notable, and the ultrasound allows to select that precise area. Our group published last year the efficacy and safety of this method using semi-automatic guillotine-type needles, and we showed that it is effective, it is safe, it is well tolerated, and it provides samples with good quality for histological analysis. On the right, you can see two images of the procedure using ultrasound. On the top image, you can see the administration of the anesthesia using ultrasound, which is also an advantage. We administer the anesthesia up to the fascia without introducing anesthesia inside the muscle, which might change the histological architecture. On the bottom image, there is the guillotine-type needle inside the muscle with its indentation where the muscle tissue stays after retrieval. However, there is a knowledge gap. We have no studies directly comparing the two methods, so we don't know, we are not sure if one is better than the other. So what we propose is to compare prospectively both methods in a multicentric study. So we want to have two groups of patients, one that will undergo ultrasound-guided muscle biopsy in our center, according to the protocol that I've just showed you, and another that will undergo open surgical muscle biopsies as per clinical practice in a partner center. So, our center particularly is a tertiary center. We have a multidisciplinary team. We have a group of dermatology, rheumatology, physiatrists, and most importantly, we have a neuropathologist that works with us, and it's used to assess our muscle biopsies, and it's going to work with us throughout this study. We are also part of national and international networks. We have around 250 patients followed in our clinics, and we have around 50 new patients every year, so we believe that we'll have enough volume of patients to proceed with this study. Regarding inclusion criteria, we expect to include every patient that would undergo muscle biopsy as per clinical practice. So we'll include patients with a suspected IIM, and that per clinical judgment will undergo a muscle biopsy. And we'll exclude the patients that would normally be excluded in clinical practice, so patients with known bleeding disorders, active infection, or the inability to consent to the procedure. We'll collect data, of course. We'll collect patients' demographics, such as age and gender, clinical data, such as myositis subtypes and MSAs whenever they are available, previous and current immunosuppressants, and analytical and clinical status by the time of the biopsy. We'll collect biopsy data, such as biopsy technique and biopsied muscle, including laterality, And procedure tolerability, VAS, so visual analogical scale for pain, during and after the procedure, complications, and the necessity for re-evaluation the patient after the procedure. We'll collect histopathologic data, such as the quality of muscle sample and histological diagnosis, and the procedure costs, direct and indirect, whenever that's possible. Our primary objective is to compare the diagnostic yield of both methods. As I was saying, there is a knowledge gap, there is no comparison between both methods, so we want to compare the diagnostic yield that will be defined as the proportions of biopsies that allow pathophysiological diagnosis of IAM. Secondarily, and really important as well, firstly, we will want to compare reported pain during and immediately after the procedure, compare short and immediate adverse events, and the necessity for the reevaluation of the patient after the procedure. This is a feasible study. Muscle biopsies are already performed in clinical practice, so we are not implementing a new method. And most centers already perform open muscle biopsy, and we showed the efficacy and safety of ultrasound-guided muscle biopsy, as I previously showed to you. And we, at our center, are going to perform the ultrasound-guided muscle biopsy, and we'll partner with a center that will perform the open muscle biopsies, and we'll expect to include around 30 patients, more if possible, in each of the centers. Timeline-wise, we expect to spend 1 to 3 months in the ethics approval, then the next 3 to 4 months for patient recruitment and data collection, 2 to 3 months for analysis and manuscript preparation, and then submission for publication. We allocate our budget to data collection and statistical analysis, PRO collections and patient follow-up, because patients will need to be contacted after the procedure to assess for complications and pain after the procedure. Research costs, and I believe this is really important because we proposed to perform this study with 2 centers. So histological analysis will be done expectedly by 2 different neuropathologists, and sometimes it might be necessary to transfer biopsies from one side to the other, and this, of course, has costs. Another option is to do all the analysis of all the histological analysis in the same center, and this will also have a cost to transfer biopsies from one side to the other, and of course, publication costs. Of course, we expect to have an impact. First of all, in our patients, they are the center of our work, and we want them to have the best experience possible throughout diagnosis, follow-up, and treatment. So we expect them to have a procedure that is less invasive and hopefully with reduced complications. We also want to have an impact on clinicians. Most of us here treat patients with myositis, and we want to have the best diagnostic tools to do the best diagnosis possible. And with the ultrasound-guided, we expect to assess the area of the muscle that has more inflammation and to have a greater likelihood for a positive or an histological result compatible with myositis. And we want to have an impact in the healthcare system. If we show that this procedure has lower costs, we can better allocate resources for our patients and for all patients. So as take-home messages, I want to state that as far as we know, there are no direct comparisons between these 2 methods, so that's what we propose. And we expect to have a procedure that is less painful, has greater accuracy, and less expensive. And I want to finish by thanking everyone at our department, the Myositis Multidisciplinary Team, but most of all to patients and their families, and my project supervisors, Dr. Raquel and Dr. Fernando. Thank you so much.
Thank you, Filipe. Thank you. Now we are open for discussion. Hector.
Thank you for your presentation. Has the issue regarding diagnostic yield, what were your findings in your retrospective study when you compared open to ultrasound-guided?
So we found that around 70% of the patients with at the biopsy that was compared to the clinical suspicion.
But your primary objective for your proposed study is the diagnostic yield.
Mm-hmm.
So what were your findings when you looked at this retrospectively? when you compared open, so you've got an existing data set where some patients...
But we have no comparison. The first study is just the efficacy and safety of ultrasound guided without comparison.
But you must have some data already.
No, because our center did not perform open biopsies. It was performed by the neurology department in our center.
Okay, but okay. So your histopathology department will have some biopsies from open biopsies and from ultrasound-guided biopsies?
Yeah, we have the data on the ultrasound-guided, yes.
But maybe you can compare from the literature the accuracy or at least the diagnostic yield, what is known from other cohorts between the diagnostic.
Yeah, it was really difficult to find that in literature, the precise diagnostic yield on open muscle biopsies.
But surely you can go to other centers in Portugal who have done open biopsies and found what the yield was and compare them to yours. Because that's on the basic histopathology report, is what's the size of the biopsy. So why do you need to do this prospectively? And also, it's just with your secondary objectives, you've got an inherent bias that your center does ultrasound-guided biopsies. So at least 2 of those secondary objectives will show a favor towards ultrasound-guided biopsies.
So you're asking if, because we perform ultrasound-guided, we'll be biased on the analysis for pain and short and immediate complications?
How will you address this?
We will compare the idea is to compare the open muscle with other centers. So other centers will assess complications by themselves. We can sort that by having someone that is not involved directly in the procedure, assessing complications and VAS for pain.
Okay, thank you.
Yes, please.
So a couple points. I think, looking at the diagnostic yield, it looks at more than just the biopsies, as we've learned from the conference. You're looking at the clinical pattern of weakness in the labs, so that's helped support it. You're not looking at the biopsy blindly in terms of a diagnosis. And for one, your yield of the biopsy, you don't go after the most affected muscle. Go after the most affected muscle, you're going to get end-stage tissue. So you usually want something that's mildly affected, but not severely affected. So something like an MRC grade 4. The level of I do all open. And the fact that you're not going to go, and I do my EMGs, it's painful. And the people that I've had, it's a big difference that have had a biopsy at other places that don't use anesthesia. You absolutely can use anesthesia. And you don't do it just to the fascia. You go into the muscle and you numb the muscle up. It is not going to give you perifascicular atrophy. It's not going to give you necrosis that fast. It's not going to give you inflammation. It's not going to give you rim vacuoles, P62 inclusions. It's just not going to do that. You don't want to do a biopsy, say, a week after you've done a needle EMG in that muscle. But you most certainly, and if you numb up the muscle in an open, they're not having that problem. And it depends on the biopsy that you're doing. If you're doing a little EDC muscle, I can make an open incision and 1/2 inch. If it's somebody you want to do a quadriceps muscle biopsy and they're a little obese, you have an inch and 1.5 incision. So there's a lot that goes in to this study that you might not get. And it really ought to be the same physician seeing the patient, diagnosing it clinically and doing the biopsy to see how much it's supported rather than taking it from different clinicians at different sites, different muscles with a variety of different inflammatory myopathies. And you should numb up the muscle. That should be the fallacy that you don't numb the muscle up for a biopsy because it's going to change the architecture. It's incorrect.
Was there a question?
If you want to comment briefly.
Okay.
It's okay. Thank you.
I just, if I can say something, we discussed with the neuropathology the question, the reason because of the anesthesia inside the muscle, and what we discussed was that it would change the architecture, and that's why we perform it like that. Okay. Thank you. Professor Shiri.
Short question related to Hector's one. I just want to be sure the biopsies will be done in 2 different hospitals. Will they be evaluated by the same histopathologist?
Sorry, the echo is really bad, but if I understood the question was the biopsies are going to be done into centers, or are we going to assess?
And who is going to evaluate the
okay.
biopsy? Single histopathologist, one person?
Yeah, that would be the biggest reason for funding, I believe. We would be able to have the same person do the biopsies, to analyze the biopsies. Our neuropathologist, the person that usually works with us. And just to answer what you were asking before, the reason to do it prospectively or retrospectively, I believe that one of the reasons to do it prospectively is that we are sure that we are collecting the same variables, as opposed to doing retrospectively. We might have lost data, even in the clinical files and so on.
I guess my issue is the first thing you do in a scientific project is look retrospectively to see what data is available. And we've not got that data, so it's just perhaps you could answer one of your objectives with some retrospective analysis first.
Thank you.
That was the point I was...
Yeah, thank you.
Okay, so thank you. We are short, sorry. So thank you, thank you, Felipe. And thank you for the 6 presentations, and once again, thank you all. And I would also like to thank our jury panel, not only for today, but all the candidates. So Lucy Wedderburn, Charis Papadopoulou, Christopher Nelke, Hector Chinoy, Ingrid Lundberg, Jens Schmidt, Pedro Machado, Hiri Venkowski, and Yves Allenbach. It was a long work, so thank you all. And thank you once again, Irene and Laz. I don't know if you want to say a last word of farewell.
Yeah, we'd just like to say thank the young, there's 6 of them. I think that, what we've heard today is very encouraging. I don't understand all of it. I only know a little bit of it, but I'm sure that the audience will understand most of it, and our judges will as well. And I'd just like to say thank you to everybody that's given their time before this, before the presentations, because there's a lot of work that went on before the meeting, a lot of assessments, a lot of emails backwards and forwards, especially to Raquel for all the work she's done setting all this up. And there's one other person I'd like to thank, really, and that's Jens, because back in Berlin in 2019, I think it was his idea of a speed funding, which we took up to sponsor. And if it wasn't for that, I don't think we'd be here today doing speed funding. So thank you to Jens, and thank you all for coming and listening to our youngsters. Thank you.
mais grave é que eu nem sequer consigo.
Esperemos que a sala cheia nos dê perguntas para as pessoas. Depois então, portanto, eu começo com este, depois o Rafael fala deste, depois eu falo do GR Ruim.
Opa, isso pode ser alternado.
E Jessica Day e o Nathan Rutledge. Ok, esses são nomes ingleses, são fáceis. Portanto, esta a senhora é da Austrália e este senhor é britânico, é aquele rapaz que eu já ouvi apresentar há bocado. Nathan.
O primeiro é mais complicado. O primeiro,
Agora,
eu não sei.
Se é este senhor da frente, será que
Podemos pôr aqui
dele.
o...
Não, é que eu já vi estas pessoas todas que eu faço parte da organização, mas isto é muita gente. Isto é muita gente, eu já não... Não, não é, na cara não me parece que seja ele. Deve ser...
Não, mas
é o...
Conrad, eu estava a ver o outro.
Sim, o Guerra, é isso, eu também estava a pensar nesse.
Também podemos fazer ao contrário.
Acho que é.
Sim.
Eu por mim
Ou então
tratava.
Sim. Ou então podemos fazer, pôr no Google Translate a ver como é que ele diz em holandês.
É que realmente é chato a pessoa dizer mal. Nem se esforçar, não é? Porque a gente não sabe mesmo o que é. Se calhar eu vou lá para
É ele?
Eu acho que é. A cara dele parece. A cara dele parece.
É, sim. Bem, ele é um bocadinho mais velho que isso, mas eu sempre
Mas
já vi.
parece.
Mais vezes, acho que sim. Perguntem-se antes, não é?
Pá, eu acho que sim, vou lá.
Yeah, right. E o Conrad é aquele que estava ali?
Portanto, o Nathan, portanto, falta
Eu não sei.
Jessica Day
E o
o... Mas
será
esse
que
senhor eu já ouvi antes, no outro
é
dia.
aquele ali?
Acho que não é. Achava que não era. Mas pronto. Eu já ouvi, pelo menos porque eu acho. Já estamos às 10h30. Acho que sim. Senão começamos já a atrasar. Acho que sim. São 10h30. Acho que
Good morning, you all. Welcome for this session on diagnosing myositis. My name is Rafael Roque. I'm a neuropathologist here in Lisbon, and I'm co-sharing with my colleague Ana Teresa Melo, rheumatologist here from Lisbon too. So the first hour will be 2 lectures on the diagnosing myositis, and then we'll have 3 oral presentations. Interestingly, all of them are about IBM. So we are going to start.
And I would like to welcome our first speaker. Conrad Weil is a neuromuscular clinician and researcher at Washington University School of Medicine in St. Louis. He is interested in diagnosis of patients with hereditary and acquired myopathies.
Great, thank you for the introduction and the invite. I changed the title a little bit. I changed it to genetic advances rather than genomic advances. And I realize we're talking about diagnosis of myositis, but I'm really going to be talking about how we don't miss diagnosis of hereditary diseases. And I think that we're at an inflection point. I remember whenever I was training and I'd see a patient with what I thought was a hereditary disease, and I'd be like, I don't want to miss an inflammatory muscle disease. I don't want to miss a myositis. But I think we may be flipping it a little bit, where we see somebody who has an inflammatory disease, and I think we also don't want to miss that potentially we could be seeing something genetic, because there are therapeutic interventions that can be happening for hereditary diseases. So briefly, I'm going to talk about hereditary myopathies and kind of the differential diagnosis of myositis, some genetic association studies, rare variant studies, and then finally, genetic testing in clinical practice. So just to kind of ground us a little bit in patients that we might see with late adolescence and adulthood presentations of hereditary myopathies, I think it's important to kind of categorize them. And one is in categories of dystrophinopathies. These are patients with Duchenne muscular dystrophy or Becker. And I want to point out that these are often due to deletions in the DMD gene, and that's going to be important as we start to understand our genetic analyses. Facioscapulohumeral dystrophy presents often late in late adolescence or early adulthood, has a distinctive presentation of facial weakness as well as scapular winging. Myotonic dystrophy, and so these are now, again, the most common. So if you were to think of the most common, it would be a dystrophinopathy. Then we would move to FSHD and myotonic dystrophy, so DM1 and DM2, DM1 being myotonic dystrophy type 1. And it's associated with an expansion in a 5' UTR region. And then finally, limb-girdle muscular dystrophies can be both recessive and dominant, and those have a vast array of genetic causes, greater than 35 different subtypes. And finally, hereditary inclusion body myopathies and myofibrillar myopathies. And these are kind of how I think of as some of the more common things that we might miss in a patient that we think has a myositis. Again, I want to point out the 3 most common causes: a dystrophinopathy, meaning Becker or DMD, FSHD, and myotonic dystrophy. These all are due to really large arrangements that are not picked up on routine genetic testing. And so whenever you have a patient or whenever you give a panel-based genetic testing, you may not find these variant mutations, in particular FSHD and myotonic dystrophy. And so you need to intentionally be thinking of those, and we're going to go through that in a minute. This is a paper by Jens. It's really just a nice way of kind of opening the conversation, which is basically, you know, we create criteria. So the 2017 Euler criteria was created really to distinguish IIM from mimicking conditions with high sensitivity and specificity, and also to put things into subgroups. And so what Jens did is they looked and they took a cohort of 100 patients with hereditary myopathies, all late onset hereditary myopathies. And they said, how do they perform in the Euler criteria? And what you can see here are graphs of each of the different subtypes, so DM1, DM2, FSHD, Becker, and limb girdles. And you can see that in many cases, it would fulfill the Euler criteria. And I don't think as clinicians, any of us are using the Euler criteria as the gold standard, but I do think it is helpful in just understanding that there is an overlap. And what was nice about this is it kind of said, well, where was the overlap? How did we miss kind of, you know, the diagnostic confusion? Where does that lie? And so phenotypically, there's clearly an overlap of hereditary myopathies in the pattern of weakness with both proximal and lower extremity prominent weakness. But the other feature was that muscle biopsies, which are part of the Euler criteria, 18% of hereditary myopathies have evidence of endomysial and or paramysial inflammation. And so a biopsy can be helpful, but does not necessarily mean that this is an inflammatory muscle disease. And in some cases, they have rim vacuoles, which led to the diagnosis of inclusion body myositis. Equally important, although not in this in Yen's paper, would be features that might distinguish hereditary from acquired. And whenever I think of the distinction, facial weakness, I'm not sure is helpful, but there is a characteristic ptosis or drooping of the eyelids, which is what you see in myotonic dystrophy and FSHD that I don't feel like I've seen in patients with IBM or other facial weakness of that stereotyped region. Electrodiagnostic features would help you find myotonia and denervation. And so if that's not common in your practice, I think it can be helpful in making sure you don't miss a patient with myotonic dystrophy. And finally, of course, family history, which can be very helpful. So I want to pause at that point. So really just bringing you into the idea that it can be complex and that hereditary patients can look like acquired myositis patients, and that we can't just rely on biopsy or phenotype. And we're going to come back to how we can use that in clinical care. But I thought it was important to kind of go over what genetic studies have been done in myositis studies. And so genetic association studies have been clearly done. There are multiple papers, and I do not want to think that these HLA regions of association that I put up here are the only ones that have been reported. These are helping to understand susceptibility loci. These can vary from ethnicity. They're not something that I think we can use for diagnosis. They may help stratify in clinical studies. They may help us think about who might respond to a certain therapy, but I'm not quite sure they're helping much with diagnosis. With that being said, I think they're very helpful. And what is being done more commonly now, and we're actually going to hear a study on IBM, but about a GWAS study, so a genome-wide association study. And so a genome-wide association study actually takes a cohort of patients and looks by doing a genotype SNP array, and then looks to see what variants are overrepresented in genomic regions that can help be overly seen in a population of patients. And so what you see on the bottom there is what we call a Manhattan plot, and anything above that dotted line predicts that it might be overrepresented in that group. And I like this graph here on the side because what you can see is in a GWAS study, what we're looking at are common variants with potentially large effects, or common variants with small effects. And so, so what we're not looking for are what's in that upper right corner, which is highly penetrant mutations. You're not going to find that in a GWAS study. That's what we find in a Mendelian inherited disorder where it's highly penetrant. But the other thing we're not going to find are rare variants with small effects. And we're going to talk about rare variant analysis in just a second. So really the GWASs are helping us kind of look at common variants with large effects and common variants with small effects. And so this is an example of a GWAS study that really is an amalgam of many different studies from the Myositis Genetics Consortium. And this is kind of what they identified. And so they were able to take, about 1,000 patients that they ended up putting through kind of their pipeline. And then if you look at the Manhattan plot there, what you can see is the most striking genetic association is in HLA, is in HLA region, so over a 20-fold association. So that's a common variant that has a high, effect. And then they can find other ones which are much less, have much less effect. And so, these are helpful. These aren't used in diagnosis. And so you might say, well, why are they helpful? Well, I mean, the HLA one's important because it tells us that this is an autoimmune disease. But the other reason is that we can start looking at pathways and we can really start to understand kind of therapeutic targets and really use this to help us understand kind of the pathophysiology. And so that's where I think that it can be quite helpful. All of these hits that are identified, they can be just associations, and are there ways of actually validating them? And so can they help us identify therapeutic targets? Can they help create model systems? And so I think they're quite helpful in those, but not really, in my mind, helping with diagnosis. So what about this idea of rare variant analysis? And so this is the idea that there are variants that are quite rare, that are found in only 0.001% of patients out there, not common variants like an HLA subtype. And so this was a study that we did, as well as those with Pedro Machado's group. And what you can see is that what we did is we did sequencing of patients. In this case, we used inclusion body myositis. We sequenced patients with inclusion body myositis. We looked, in our case, we used targeted genes. In their case, they looked at an exome. And they were actually able to identify rare variants in candidate genes that they thought would be important for disease. And so in this case, both of us found about 4% of our patients with presumed sporadic inclusion body myositis had rare variants in one of two genes. One was sequestosome 1, and the other was a gene called VCP. Sequestosome 1 and VCP are important in autophagic degradation, and they are found in a form of hereditary inclusion body myopathy known as multisystem proteinopathy, or classically called IBM PFD. And how do we know that these are enriched? So what you can see is, if we take VCP at the bottom, these were the variants that were identified. If you look in a population database of 60,000 patients, you can actually see that there were 600 patients with presumed no disease at all that were identified in NOMAD. So that frequency would be 0.075%, whereas in our populations of IBM, it's enriched 3.8%. And the same is true for sequestosome, where the frequency is much, much lower than what we found in our cohort. And so that's kind of how you do rare variant analysis and can show kind of enrichment. Now, many people would say, and I've heard this many times, that I just got the diagnosis wrong on these patients. And that's a fine statement. I'm okay with that. But I do think that we are showing actual enrichment because of these variants. Many of them are actually common, not common, but they're overrepresented. They're not zero in population databases. So can we do another approach in myositis? So this is another example of how we did variant analysis. And so what we did is we took patients with inclusion body myositis, and we did exome sequencing on them, where we could identify every coding variant in these patients. And we made some assumptions. And so whenever you do large data sets, you sometimes have to make some assumptions. And so we made an assumption that we were going to identify rare variants, meaning a frequency of less than 0.1 in the population, that these variants in the patients had to be a missense mutation or result in a loss of protein function. And so whenever we did that, this is what I just said, it also had to be shared in more than one individual in our cohort. So the mutation in the gene had to be found in another patient in the same gene. It had to be statistically overrepresented in controls. We used actually an ALS cohort control, but we validated it now later. And so if we use this criteria right here, all of us have about 500 rare variants that are coding in genes. So we're looking at patients and seeing if we can overlap these. And so, can we do this in IBM? And I will say, doing that simple approach that I just said, we found nothing. Okay, we found nothing that correlated in those 62 patients. Had we had thousands of patients, we might have been able to do that. And so that's one limitation we have, I think, in myositis. I think our cohorts are growing and growing, but we're not Alzheimer's disease. And so these rare variant analysis can be challenging. And so what did we do? We said, well, let's use other omic approaches and see if we can enrich this data set. And so we actually worked with Rudy Clay and Matthias Vogard, who had done proteomics on what were present in the rimmed vacuoles. So these are proteins that were enriched in the rimmed vacuoles in inclusion body myositis. And what we did is if we took, I'm sorry, that's yellow and white. That doesn't go very well. But what this is showing that there were about 213 proteins that were enriched, that were in the patients, or sorry, in the proteomic data set that were at the vacuole. So the vacuole had an enrichment of about 213 different proteins. We then overlaid that with our data set of rare variants, and we found 17 patients that had that. And then we overlaid, 17 genes, 17 genes. And then we overlaid that with how many would be in our cohort. And so there were still, of those 17, 16 of them would have been enriched in another data set that was not relevant to the disease. And so that left us with one variant that was uniquely enriched in our patient data set as well as in the proteomic data set. And so that was in a gene called FICO1. And the enrichment was about 0.029, so we were quite excited that we actually had enrichment of a missense variant in this gene in 7 of our 62 patients. And so, again, low effect, a low effect. This is not a diagnostic test. This did not track with the patients. There was no hereditary component to these patients. So what is this actually telling us? And so this is telling us perhaps that we need to be thinking of targets. And so FICO1 ends up being that protein right there. It's important for trafficking an autophagosome. An autophagosome is the degradative component of the system of autophagy. And what we can see here is that mutations in that region may be important for affecting autophagic degradation. And so this then fits with our other rare variant analysis in IBM, where sequestosome and VCP and FICO1, all of them may be synergizing kind of at that region of autophagic maturation of an autophagosome. And in fact, if we look at the rimmed vacuoles, we actually see accumulation of FICO1 and autophagic proteins. And so that's kind of how rare variant analysis can be helpful. I would say we need to be thinking about rare variant analysis for pathway discovery. And as we do more and more proteomic data sets, more and more omic data sets, and start overlaying those and phenotyping patients better and understanding enrichment in different populations, I think that we will actually start to really identify therapeutic pathways in many different myositis disorders. And so I think that's an exciting thing to think about. And of course, there needs to be functional validation for these. Otherwise, they're just associations. There's really no determination whether they're actually causative. So that would be the kind of the next step. So I'm going to switch gears again and really talk about kind of genetic testing in clinical practice. And so genetic testing in clinical practice, there's a couple of ways that we can do that. This is a busy slide, and it really goes through. You can do individual gene testing. So if you know somebody has a genetic disorder or has a family history, you can do an individual test. Typically not often utilized. Panel-based genetic testing is probably the most common thing that we do. And that really is of known muscular dystrophy genes or likely genes. Different panels have different testing panels. So often a patient will say, I've had genetic testing. Well, you don't really know what kind of genetic testing they had. Certain genes won't show up. Myotonic dystrophy won't show up. FSHD won't show up. You have to intentionally test for those. Whole exome sequencing, as I just described, finds everything. Whole genome sequencing finds even more, but is somewhat cumbersome to utilize. And more RNA sequencing for muscle can be used, and there actually are companies that are now doing RNA-seq analysis from skeletal muscle. You know, there are barriers to genetic testing. Payers don't want to pay for it. I would say patient knowledge. I would say physician knowledge. And really, this is really striking. This is the current clinical criteria for hereditary muscle disease by the AANEM, which is a neurology kind of governing body. This is from 10 years ago, hasn't been updated, but it actually says that you should do your exam, see the patient, and then kind of guess what do you think their diagnosis is, and then just test for that single gene. That's what the recommendations are. The expert recommendation is the last one, which is, you know, if you ask me, I think you should do a panel. So the expert would say do a panel, but the guidelines actually suggest that you should actually look for individuals. So I think that's quite challenging. So what is, you know, exome sequencing or next generation sequencing, and how has that kind of transformed the genetic testing landscape in clinical care? And so, you know, it used to be that you would have to do something called Sanger sequencing, which is you would sequence here and you'd kind of move slowly across and then you'd stop and you have to do it again. With next generation sequencing, what you can do is you take the DNA, you then fragment the DNA. So you basically shear the DNA into small pieces and then you amplify each exon. And whenever you do that, you then sequence everything. So now you sequence all these little fragments at once, and so you do this kind of massive parallel sequencing. And then it's where the analysis happens, where you then overlay and say, okay, there's this region, this patient, you know, this sequence was in this region, it overlaps with this sequence here, you start to overlap them. But what you can see is whenever you do that overlapping, you're likely to miss deletions, you're likely to miss duplications, you're likely to miss some chromosomal rearrangements. And so next-gen sequencing is not perfect. And so I think understanding where its imperfections are can help you understand sometimes the genetic testing result. And so this would now be a patient with kind of proximal weakness. And this is what we get whenever we do genetic testing on somebody. And so it can be quite confusing and overwhelming for clinicians and patients. So now you're looking at this, you've got several genes, and you've got these things called variants of unknown significance, and it can be quite challenging. You also have up at top, it says likely pathogenic. So if we look, the gene is AMPD1. It should be inherited in an autosomal recessive manner. And then if we look at the protein change, it's a glutamine 45 terminating. And then it says likely pathogenic. All of us could have figured that one out. You stop the protein being made at amino acid 45, it terminates, it's not going to make a functional protein. That's where the likely pathogenic came from. Not because the company knows that there's another patient out there that has a pathogenic mutation. And so they're just interpreting, they're just classifying that variant. And I think that's a really important aspect that I want to put on everybody, is that genetic testing labs are just classifying the variant. We are interpreting the variant. It's the same as the chest x-ray from the radiologist telling you the patient has pneumonia, and you see the patient, they're breathing fine, they're afebrile, you know what they have, they're just helping you with it. And so I just want to make that a point, because I think more and more we're getting to an era where someone comes in and says, I have this because of my genetic test result. And so that's just one thing I want to say. So how do we get all these variants that occur? And so these variants occur because the reference genome was made by one individual, and so everything's compared to that one individual. And so what that means is that many ethnicities are underrepresented in these databases. And so as we start to build these data sets, we need to actually have more and more patients from underserved areas, from ethnicities that we don't typically capture, because whenever a genetic test result comes in, if you are from sub-Saharan Africa or have an ancestry there and you aren't represented in the databases, you're going to have tons of variants of unknown significance. So the burden is actually placed on a subset of patients that are not Western European or Caucasian. This is the first example of how much genetic variation we have. And we've known about common genetic variation. We've done crime scene analysis. We've done paternity testing. Those are common variants. But the vast amount of rare variants was actually identified only about a decade ago, in which all of us have about 3 to 4 million variants, of which 30 to 1,000 are rare and some are unique to you. And this graph actually shows an individual with their own novel mutations. Of these, 250 are loss of function. We all have loss of function on 2 different alleles. So we are all knockouts of about 50 to 100 variants. And this has been proven in larger data sets where, as you can see here, we are homozygous protein-terminating variants. Each of us has approximately 30 loss of function variants. So we're all knockouts, yet we all have disease. And so how does this affect kind of clinical care? Well, this was a really important effort by the Muscular Dystrophy Association where they actually brought in, they allowed kind of point-of-care genetic testing. And point-of-care genetic testing allowed us to do testing in the clinic. And normally what I would do is I'd say to a patient, let me find out if insurance will cover it and all of that. And so they actually allowed us to do that. And this is the data that they show you, which is that they were able to show you the landscape of hereditary muscle disease in our MDA clinics. This is actually the data, which is they only solved about 24%. And really, they left 76% with non-diagnosis, of which half had one of these variants of unknown significance in the gene. And this is borne out to be true in other studies here, where even if you sequence more, so you might say, well, 35 genes wasn't enough. We should just sequence more genes. Let's just sequence more genes. And so in this study, they sequenced 169, and then they added on another 260 more genes to the panel. And whenever they did that, they only incrementally, 2%, increased the diagnostic rate. So just a teeny, teeny amount by adding more genes. But what they did do is whenever they did that, they actually just added more variants of unknown significance. So now more diagnostic confusion for the patient. And so I think that's really an important thing to do whenever we do these genetic testing. And I'm going to skip through this, but I also want to talk a little bit about free genetic testing programs. So at least in the US, there are many what they call free, I would call them sponsored genetic testing. They are typically sponsored by a company. They have gene panels. The goal of those companies are to identify patients with a disease that company is interested in. I found it just amazing that Sanofi Genzyme has been going to rheumatology clinics and wanting people to look for patients because they want to identify patients with Pompe's disease. And so, it's just important to understand that. And I would say that the sponsored genetic testing programs, for us to do it is easy, but the burden of pre-test genetic testing is on us then. And so you really need to be able to explain to the patient kind of, what their genetic, the implications are. And so this is an example where they do a broad test. They don't actually, they're choosing genes just broadly. They're not choosing genes necessarily that are the ones that should be looked at, mainly so that they can identify a GAA mutation. I want to go through just some really quick cases. The goal of these cases is really just to understand the genetics. So I'm going to run through them really quick because I'm short on time. But this is a patient who had kind of progressive weakness. He didn't have a family history. He had proximal weakness. All these patients I'm going to talk about have kind of a limb girdle pattern of weakness. He had what looked like a rim vacuolar myopathy, an inclusion body myopathy. I did something that I would not recommend anyone do. I was young, I was arrogant, and I did exome sequencing on the patient. I was like, I'm going to do an exome sequence on this patient. I did the exome. We actually found the cause. So the cause was a mutation in BAG3. But what I didn't realize when I did the exome sequencing is this 45-year-old man had a BRCA mutation. And it was on me, a neurologist, to now explain to this patient the risk of his BRCA1 mutation for a male of 45. And so I just think it's important that we understand what we're looking for and the implications of the genetic testing that we do, and why sometimes panel-based sequencing is better, because we're actually going to look for things that we can actually understand. And so that, my lesson learned was that pre-test genetic counseling is essential. I didn't even explain to him we could find that. I didn't even think about the implications for his family. Genetic counseling is something that we have in our clinic, but it's something you can engage in. This case is another patient, and I'm gonna blaze through it really quickly. Again, it's a 49-year-old man with progressive weakness. He had scapular winging, he had finger flexor weakness, he looked like inclusion body myositis. He had his genetic testing had been done and it was negative, so came to me, said genetic testing was done. He had FSHD genetic testing, which was reasonable because of the scapular winging. He had an NTC51A that was positive. And his biopsy showed inflammation, and this was his biopsy. And it was actually the pathologist actually read one area of focal invasion. I'm not convinced I saw it, but I was trusting the pathologist. The patient came back to me and said, my brother developed ALS. And so this made me think that perhaps he could have one of these myopathies that had overlap due to mutations in VCP. But I was convinced he had genetic testing, and I was convinced that VCP was on the limb-girdle panel. But when I actually looked, and so you got to go look at your genetic test that you did, and you got to look at all the genes that were tested, VCP hadn't been added to this panel yet. And so I went back and I tested him for VCP, and he had that mutation. And so the important part here is that not all gene panels are equal, and a comprehensive panel may not be comprehensive. And even though the patient had prior genetic testing, even if it's in the recent past, kind of to understand that. In this last case, I just want to get through really quickly, basically to say that he had previous genetic testing on a sequencing panel, but what he didn't have was sequencing of his entire DMD gene. And so he'd been going along for years with a diagnosis of myositis, and he actually had Becker. And really it took understanding kind of his history of what he had. And so I'm gonna end there, and thank you. So, thank you.
Thank you so much for your kind presentation. We only have time for 1 or 2 quick questions if someone wants to do a question, because we are a little bit of time.
Looks like there's someone over there.
Is that working? Thanks, Chris. Just a quick question. As many of these genes you mentioned can cause ALS and vacuolar myopathy and so on and so forth, have you looked at the 16 proteins that are shared with ALS in your proteomic study to see if those provide any insight?
Yeah, so we have looked to see if there are an enrichment of kind of genes in other degenerative disorders, ALS and things like that. And we haven't seen an enrichment in those types of genes in sporadic IBM. That's not the case for hereditary causes of inclusion body myopathy, but not for sporadic IBM. Looks like there's a question over here.
Do we have a microphone? Or you can come here, please.
Olivera Chasar-Borata, neuropathologist from Uppsala. Thank you for a nice lecture and beautiful histopathological pictures. And overview over genetic analysis in neuromuscular disorders. There is one analysis that I missed actually on your list, and it is genome-wide methylation analysis. Do you think that methylation profiling has a future role in diagnosing of neuromuscular disorders? Methylation analysis, genome-wide methylation profiling, it has contributed a lot to diagnosis of brain tumors and so. Do you think that it has future in neuromuscular diagnostics too?
Yeah, so you're asking about methylation?
Yeah.
Yeah, absolutely. So I think there are, we're only looking for what we see, and we're only looking for what we looked for. So yeah, there absolutely are other approaches that would be worthwhile. So looking at methylation status, which can lead to epigenetic changes that can reduce gene expression, increase gene expression, all of that is ripe and ready. Yeah, absolutely. I think that's a very reasonable thing.
Thank you.
Thank you. Thank you.
Okay.
It's with great It's for me a great honor to introduce our next speaker. Ichizo Nishino is Director of the Department of Neuromuscular Research at the National Center of Neurology and Psychiatry in Tokyo, Japan. His laboratory serves as a nationwide referral center for muscle disease and reviews more than 1,000 muscle pathology cases annually. Thank you.
Thank you very much for your kind introduction. So today I'm going to talk about muscle biopsy and histopathology, but I'm going to mainly talk about pathology. So this is my disclosure. So there are 4 well-established myositis subtypes, as you all know, and immune-mediated necrotizing myopathy, dermatomyositis, antisynthetase syndrome, and inclusion body myositis, but there are others. But today, for the sake of time, I will focus on those well-established 4 subtypes. So immune-mediated necrotizing myopathy. So the pathological features of immune-mediated necrotizing myopathy is, of course, the most important feature is necrotic and regenerating, the presence of necrotic and regenerating fibers, as its name suggests. And although this condition subtype is classified as myositis, we rarely see the remarkable lymphocyte infiltration. And there is minimal or mild MHC class I expression, which is a non-specific marker of immune activation. And this condition is believed to be associated with or due to complement activation. And you see, we'll see our sarcolemmal C5b9 or membrane attack complex depletion, MAC deposition onto the sarcolemma, which is believed to cause myofiber necrosis. And more recently, an additional pathological feature was reported, which is granular sarcoplasmic P62 staining, as I mentioned later. So this is one of the representative case of immune-mediated necrotizing myopathy. Of course, you see necrotic fibers. And once myofibers are necrotized, of course, there will be regeneration. So not surprisingly, we see number of regenerating fibers. But importantly, again, there is no lymphocyte infiltration. Even if we see it, typically very, very little or minimal. And the MHC class I is usually very minimal or mild or even absent. And again, importantly, this condition is believed to be due to complement activation. And so you can see that the membrane attack complex, or MAC, is deposited onto the sarcolemma, which is believed to lead to myofiber necrosis. However, in our daily practice, this finding can be seen in a wide variety of myositis subtypes. For example, yes, we see in IMNM, yes, we see more often we see it, but even in other conditions like antisynthetase syndrome and dermatomyositis, we see them. So for this reason, of course, it depends on how you choose the cohort, but in this study, the sensitivity, specificity is like 60%, and positive predictive value is very low, like 20%, and negative predictive value is very high, 90%. So practically, even if you see it, you cannot be very confident that whether this is your case is IMNM or not. Then what about this one? So recently, Werner Stenzel's group reported granular sarcoplasmic P62 staining as an additional feature of IMNM, or immune-mediated necrotizing myopathy. And they mentioned that they reported that this phenomenon is associated with chaperone-assisted selective autophagy, CASA. But the question here, key question for us, is whether this is specific or not. So we actually, every year we have, medical students, a laboratory, and they stay 2 months and they do some, research work in a small project. So I gave them a very small project So I asked them to investigate the specificity of this finding. Yeah, so they checked of course, they checked the number of cases, like 150 IMNM cases and 255 non-IMNM cases, including actually muscular dystrophy and other hereditary muscle diseases. And the method-wise, I asked them to scan all the immunohistochemistry slides and measure the optical density. And of course, the staining condition may be different from slice to slice, so it has to be corrected by, for each slide. So I asked them to correct the optical density by the optical densities in the perimygium, so blue one. So then this corrected optical density was used to compare between IMNM and non-IMNM cases. Very interestingly, so you see the high, can you see? I don't see maybe, sorry. So as you can see, those with high, optical density, high OD, yes, thank you very much. So this one is seen only in IMNM, but not in non-IMNM cases. So looks very promising. So if you set the cutoff, of course, we did some calculation, but if you set the cutoff of 0.3 OD level, actually specificity is 100%. Although sensitivity is not very high, but specificity is 100%. So if you set the criteria that one positive fiber, the optical density higher than 0.3, and if you find even just one fiber at 20 power field, then you can make a diagnosis. Yes. So yes. So you can make a diagnosis like this. So probably of course, we need further investigation, but probably this granular sarcoplasmic P62 staining is much more specific and more useful for the diagnosis of IMNM. So I hope those young people will write a paper. One of the guy is actually 19-year-old. So I really want the paper from that, with that young guy as a first author. So let's wait. Another important issue is the presence of pediatric cases of IMNM. So we have ever reported the series of nine cases. And as you can see, the patient one, the onset is 10 months old. So there are very, very young onset cases, so you have to be very careful about it. And not surprisingly, half of them are actually misdiagnosed, at least clinically, initially misdiagnosed with muscular dystrophy. And of course, it's not so surprising if you see a patient like a 2 or 3 years old, kid with progressive muscle weakness, very high CK. Of course, you will think about a possibility of muscular dystrophy. And even on histochemistry or even on muscle pathology, actually, it looks really dystrophic. This case was not included in that series, but this case, this muscle pathology, muscle biopsy was obtained from a 16-year-old girl who had 8-year history of muscle weakness, and she was clinically diagnosed with limb-girdle muscular dystrophy, and she didn't receive muscle biopsy until age 16, so which really delayed the diagnosis. So I think there is always a discussion whether muscle biopsy is still necessary or not. Yes, but of course I'm biased, but you still need it. And once you think it may be necessary, I think you should do it as soon as possible to preventing delaying the diagnosis. Okay, so let me go into the next one, the dermatomyositis. Probably the best known and the pathological hallmark of dermatomyositis is perivascular atrophy. This has been known for many, many years. But importantly, I don't go into the detail today, but importantly, perivascular atrophy is not just atrophic fibers at the periphery of the fascicles. They often accompany punched out vacuoles, and often COX activity is diffusely decreased in the region. So when we judge perivascular atrophy, we don't just look at the size of myofibers at the periphery of the fascicles, but we also check other features as well. And also sometimes we see microinfarction in dermatomyositis cases, and the microinfarction is a cluster of necrotic fibers without phagocytosis. They're very interesting. Nobody knows why. So typically, whenever you have necrosis, necrotic fiber, typically macrophages can come in and they clean up all the destroyed cytoplasm. But for some reason, there's no macrophage invasion. So there must be some mechanism which has to be dealt with. And then interesting, again, I don't go into the detail, but in that region, very interesting fibers are seen, which we call CNPR fibers. This means centrally necrotic, peripherally regenerating fibers. So in myofiber, you see necrosis within single myofiber, you see necrosis in the center, but at the peripheral part, you see regeneration. I believe that this is related to lack of macrophage invasion. So this weird phenomenon is seen in microinfarction. Okay, then on immunohistochemistry, we see capillary MAC deposition, which has long believed to be diagnostic of dermatomyositis. And MHC class 1, of course, is expressed, but with perivascular enhancement. But most important finding for us is sarcolemmal MXA expression, which we believe the most important and most useful diagnostic marker of dermatomyositis. And if you do EM, yes, you see tubular reticular inclusions or TRIs in the vessel. But I don't think we do, all the laboratories do EM as a regular analysis. So I'd like to focus on sarcoplasmic MXA expression. So this is a representative image of MXA immunohistochemistry. As you can see, in normal or control muscle, we never see any expression at all. But in dermatomyositis, you see scattered fibers, myofibers express MXA. MXA is the marker of type 1 interferon. So now we think that this is dermatomyositis can be regarded as type 1 interferonopathy. So we previously investigated the sensitivity and specificity of this finding as compared with more classical findings, perivascular atrophy and capillary MAC deposition. So both perivascular atrophy and capillary MAC deposition, not surprisingly, their specificity is very high, more than 90%, especially perivascular atrophy is 98%. So practically speaking, if you see it, you can probably make a diagnosis of dermatomyositis. But the problem is sensitivity. Sensitivity is not that high. So it means that there's high chance that you may miss the diagnosis because they are not always, present in your section. But when it comes to MXA, myofiber or sarcoplasmic MXA expression, the specificity is high as perivascular atrophy, 98%, and sensitivity is much higher than those of other two findings. So we believe that this is the most useful diagnostic marker of dermatomyositis on muscle pathology. For example, this case this muscle biopsy was obtained from 1 year and 9 month old girl who was suspected having dermatomyositis. clinically. But muscle pathology is like this. It really looks normal without perivascular atrophy. Importantly, I'm not selectively showing the normal-looking area. The whole area, entire specimen was like this. So previously, I believe that we would send back reports saying that, okay, muscle pathology looked normal and no diagnosis. But with MXA immune histochemistry, look at this one. Of course, not all fibers are positive, only the fibers at the edge of the specimen, but they are clearly positive. So now we can confidently say that, okay, this case has dermatomyositis. So actually, this improves our diagnostic probability or rate very much after the introduction of MXA immunohistochemistry. Now that, this means that with MXA expression, we can confidently say that particular case has dermatomyositis, regardless of clinical information or clinical features. So this led us to identify cases with dermatomyositis without skin changes. This condition is referred to dermatomyositis sine dermatitis. I'm not sure if my pronunciation is correct, but so sine dermatitis. And interestingly, most of the cases are associated with NXP2 antibodies. So this means that it is well known that CADM, clinical amyopathic dermatomyositis, is highly associated with anti-MDA5 antibodies. This was well known. So, but at the same time, on the other side of this spectrum, now we know that there is dermatomyositis sine dermatitis, which is associated with anti-NXP2 antibodies. Okay, so there are 5 antibodies, autoantibodies specifically associated with dermatomyositis that are anti-T1 gamma, MDA5, MI2, NXP2, and SAE. And I think everybody knows that there are clinical features specifically associated with the positive autoantibodies. For example, T1 gamma is highly associated with malignancy. MDA5, as I already discussed, associated with CADM. NX2, sine dermatitis, or juvenile dermatomyositis, something like this. But for us, the key question is whether there is any difference, in muscle pathology depending on the positive autoantibodies. So I don't think I have enough time to go into the detail, but the answer was yes. So, my former postdoc, Jantima Tambun, who was from Thailand, she actually worked really day and night and investigated, reviewed all the slides. And she identified, for example, T1 gamma positive dermatomyositis have more frequently show punched up vacuoles, capillary MAC deposition, and MHC class 1 or HLA-BC was more predominantly expressed in perivascular fibers. MI2 positive cases are associated with perivascular necrotic fibers at the perivascular region, and also often with alkaline phosphatase expression in perimysium, and MXA positive fibers at the periphery of the fascicle. But interestingly, capillary MAC deposition is rare in this condition. MDA5 cases, of course, MDA5 positive cases, they actually typically don't show much muscle weakness. So our cohort, they received muscle biopsies, so our cohort may be biased. But interestingly, we don't see, for example, perivascular atrophy, no capillary MAC deposition, very, very rare. And there are, of course, MXA positive fibers, but the distribution is very different from other subtypes. Typically, they are often distributed in perivascular region, but in an MDA5 positive cases, they are distributed in a scattered manner. And NXP2 positive, yes, as I mentioned, there is microinfarction is really highly associated with NXV2, and capillary MAC deposition is very high. And so the answer is yes, and then there is some difference. I can't tell you all the details, but this means that pathology is different. But this means that probably, although they are all dermatomyositis, but probably there is subtype specific mechanism, different mechanism, should be there. Okay, the next one is antisynthetase syndrome, or ASyS. Okay, so there are two lines of pathological features. One is perivascular necrosis, and the other one is perimysial pathology, which are actually, I already mentioned. This is really similar to MI2 positive dermatomyositis for some reason. But anyway, this is seen in this condition. And MHC class 1 is, of course, always expressed in any type of myositis. But importantly, MHC class 2, I believe this is more useful for the diagnosis of myositis, is expressed in the perivascular fibers. Okay, first, perivascular necrosis. You see necrotic fibers and regenerating fibers at the periphery of the fascicles. This is typical perivascular necrosis. By the way, this is a anti-Jo1 positive case. And in the same case, you see connective tissue is fragmented in the perimysium, and alkaline phosphatase is expressed in perimysium for some reason. Actually, this condition was reported really many years ago, in 1970s, actually. But in that paper, they said that around 20% of cases of myositis show perimysial alkaline phosphatase expression, but they don't know, of course, what it means. But it looks like this is more frequently seen in antisynthetase syndrome. But here, I'd like to emphasize the importance of MHC class 2 expression. Interestingly, MHC class 2 is often enhanced at the perivascular fibers. And this is, okay, graph is a little bit difficult to see. You see, this is all antisynthetase syndrome, and this is subclassified by autoantibodies. And IBM, Dermatomyositis, IMNM. So actually, for example, IMNM, of course, there are rare cases with MXA class 2 expression, but it's rare. So typically, there's no MHC class 2 expression. Dermatomyositis, 15%. Again, typically, there is no MHC class 2 expression. So typically, we see only two conditions in which we see MHC class 2 expression. One is IBM, inclusion body myositis, which I will discuss in the next. But the other one is antisynthetase syndrome. And when you look at this one, so actually Jan Tima classified the pattern of the distribution of MHC class 2 positive fibers into 5 types. So basically, blue one indicates diffuse pattern, and the pink one indicates perivascular pattern. So you see, most of the positive cases show pink color. So it means that antisynthetase syndrome cases, most of them show perivascular enhancement on MHC class 2 immunohistochemistry. In contrast, IBM actually cases, almost all cases show MHC class 2 positive fibers, but look at them, most of them are blue. So it means that no perivascular enhancement, diffuse or scattered, or manner. So this actually helps making the diagnosis. Yes, and they also, another question is whether there is any difference in terms of positive autoantibodies, even for antisynthetase syndrome. The answer is yes, but again, I don't go into the detail today. Okay, so again, this is the same slide. One day, when I was giving a lecture in Japan, and I showed this slide, and some guy, a rheumatologist, said that, look at this one. Perivascular fibers are smaller in size. So this must be perivascular atrophy. And I think many of you now, still, I think, I'm sorry to say, but still think that myositis with skin changes are dermatomyositis for you guys, right? So antisynthetase syndrome often shows skin changes, or clinically often diagnosed as dermatomyositis. So that guy said, see, even on muscle pathology, this is perivascular atrophy. This must be dermatomyositis. That's what he said. So how can I say no? So if you remember, MXA is very important diagnostic marker, right? This is the picture on the right side is exactly the same case. So you see no expression in the myofiber at all, right? So this clearly indicates that dermatomyositis and antisynthetase syndrome, they may be very similar, clinically or even pathologically may be very similar, but there must be different mechanisms. So they are different diseases. Finally, inclusion body myositis. I think I have 5 more minutes or 4 minutes. So there are 3 lines of pathology, pathological changes. One is T-cell pathology. Number 2, rim vacuolar pathology. And number 3, mitochondrial pathology. And finally, I'd like to discuss the importance of MHC expression. T-cell pathology, I think all of you know that cytotoxic T-cells surround and even invade into non-necrotic fibers. This is really the pathological diagnostic hallmark of IBM and also polymyositis. And Steven Greenberg reported that those CD8 or cytotoxic T cells actually express KLRG1, which is a marker of highly differentiated, memory effector memory T cells. Then rim vacuolar pathology, of course, as its name suggests, we see rim vacuoles. And in addition to that, we see P62 and TDP43 cytoplasmic aggregates, which are believed to be the upstream mechanism to lead to produce or to form rim vacuoles. And another important feature is because the TDP43 is normally localized in the nucleus, and they have a role regulating transcription. But look at this one, yes. So in those fibers, in those myonuclei, you see TDP43 positivity. But look at this particular myofiber. This myofiber has aggregate, TDP43 aggregate, and look at those nuclei, myonuclei in this particular fiber. Nuclear staining is absent. So this clearly shows that normally, the TDP43 is supposed to be present in the nucleus are actually depleted from the nucleus. And because they all go to the aggregates. Actually, this causes, this was reported by Tom Lloyd's group, this causes, of course, abnormality in the splicing. So this results in cryptic exon inclusion, yes. So you can see. And finally, another one is mitochondrial pathology. Of course, in elderly people, we sometimes see ragged red fibers and COX-negative fibers. But many people believe that the frequency of this type of abnormality is more frequent. More frequent than age-matched control. So in this slide, you can see SDH positive fibers. They are ragged red fibers or ragged blue fibers, and they are COX negative. And if you do SDH COX double staining, of course, they are ragged red fibers, COX negative, and this is COX positive ragged red fibers. And so, interestingly, there is one condition reported from, again, Werner's Cancer Group. They reported that, there are conditions in which you see T-cell pathology and mitochondrial pathology, but without rim vacuolar pathology. But even in those cases, as you can see here, there is molecular evidence of cryptic exon inclusion. So this is a molecular evidence of rim vacuolar pathology. So practically speaking, this is probably sort of IBM. This condition is PM mito. And the MHC, of course, MHC class 1 is always expressed in any type of myositis, but MHC class 2 is ubiquitously expressed. This is very important, and this was already a long time ago. It was reported that all IBM cases show MHC class 2 expression, while other conditions are relatively rare. This was also the case in our series. In this case, among 101 cases, only one case was negative. But retrospectively, the condition, the sample was very poor, so probably we shouldn't have included this case. So practically speaking, all cases are positive. But you have to be careful. Look at this one. This is a really the picture at low magnification. This is MH class 1 and the class 2. Of course, in this area, you'll see classical finding. But if you biopsy only this area, you may well miss the diagnosis. So this really indicates the importance of the selection of the biopsy site. So for this, I think the MRI-guided muscle biopsy is required. So you should target the edematous area on muscle MRI and should avoid muscle with a marked fatty infiltration. Okay, this is a summary. I already used up my time. But we have videos, teaching videos for the muscle biopsy technique and also fixation. So please, this is really endorsed by many societies. So please use this one. And this is my acknowledgement. Thank you very much for your attention.
Thank you for this elucidative lecture, and for this interesting data as the pattern of P62 in necrotizing myositis as an instance. So we have a little out of time, so we have a time for a question or comment. If you... Any comments? No.
Actually,
okay.
We are kind of late.
Okay, thank you.
So now we are going to start our selected oral presentations, and we'll start with Geert Bruin from the Netherlands.
Okay, good morning. Today I will present recent work in our lab that is aimed at the detection of an autoantibody marker, anti-CN1A or anti-5-C1A antibodies. And we developed a new technology to detect these antibodies by immunofluorescence. These are my disclosures. So CN1A, anti-CN1A antibodies is one of the many autoantibodies in myositis, as you probably know. A unique feature of CN1A is that it's particularly frequently found in IBM. So from that perspective, it's helpful for the diagnosis of IMNM. What is CN1A? CN1A is an enzyme that is particularly found in the cytosol. It's involved in nucleotide metabolism, and it's particularly highly expressed in muscle tissue. If we look in more detail in muscle tissue, then CN1A levels in type 2 fibers are higher than in type 1 fibers. And if we look at longitudinal sections, then CN1A is particularly enriched close to the Z lines. If we zoom in a little bit, and this is a section of an IBM patient with an internalized nucleus, then in red you see the CN1A staining, which is most highly visible, or most strongly visible, close to the sarcolemma and close to the nuclei. So it's particularly abundant in the perinuclear regions. This is not a unique feature for IBM. This is also seen in muscle sections from other individuals. If you look in the literature, anti-CN1A antibodies were identified in a little bit more than a decade ago. There are quite some studies that address the frequency of anti-CN1A antibodies in IBM. But there's quite some variation. These frequencies vary from 30% up to about 80%. But if we focus on those studies that include at least 50 IBM patients and have a specificity of at least 90%, then you can see that the sensitivity is generally between 30 and 40%. There's one exception, 47%, which is, I think, a good representation of the frequency by which these antibodies can be detected with a variety of assays, particularly ELISA. Well, anti-CN1A is not unique to or specific to IBM. It's also found, but less frequently, in other autoimmune diseases, including myopathies, but also in Sjögren's syndrome and SLE. In this analysis, the detection of anti-CN1A antibodies was performed with 3 synthetic peptides derived from the CN1A protein sequence. And these 3 peptides represent the major linear epitopes that we have identified previously. To better understand what is happening with CN1A in cells and to see whether the behavior in cells might be related to the loss of tolerance against this protein, immunological tolerance against this protein in IBM, we started to express this protein in standard eukaryotic cells like HeLa and Hep2. And if we do this, then we saw that the protein also accumulates particularly in the perinuclear regions, in filamentous structures. Actually, these time-lapse movies show transfected cells that express the GFP CN1A fusion proteins, and the time-lapse concerns the 20 to 40-hour period after transfection. And what you can see is that once the proteins become expressed, there's small filament forms which then in time assemble into these larger filamentous perinuclear structures. We also analyzed non-fusion CN1A, non-GFP fused CN1A protein, and this results in the same distribution in these cells. So Fleur Brinkman in my lab asked the question whether this subcellular behavior could be employed to detect anti-CN1A antibodies in patient sera. So what she did is she took Hep2 cells, transfected them with a construct for the expression of the GFP CN1A fusion protein. Once it's expressed, it leads to these fluorescent, GFP fluorescent perinuclear filamentous structures. These cells are then fixed and incubated with patient sera and imaged by microscopy. Well, the upper part of the figure here shows again the expression of the GFP fusion protein based on the GFP fluorescence. And if we stain the cells also with an anti-CN1A antibody, this is not a patient serum but a reference antibody, then you see that there's full overlap of these signals. And if we analyze the proteins that are expressed by Western blotting, you see that both anti-CN1A antibodies and anti-GFP antibodies stain the same protein. So the fusion protein is apparently intact in these cells. So these cells were stained with patient sera, and in this part of the slide you see that there's the cells are incubated with 4 IBM patient sera, 2 which were known to be negative for anti-CN1A, the upper 2, and the lower 2 which were known to be positive for anti-CN1A antibodies. And you see there's a clear co-localization of the CN1A or the serum signals with the GFP signals. Fleur did the same for a number of sera from other diseases like DM, PM, SLE, and Sjögren, all of which were known to be positive for anti-CMNA antibodies. And again, this immunofluorescence assay allows the detection of these antibodies. So she also compared the immunofluorescence data she obtained for 82 IBM patient sera and various numbers of other patient sera with 2 ELISA assays, one the 3-peptide ELISA assay I already mentioned before, and a full-length recombinant protein ELISA that was developed also in our lab, which is probably quite similar to the commercial anti-CMNA ELISA assay developed by Euroimmune. What you can see is that 38% of the IBM sera were positive in the peptide ELISA as well as in the full-length protein ELISA, but 59% of the patients were positive, the sera were positive in the immunofluorescence assay. And this is not associated with loss of specificity, because the specificity in the immunofluorescence based upon just the disease-controlled samples is at least as high as in the ELISA assays. Well, this graph shows the correlation of the reactivities in the 3 different assays: A, the peptide ELISA; B, the full-length protein ELISA; and C, the immunofluorescence assay. And you see that in most cases, those that are the IBM sera that are positive in the immunofluorescence assay are also positive in one of or both of the other 2 assays. This correlation is less apparent in the other diseases, but you can see also that the number of sera that were positive is also quite low, so should be careful to draw conclusions about this. Fleur also analyzed the titers by the immunofluorescence assay of the anti-CMNA positive sera, both for IBM and for the other diseases, by dilution series. One example is shown on the right, and you see there's quite a heterogeneous distribution of titers in IBM, and the same seems to be true in the other diseases. And there doesn't seem to be major titer differences between IBM and PM, for example. So this brings me to the conclusions. We conclude that the anti-CMNA antibodies can be specifically detected by this immunofluorescence approach. The sensitivity of this method seems to be higher than that of the existing ELISA assays. And what could be the reasons for this? We hypothesized that the conformation or the molecular nature of CN1A expressed in these cells might be different from the recombinant protein that is used in ELISA, for example, by post-translation modifications. We have already identified a couple of methylation sites in CN1A that may play a role, but what we think is more likely explanation is that specific epitopes are formed by the polymerization of CN1A monomers. If we produce CN1A recombinant protein, then we see that it spontaneously assembles into polymers of individual CN1A protein molecules. And well, you can imagine that this leads to the formation of new epitopes that might be relevant for autoimmunity. Well, there's quite some variation in the titers, as I showed, particularly for the IBM patients, but probably also for the other autoimmune patients. There doesn't seem to be real titer differences between the other diseases that are positive for CN1A and IBM. Well, I would like to thank particularly Fleur Brinkman and Isa Verlangen, who performed most of the work on this project. And I would like to thank Imus Bad Rising and Hector Chinay and Janine Lamb for providing patient sera for these analysis. And I thank you for your attention.
Thank you. Thank you for your presentation. Some questions? Yes, please. You have the micro. Yeah.
Hi, thank you for your communication. I have a quick question. So my experience with immunoblotting of anti-CN1A is actually totally different from this. we have a lot of false positives. So I wanted to ask if you have any experience with this. Thank you.
Can you please repeat the question because it's very hard.
So if you have any data or experience with immunoblotting for detecting anti-CN1A, because at our center, at least, the specificity seems to be very low. We have a lot of false positives.
So you are saying that the specificity is very low, or?
Yes, for immunoblotting instead of ELISA or immunofluorescence. I was asking if you had any experience with anti-CN1A using immunoblots, line blots.
With line blots, well, yeah, we don't have any experience with line blots, but I know from people who are routinely using line blots that they also say that the ELISA, the CN1A ELISA, is way more specific and better performing than the line blots. So I think this is related to your experiences.
Thank you.
Well, we have enough.
Thank you for the question. Very quick. Thank you very much for the presentation. My question is regarding the positive patients with Sjögren's disease that you had. Do you think that it might be a link with this positivity and IBM?
You are referring to the Sjögren's syndrome patients, whether there's a link? Yeah, I don't know. I'm not a clinician, so I just received the samples from clinicians which are diagnosed. So I cannot say anything about potential relationships between these.
Thank you.
Okay, thank you. Thank you very much. We move on. Our next presentation is from Australia. Jessica Day is the first author, also about IBM. So
thanks very much for the opportunity to present our work identifying a circulating protein signature that distinguishes IBM from histological polymyositis. These are my disclosures. There are none relevant to this study. So we know that inclusion body myositis involves a complex interplay of inflammatory, immune, proteinopathic and metabolic processes, and conventional blood-based disease markers fail to capture this biological complexity. Around 50% of patients are initially misdiagnosed, and there is thus an unmet need for minimally invasive, biologically grounded biomarkers. So the aim of so most clinical blood-based biomarkers rely on single proteins. However, that obviously only captures one biological axis and may have limited discriminatory performance. And so as such, there's increasing interest in multiple fields in exploring multi-protein signatures, which might capture combined biological effects and interactions between pathways. And perhaps these are more appropriate for a complex disease with multiple biological dimensions like IBM. So the aim of this study is to use multi-omic profiling to identify a circulating proteomic signature of IBM and to cross-reference our findings against tissue-level molecular data. So to support this study, we accessed samples from several biobanks. Our serum samples were retrieved from the South Australian Myositis database, where patients are classified based on histopathology. And so we included patients with inclusion body myositis, as well as what we've defined as histological polymyositis, with lymphocytes surrounding and invading non-necrotic muscle fibres, MHC expression, and the absence of IBM-defining histopathology. The clinical features were reviewed and incongruent cases excluded, and all of our cases met appropriate classification criteria. We also had access to some healthy control blood. And for our tissue validation cohort, we used a separate biobank, which includes biopsy-confirmed myositis samples. All patients met EULAR-ACR classification criteria with appropriate subtyping, and we also included some non-inflamed muscle samples as controls. So we used two blood-based discovery approaches. We first used unbiased approach, mass spec-based proteomics, which in this method, serum proteins are enzymatically digested into peptides and then undergo mass spec for protein identification and quantification. But we also used Olink, an affinity-based proteomic platform, which is an antibody-based targeted approach, where Olink uses paired antibodies which are conjugated to oligonucleotides, and when both antibodies bind the same target, those oligonucleotides come into proximity and undergo extension amplification, generating a DNA barcode, which we can then read with a next-generation sequencing readout. So why did we choose these two approaches? It's because they're inherently complementary. So plasma proteins span an extremely wide dynamic range of concentrations in the blood, from grams per litre for abundant proteins like albumin, but down to even picograms per litre for low abundance cytokines and interleukins. And no single analytical platform can reliably measure proteins across this entire range. And so we chose mass spec and Olink to provide broader coverage of the circulating proteome. So this is our study population. We had 39 IBM patients and 36 histological polymyositis cases. The symptom duration was lower in the polymyositis group, and there was increased representation of extramuscular manifestations as well as non-anti-C5N1A antibodies. And it is possible that some of the polymyositis cases could be classified as overlap myositis or antisynthetase, but for the purposes of this biomarker discovery approach, our comparator group was intentionally defined based on histopathological evidence of lymphocytic inflammation. So that was our comparator. So for using the unbiased mass spec, we identified 216 differentially expressed proteins between IBM and histological polymyositis. Most proteins were up in IBM compared to polymyositis, with only 6 being down. And when we mapped where these proteins, what pathways these proteins referenced using a gene ontology analysis, we see this complex constellation of biological processes consistent with this multifactorial nature of IBM, with immune pathways, a very strong fibrosis signature, protein handling pathways, some pathways representing lipid metabolism, as well as metabolism of vitamins and proteins, and redox biology represented. So obviously we see some complex processes mirroring the complex pathology we see in IBM. So to extend our coverage of the proteome, we then applied the four Olink panels, and we chose these panels based on insights gleaned from the mass spectrometry results. So we included some inflammation panels, as well as adding a cardiometabolic panel to capture that metabolic component. And in total, across all of these panels, over 1,700 proteins were profiled. And you can see these are the PCA plots for each panel. You can see that there's partial separation between groups with healthy controls, polymyositis in dark blue, and IBM in red. And using these targeted panels, this really enhanced our discovery efforts. We identified a very large number of differentially expressed proteins, over 455 significantly differentially expressed proteins in the IBM versus PM comparator, and over 1,000 in the IBM versus healthy control. And so this is a lot of proteins to work with, and we're analysing all of these proteins with respect to their mechanistic implications. But the purposes of today's talk was to identify a minimal protein signature, or the smallest set that could robustly distinguish IBM from this histological polymyositis. So for this particular analysis, we performed a lasso regression, which is a method that builds predictive models by shrinking less informative variables to 0. And on the left, we have the lasso coefficient path plot. The x-axis is the strength of the penalty applied in this model. And as the penalty increases, we see less proteins in the model. And as the penalty relaxes, we have more proteins included. And then if we look to the right, this is the cross-validation plot. So this determines for what strength of penalty which confers the lowest misclassification error. And we can see that the lowest misclassification error occurs here, which corresponds to 14 proteins. If we move to the right, we're getting overfitting. And if we move to the left, we have too few proteins in the model to be discriminatory. And so we then applied this 14 protein signature and determined the discriminatory performance. And this violin plot shows the predicted probability of IBM for every patient in our cohort using this model. And what we can see here is that the IBM patients are clustering at high probabilities. PM is clustering at lower probabilities with separation between groups. And this is the resultant ROC curve with an area under the curve of 0.795. So this is where the proteins sit in the volcano plot. You'll see it's not always the most differentially expressed protein that was selected in this model. And this is the identity of those proteins. So we have representation of T cell or sometimes NK cell markers, particularly cytotoxic T cells or activated T cells. We had some representation of we have a B cell marker there. We also have some myeloid cell markers and other inflammatory signaling pathways, including the RAGE receptor. We have a vascular marker, as well as some proteins involved in neuroendocrine signaling, neuropeptide generation. We also have a mitochondrial protein involved in mitophagy and a protein involved in cellular metabolism. So again, a multi-system biological signature consistent with the complex pathophysiology of IBM. So we then asked whether these circulating proteins could be observed at the tissue level, and for this we analysed a cohort of muscle biopsies using the Xenium Prime 5K panel it's a spatial transcriptomics approach, which involves interrogation of 5,000 targeted genes at subcellular resolution. And we examined 13 muscle biopsy samples across several myositis subtypes. We also had some non-inflamed muscle included. And we've analysed this Xenium dataset several ways, and for the purposes of today, I'm going to present the first pass cell segmentation-free analysis using this methodology called FICTURE, which is a computational method which decomposes complex gene expression into recurring spatial patterns, which are called factors. And these factors may reflect cell types, cell states, or tissue niches. And this is an example of some representative output. This is one of the IBM patients, and some of interesting factors that emerged in this patient. For instance, factor 1 has a variety of genes that suggest this is a myeloid factor, and we also see representation of one of our biomarkers of interest. And factor 6 has a constellation of genes suggesting it's a cytotoxic T cell factor, and again, it has representation of one of our markers of interest. So what we did is we examined the transcripts reflecting our serum biomarkers and whether they appeared within the FICTURE factors for each patient and what those factors represented. And we looked at whether the transcript was within the top 20 genes identifying or defining that factor, or whether it was present outside those top 20. And what we observed is that KLRK1 and CLEC6A appear highly represented within factors almost exclusively in IBM, and that these factors correspond to immune cell programs. But in contrast, interestingly, markers like ENG, that vascular marker, is diffusely expressed, broadly expressed across the whole cohort, including in some normal cases. But the quality, and it corresponded to an endothelial factor, but the quality of that endothelial factor seemed to differ, with it being associated with more stress or inflammatory signatures in the disease cases. And some of those neuropeptide markers were broadly expressed in skeletal muscle, sometimes correlating with some neuronal markers as well. So in conclusion, IBM is characterised by a circulating proteomic signature that encompasses inflammatory, immune, metabolic, fibrotic, and proteinopathic related pathways. We found that complementary serum proteomics approaches spanning the protein dynamic range enhance biomarker discovery. A 14-protein signature discriminates IBM from histologically defined polymyositis, and spatial tissue analysis identified KLRK1 and CLEC6A as strongly spatially implicated in IBM muscle. So we have some ongoing tissue-based proteomic profiling to further validate these results, and we'll perform blood-based validation in a larger cohort. So these are my acknowledgements. This work occurred at the Walter and Eliza Hall Institute, which is my institute, in the Wicks lab. And this is a collaboration with Vidya LeMay from the Royal Adelaide Hospital. Thank you.
Thank you. Do we have any question, please?
I wanted to ask about the PM, patients, because P62, TDP43, even though the sensitivity might be high, but it's not 100%. So, because PM still, I find it controversial, like does it exist, or it's really rare? And when you were also looking at histopathology, did you just look at the TDP43 and P62, or did you look at COX-negative fibers as well? How do we know that the 36 patients of PM were really PM patients?
Yeah, great question. So the PM cases, we specifically chose patients that didn't have any evidence of mitochondrial dysfunction with the COX-SDH stain, no P62 aggregates on P62 staining. it is possible that they could have been some early IBM cases in that cohort, but and no rimmed vacuoles. So we specifically sought cases, patients, where those features were lacking.
Okay, so finish for now. Thank you. And now our last communication, Nathan Ruthledge from the UK.
Hi everyone, I'm Nathan. I'm a PhD student in my second year. Those here this morning might recognize me, and you'll probably recognize a few of the slides I had up. I've got no conflicts of interest. And I should probably start by thanking all the authors on this, because I've kind of swooped in here at the end of this GWAS. Apparently it's been going on for a long, long time. So I'd like to thank everyone who's been involved in this. And if you're not, we're going to be sending out the paper hopefully soon. If you're not on there, please contact us so we can make sure you're on there, because I know a lot of people have been involved in this. So as you're probably already well aware, IBM is part of the idiopathic inflammatory myopathy group, and it's got a complex etiology. You have the T-cell migration into the muscle, as you've seen before, but they also have these degenerative aspects, the rim vacuoles, the protein aggregates, that you can often see in things like Alzheimer's in the brain, but we see them in muscle. Now, one of the potential reasons for this could be the autophagy and the proteasomal degradation pathways being dysregulated in some way, and there's been some rare variant analysis that kind of backs up this idea that might be the case. Now, previous genetic studies of myositis have included IBM, but not to that large degree. So we don't really know the genetic architecture of IBM that well. Now, finally, we have the first large-scale IBM GWAS. So we've got, to begin with, over 1,184 samples of IBM. After quality control, that became 1,036. We also have over 7 million SNPs after imputation involved. And to make sure what we're looking at here is not affected by population, we've basically kept it to be a European, in a word-centric background when it comes to what is actually included. So what we're looking at, hopefully, is IBM and not population differences. So here is the nice Manhattan plot that you can see. Some things you might notice before, so the PTPN22 has appeared before in other myositis GWASs. Of course, you've got a massive HLA region, as you'd expect to see, but there are some new cases, such as RMIT, RMI2, ORMDL3, and also AHRR. Now, if you look at all these lead SNPs, the main thing that's causing the spike, you'll see that most of them are intronic. So they're not actually within the exon or the impacting of a gene, but they're lying in between segments, or in some cases actually in between genes. And also, should be noting, while we have got our genome-wide significance lying that dotted line, there are quite a lot of interesting ones that are just below kind of that borderline significance. That could still be interesting to look at as well. So if we zoom in onto that Manhattan plot, and let's look at that HLA region. When we zoom in, you actually see that the region we're looking at, the most association was the HLA DRB1 030101 amino acid change, which has come up before with IBM association, and it's great that we're seeing this come up in the GWAS, really further cementing that association that we've seen. We can then also look at other regions. So we zoom in on AHRR, for example, the highest peak that was in an HLA region. You can see a plot like this. So this is on locus zoom, and we can see is the purple SNP in the top is our lead SNP, and all the different colored SNPs are basically telling you, if you inherit this lead SNP, how likely are you to inherit the other colored SNPs? So the ones in red, you're most likely to also inherit them as well. And then this bottom one is telling you what's causing the most contribution to this GWAS in terms of why it's becoming genome-wide significant. And what you can see here, there's a very specific region here of AHLR, and a very specific SNP that's come up. And we can look at another one, so PTPN22. Now, normally, if I was to see this, I might be a bit concerned because there are no SNPs in LD when it comes to it. It's literally just lead SNP and one in LD. But, as you might be aware of PTPN22, this is a SNP found in the exon. It's appeared in other myositis and other also autoimmune disorders that's come up, which might explain why it's so specific when it's coming up here. And then finally, we can look at another region, which is a bit more complicated. So this is the ORMDL3 region. And we have our lead SNP in purple, and you see that there are a lot of other SNPs in red And you can see that actually a lot of these SNPs could be contributing to what we're seeing in this GWAS, and any of them could be quite interesting. And you might also see they're not just ORMDL3, it expands across a few other genes and before some genes. So it's basically saying, anything here could be interesting to look at. So we can also look at the previous GWASs that have occurred. What we found in our GWAS, has it appeared in other myositis GWAS? And you can see here, so on top is from our GWAS, and below are from the previously published GWAS, as you can see for PTPN22, SH2B3, and even NAB1, which is just below for us, that these loci are pretty much shared with what we saw before. So it's not an IBM-specific thing, it's more of a myositis loci that's come up. I mean, interesting for PTPN22, in the myositis it's just below, but for IBM there seems to be a stronger power there. So we can then further on and actually look at all those SNPs, because we said they were kind of a lot of them were in the intron regions. So do they have effect on gene regulation maybe? Are they in kind of in enhancer regions or promoter sites, or have an effect on transcription start sites? Now, this basic table, I've used something called RegularMDB, which is a software to predict this. The more ticks you see associated with the lead SNP, the more evidence that that might have an effect on gene regulation in somehow. Also, there are other EQTLs available for it. So there's giving more evidence that actually, yes, these SNPs might have some effect here. So I've mentioned all these genes, but what are they? Why could they actually be interesting? So we start with AHRR. It's a transcription factor. It's got a main role in inflammation. As I said, PTPN22, quite well known, quite well associated at this point. SH2B3, again, is involved in the T-cell activation. CD200 is an antigen involved in T-cell immune responses. GSDMB is quite interesting. It's a gene that's involved in cryopoptosis, which is a form of apoptosis related to the inflammatory pathway. Ataxin-2, that came up on the SH2B3 loci, is actually well associated with ataxia and other neurodegenerative disorders, but normally, in these cases, it's when there are expansions. And then finally, PDCD6. This has appeared on the AHRR pathway locus, and it's quite interesting, I think, because it's involved in T-cell apoptosis quite specifically. So then to follow this up, now we have our GWAS genes and loci to look at. Do they have any effect on expression in muscle in IBM? So we've got two cohorts. One is a collaboration at UCL with British and Italian RNA-seq for muscle. Another one was already published Finnish data sets, so two different population data sets to look at to see, okay, in our GWAS genes, what's happening in the RNA-seq. And you can see on the left is just your overview of the volcano plot of expression. And here on the right, we can see is in orange color, it's quite small, but they're all the GWAS genes. So all the genes from the GWAS loci. And you can see that most of them, actually, there isn't much change in expression at all in RNA-seq muscle. And in black, in the top, they are the kind of the top 50 most dysregulated genes when it came from both data sets. But there are a few that do appear. So PTPN22 does come up as being overexpressed significantly. But one very specific one that comes up is IKZF3. It actually appears in that top 50 when it comes to the UK data set, and quite close to it when it comes to the Finnish data set. So what is IKZF3? It's a transcription factor. Its main role is to be in B-cell regulation, but it also has involved in T-cell apoptosis, and one of its many targets is CXCR3. It's also come up before, and it has involved in basically migration of T-cells to inflammatory sites. So it can be very interesting when it comes to that pathway of IBM. This could be involved. And if I go back to that plot I showed you before of ORMDL3, you'll see IKZF3 is quite a big gene on here, and most of those lead SNPs, or most of the SNPs, were before IKZF3 in that region. So I put all of the SNPs into RegularMDB again to see, okay, could any of these SNPs have any potential impact on regulation of IKZF3? And the hypothesis is actually just maybe they could have an effect. They could be along the transcription start site again, or it could be infecting a promoter. So there could be that this might be one of the reasons why we're seeing that overexpression, along with what you might already expect to see. So to conclude, we performed finally the large first GWAS of IBM. We found some hits that have come up before, like PTPN22, as well as the HLA DRB1 hits for IBM. We found new hits, such as the AHRR locus, the ORMDL3, and the RMI2. The further RNA-seq follow-up showed that actually most of the genes, when it came to RNA expression, weren't that well differently expressed. But one of them was IKZF3, and maybe that could be interesting to look at. Is that actually affected from our GWAS hit that we're seeing? So I'd like to thank the Myositis UK for funding my PhD, because the International Myositis Society for having me, and also my lab, and also my supervisor, Pedro, for all the help. Thank you.
Thank you for your presentation. We have time for one question, one comment. Yes.
Thank you for a wonderful talk. I'm Tomoyo Yoshida from Japan. Just a brief question. Did you look at the co-localization with EQTL, for example, with some GTEx data or other EQTL resources?
So we did look up EQTLs in terms of what we're finding with other data. I don't think so, but we looked up if there are any EQTLs from GTEx data, and there were, again, there are a few from the hits that did come up, and that was kind of included in that. I know it's quite a big table, so quite small, but that was included in that table there at the end.
Thank you.
Thank you. Any more questions? Okay, so thank you.
So we are really on time, so we have to finish. Thank you so much for being here.
Thank you.
So this was the right time.
Yeah, I know.
We recovered.
A minute or two.
We recovered because it was.
Nice to meet you. Nice to meet you too.
Because we were a little bit late.
Yeah, I know, but you recovered with the talk, so that's great.
Thank you.
You're on time. So we have 3 talks. The first one is by video.
Yeah. Will Kevin be online with us? I don't think so. I don't think so. Yeah. They told me the communication was prepared, was ready, but I'm not sure he's online.
I can. Está tudo ok? Tem alguma... Yeah. Back now? I don't think it's back. Yeah, and times I don't think they have back. Yeah. Okay, sounds good. Thanks, Kat. And thanks for having me. Obrigado.
Okay, thank you. Não sabe se o Kevin vai estar online connosco.
Não faço ideia, posso perguntar ali.
Eu perguntei, mas
I confirm that he's not online. Just
Sorry?
I confirm that Kevin will not be online.
Will not be present. Okay.
It's gonna go probably about 2 minutes. They asked us if that's okay because they wanted to.
Okay.
Then we have mine videos. Okay. So how do you want to do it? We can alternate. We should get started.
Yes, maybe
start with the video.
If you want to start,
okay.
Maybe I can present Kevin.
Sounds good. We just briefly introduce ourselves. And,
okay.
And,
Good afternoon, everyone. So we're about to start with the inclusion body myositis session. If you can please take your seats. So I'm Elina Def, a neurologist at Mayo Clinic.
My name is Miguel Pinto. I'm a neurologist and neuropathologist at Oporto here in Portugal. And welcome, we would like to welcome you to this IBM clinical section. And without further ado, I think I'm going to introduce our first speaker, which is Kevin Austin. He was not able to travel from Australia, but we are happy to have one virtual communication that he prepared for us.
Someone able to pull Kevin's recording of his talk? There should be an audio.
Today from down under,
Thank you for the opportunity to present some insight from a research into living with the challenges of inclusion body myositis. Some of you may be aware of the research that I led over the last few years, exploring patient and carer priorities in IBM, working with Professor Merrily Needham and her team in Perth, Australia. If you haven't read this work, I'd encourage you to do so. However, the main purpose of this short presentation is to give you an insight into what it is like living with the daily challenges of coping with IBM. Or as one of our study participants put it, why having IBM truly sucks. Our research and work done by others illustrated the enormous impact of IBM, not only on suffering, but also partners, carers, who by necessity became carers rather than lovers. The biggest drivers of frustration and sorrow in IBM can come from activities and tasks that most of us take for granted. For me, some of these have become the greatest impact. At the very beginning of the day, you slowly become aware of your surroundings. Your first thought flows into your consciousness, toileting. You think how you feel. You need to make a move. What time is it? Too early to wake your loving partner and carer. Can I wait a little longer? And she still seems deeply asleep. No, I can't until the daily challenge begins with the first and major challenge for the day. Can I roll over on my back? Not quite yet. Will need help. The next I can manage, I call to rise to wake up. He immediately silently gets out of bed and makes her way to the end of my bed and removes the sheet and doona and pulls it back across the blanket frame to release my leg. Next he moves my leg so they straight and puts the slippers on. Next I see they move as far left of my body as you can both manage. I now need to get my right hand under my hip at the focal point, and she swings my feet towards the right side of the bed and lowers the bed until my feet are just touching the floor. She then raises the bed head upright as far as it will go and takes my left hand and pulls me up into a seated position using a forearm frame. I slowly transfer up a small ramp into the ensuite bath where our toilet is located. We twist around and back into the toilet itself. She raises the toilet seat using the electric toilet raiser, which has been fitted with a bidet. I lower myself using grab rails slowly onto the toilet seat and then use the toilet raiser remote to lower myself onto the toilet itself. If you've been following this, you may have an idea of how tired I feel, and all I've done is wake up and move to a toilet. With luck, you then successfully use your bowel and clean yourself up using the automated bidet. Now comes the biggest challenge of the entire thing, getting up. The various steps in getting up from the toilet and getting dressed takes about 30 minutes, and the end-to-end cycle of waking up, toilet and getting dressed takes about 60 minutes. The stress of toileting, unfortunately, is not limited to the start of the day. Due to the unpredictable nature of bowel incontinence, which is experienced by men with IBM 9 times more than those without IBM, 2 or 3 rushes back into the toilet are quite common, with occasional accidents. So toileting away from home is done reluctantly, and with some anxiety, limiting outings and social interaction. The height of toilets is a major issue for most aging patients and those suffering from muscle weakness. I made my way into the wheelchair and traveled into the kitchen for breakfast. A third significant challenge is falling. Another activity take for granted and most usually accomplished without conscious effort. There are quite a few variables at play, but the more severe incidents are truly frightening. Severe swallowing difficulty and dysphagia, including frequent cough through the frightening choking incident, can result in not being able to get a breath into your lungs. It's hard to describe the level of fear that can be overwhelming. I typically try to calmly have a normal meal, even when a large number of precautions have already been taken to simplify the process of eating. Turning your food up to bite size, having thicker food and so on. Indeed, I consider myself lucky. Some of my IBM friends are now nilled by mouth and have to be fed from G2 directly into their stomach. Preventive exercises with an expiratory muscle strength trainer every day and resulting in me having a dramatic improvement in swallowing patients. Failure in voice and difficulty communicating clearly is yet another normal activity that I'm aiming to. My mouth and throat operate at two extremes, either too dry or too much saliva. The things are sitting in and around my throat, also making it difficult to talk, communicate or swallow. This results in severe social awkwardness, and even those supposed to constantly ask you to repeat what it is you just said. Pardon? Beg your pardon? The main impact is you tend to remain silent for more than normal through a sheer embarrassment. Progressive loss of function in both hands and further burden in trying to navigate everyday time throughout the day. Think of any activity that involves hands and they quickly become major tasks and tough to follow again. Even signing your name can seem overwhelming. These challenges that take away independence and dignity along with muscle strength can have a severe impact on your mental health. And some kind of mechanism. First of all, IBM never takes a holiday. It's a condition that impacts suffering 24 hours a day, 7 days a week. The lived experience reveals a real, sometimes overwhelming burden on both patients and carers. Major daily challenges and for patients and carers include toileting, choking, lack of hand function, difficulty in communicating clearly, and these challenges can be overwhelming. Please ask your patients how they cope, because most of us are not. Life being IBM can be really tough. It sucks. And now it's few special thanks to some people that have given me enormous support. Thank you.
Okay, so we would like to thank Kevin for this impressive message. I think that we're a little bit behind of time, but if anyone has a comment to make, please come forward. Yes, please.
Kevin is not online, not joining us online at this time. He just pre-recorded the video. Dr. Schmidt?
I would just have a comment that maybe all of us in this room really let this settle. Life with IBM sucks. There are so many neurologists, they say ALS, it's such a dreadful disease. yes, but patients only mostly live with it for much shorter time than with IBM. So IBM is like ALS, an extended version, really dreadful.
Definitely, yeah.
And I think these patient glimpses really set the stage. I'm very thankful for that.
Yeah, very touching and very powerful video with strong messages that also remind all of us on the main reason why we're all here and what brings us together. So along the same lines, there are a lot of lessons to be learned from clinical trials, and especially in the IBM population, which is the main IIM population with no approved therapy. So for that, Professor Schmidt, who's a full professor and chair of the Department of Neurology and Pain Treatment at Emanuel Clinic Rudersdorf, the University Hospital of the Brandenburg Medical School, where he also leads the Neuromuscular Center and the Center for Translational Medicine. Dr. Schmidt.
Thank you for this kind introduction. So welcome, everybody. First of all, I would like to thank the organizers for putting together such an interesting program. I really enjoy this meeting, and we're just halfway through, and I'm looking forward to the dancing event tonight, I must say. So secondly, I thank the organizers again for inviting me to give this lecture. And now a disclaimer. You see my conflict of interest here. And now the disclaimer for every clinicians looking for new information regarding IBM clinical trials and pharmaceutical industry. If you think that at the end of this lecture, you would have a cookbook on the next IBM trial, I'm sorry to say. Rather go out, have a coffee, enjoy the sun or whatever. So it's so complex, I apologize. But I will hope to at least give some more information of, yes, what we learned from the negative, unfortunately negative clinical trials. So we've just had an excellent overview of the clinical picture of IBM. The pathology has been shown in several talks already throughout the meeting so far. What we do know is that there is inflammation and degeneration at the same time in the muscle, and we have cytokines, T cells, B cells, and then vacuoles and protein accumulations. The mechanisms, as in many diseases in the inflammatory myopathies, is not known specifically. What we do know is that there seem to be several predisposing conditions like impaired regenerative capacity, premature aging, and enhanced senescence, which then set the stage that the muscle is more prone how to respond to an inflammatory stimulus, inflammation with cytokines, T cells, B cells, antibodies, and that then proteins accumulate in the muscle fibers, and that then an interactive cell stress response kicks in with various cell stress mediators, for example, nitric oxide, various heat shock proteins, also mitochondria. And then in the end, we will see fibrosis, fat replacement of the tissue, and the point of no return, at least so far. So we've seen a plethora of different clinical studies in IBM, starting at the end of 90s with prednisolone, oxandrolone, cyclosporin. They were all negative. It goes on with IVIG alone or together with prednisolone, interferon, and a TNF-alpha block. Then anti-T lymphocyte globulin, alemtuzumab, and then more recently, large clinical placebo-controlled trials. All of these were placebo-controlled trials, bimagrumab, sirolimus, and arimoclomol. And I would focus in the following on the IVIG a little bit, and then go into more details on the more recent trials, bimagrumab, sirolimus, and arimoclomol. So the number one question is, why were all these trials negative? And we don't know. We don't know if the drug was not optimal, if it was the wrong mechanism that we targeted, or if the wrong tissue was targeted, if we should rather target
the muscle than T cells or B cells, or if the trial design was not optimal. Some of the early trials, as we will see, were crossover designs, or if the outcome measure was not optimal, or if the trials were simply too short. So I would like to share with you some unpublished observations or only abstracts presented at conferences regarding immunoglobulins. This is a short retrospective cohort collected a number of years ago, and this seemed to be promising. It was collected during the standard of care in Germany, which allows to give IVIG in patients with IBM, and we did see some promising improvement of the MMT and the time patients needed walking aids. This was comparable to a cohort published at the AAN a number of years back, again as a poster, never as a paper. And we then followed up upon, and we are now putting this together as a publication with a higher number of patients, 68, again retrospective, standard of care, not placebo-controlled. And in this cohort, looking at the IBM-FRS, we do not see a consistent change, which is consistent with what many of us, unfortunately, experience with IVIG. That said, there is a number of reports that have been shown that the swallowing abnormalities can, at least for a certain time, be even improved or stabilized in IBM, and this has been shown in the clinical trials, which had the negative primary outcome, but as a secondary outcome, there were swallowing assessments, and they were positive, significantly positive, and a number of case series. So now I would like to go into more detail on the 3 negative large recent trials that were conducted. The first is the bimagrumab in IBM Resilient trial, targeting the myostatin pathway. Myostatin is the growth brake of the skeletal muscle, and there are naturally occurring mutations in cattle and in humans, very rarely. And a drug, bimagrumab, was used to pull this brake off the muscle. It worked elegantly in the cell culture and also in a mouse model, and increased the muscle mass. Then a clinical trial was mounted with 251 patients over 52 weeks, and the primary outcome measure was the 6-minute walk distance, which turned out to be negative, similar to most of the secondary outcome measures as well. The second large study was the arimoclomol study, going back to data in the ALS mouse model, with arimoclomol targeting the heat shock proteins, so to stabilize proteins in order to reduce protein accumulation. And this worked in muscle cells very nicely, and it could be prevented that proteins accumulated. And there was also promising data in the VCP model to prevent accumulation of TDP-positive proteins. The clinical trial then encompassed 150 patients over 20 months, so 80 weeks, and the primary outcome measure was the IBM-FRS, and again, this was negative, and so were the secondary outcome measures as well. Lastly, the sirolimus study, so this is a phase 2 study over 52 weeks with 44 patients. It failed the primary outcome measure, the quantitative quadriceps strength, but at least several secondary outcome measures, including 6-minute walk distance, HAC, and quantitative MRI, were positively improved. A phase 3 study as an investigator-initiated trial is still ongoing with the primary outcome IBM FRS, 140 patients, and with 1.5 years, so 84 weeks duration. There are the ongoing trials now will be Vi+Vii. The Apcuro trial has just ended, and I will come to that on the following slide. Sirolimus, I just mentioned the phase 3 study, and there is one more study going on with a JAK1/2 inhibitor, but this will go on until 28 at least, according to clinicaltrials.gov. So the Apcuro study was the anti-KLRG1 study in IBM, going back to the T-cell signaling properties, and it has been shown that this target molecule, KLRG1, is present on cytotoxic T-cells that can be found in inclusion body myositis muscle. And the clinical trial was performed in 272 patients over 76 weeks, and the primary outcome was the IBM FRS. The data will be presented tomorrow by Namita Goyal. I can share with you this slide. There were 2 different dosings that were given over the duration of the trial, 2 milligrams or 0.5 milligram Vi+Vii, and as I said, the primary outcome was negative, and also the key secondary outcome measures, unfortunately, turned out to be negative. And more details we will hear tomorrow. We already heard from our patient from Australia that dysphagia is an important symptom, and it's important to really ask the patients if they have swallowing abnormalities, and then, if so, to perform adequate testing with FEES, video fluoroscopy, or if you have the possibilities with real-time MRI. We just had a follow-up study that we presented yesterday as a poster also. And why is this so important to really look what's happening? Because there is a cricopharyngeal bar, a functional stenosis in the pharynx, which we can see in video fluoroscopy. And why is this important? If you see this, you know that there will be a high risk of aspiration with all of what can happen, pneumonia, or even death by choking big chunks. So what can be done in this respect? There are no placebo-controlled trials out there yet, but a number of case series and even one prospective study. And it did not work in all of the patients. Local injections into the cricopharyngeal muscle with botulinum toxide. But a number of patients could show positive results as shown here. For example, 10 out of 13 or even 12 out of 12 patients were positive. So what are the lessons learned from these trials? I would go for the big 3 studies. I would like to tease out the mechanisms and start with the anti-inflammatory component that we used, for example, in the study with blocking with steroids, immunosuppressants, or with the T-cell block from Apcuro or alemtuzumab. What we do see, and this is a consistent finding throughout all reports, is that we do see a high number of pro-inflammatory molecules in the skeletal muscle, chemokines, lymphotoxin, cytokines, and that these are interlinked and really can be noted in the muscle fibers adjacent to the protein accumulation. And very important is that this can be mimicked very easily in the cell culture. So this is a logarithmic scale. It's relatively little on the bottom right. a number of pro-inflammatory cytokines or chemokines are produced at the mRNA level and they're secreted. And again, this is a logarithmic scale, so a massive production of pro-inflammatory cytokines and chemokines by skeletal muscle. So we had the chance of using some of the samples from one of the IVIG studies with IVIG and prednisolone. And we could see at the mRNA level that some of the mediators were reduced. However, for example, INOS, inducible nitric oxide cell stress-related molecule, remained unchanged. And we could mimic the same situation in the cell culture system. When we exposed the muscle cells, we could induce high numbers of pro-inflammatory cell stress, NO, nitrotyrosin, and this remained unchanged even when we added immunoglobulin or prednisolone. A very new finding was also presented as a poster yesterday. Is a chronic mouse model with overexpression of lymphotoxin, and in this mouse model, it's a transgenic mouse model. We could show presence of the whole range of immune cells, T cells, B cells, macrophages. We can see protein accumulations, vacuoles, and also mitochondrial dysfunction has been shown. So we used this model to look in an automated running wheel setup and expose the mice to prednisolone, to T lymphocyte block, or alemtuzumab. And to make it short, we saw the same as in patients. This was negative. So the mice, this was not sufficient to reduce or to prevent the ongoing impairment of the muscle and the damage to the muscle inflicted by the severe inflammation. And this is in line with data from Tom Lloyd's group that showed in the xenograft model that even a total depletion of T cell reduced the inflammation and reduced MHC expression, but could not prevent the myopathic transformation of the skeletal muscle. So it could be that there is an insufficient power of the pro-inflammatory mediators with the drugs that we use so far, or it could be that the target is not right, that we target lymphocytes rather than muscle cells, or that the network of the different mediators that we target is not optimal, and that we should rather aim at other chemokines or cytokines that we haven't addressed yet in clinical trials. What we do not know yet is what sirolimus will do, because that addresses immunosuppression as well as autophagic mechanisms. So I would go on with the muscle growth, with the bimagrumab or possibly other components. Christopher Nelke, as first author, and his group have shown a few years ago an overexpression of CDKN1A in a subset niche in patients with IBM, and it was a profound upregulation, and this was in line with biopsies where the P21, this is a cell cycle stopping molecule which is controlled by CDKN1A, is impaired. So we have a higher number of cell cycle arrest, which is an indicator of enhanced senescence in the muscles of IBM. And along the same lines, a group from the Netherlands showed last year the KDM5A histone demethylase was overexpressed in IBM, and this was along with an impaired regeneration, and this could also be mimicked in the cell culture system when cytokines were exposed. There was also an upregulation of this molecule. And lastly, similar data could be shown in the mouse system with an ongoing interferon gamma installation that these mice also experienced an increased senescence, and this was mediated by molecules that are important for the regeneration of muscle, PARCs, for example, and MyoD, which were impaired in this setting after chronic exposure to inflammation inflicted by interferon gamma. So the theory is that maybe enhanced aging impairs the physiologic growth and the damage repair in IBM, and that the continued inflammation induces early senescence in the muscle, and that this makes the muscle more susceptible to other damaging factors like T cells or inflammation. And of course, the enhanced aging predisposes to degeneration. Now, lastly, regarding the cell stress and regarding the arimoclomol study and comparable studies, arimoclomol, as we said, was a heat shock targeting molecule. There are other heat shock and chaperone molecules which have been shown to be present in IBM, for example, alpha-B crystalline. And alpha-B crystalline has been shown to be interlinked with accumulations of amyloid. And in, again, the cell culture system, this could be mimicked very easily with induction of inflammatory stimulus. Alpha-B crystalline was upregulated. And then, again, coming to the nitric oxide stress, this was present profoundly in IBM muscle samples, again, a logarithmic scale, and also the NO stress was visible in muscle fibers that showed accumulations of protein, and this in the cell culture system, there was a logarithmic profound upregulation of the molecule that produces nitric oxide in the muscle fibers, and when we blocked this, we could prevent the accumulation of proteins, and we could prevent cell death. We then went on recently to transfer this to a mouse model and showed this as a poster yesterday when we took away nitric oxide and crossbred mice on the APP presilinin background, the Kitazawa mouse. These mice perform much better in the automatic running wheel experiment, and also the myopathology was improved, and there was a reduced accumulation of amyloid in these mice. Another association of protein accumulation and mitochondrial stress has been shown a few years ago by Huntley et al. That could show colocalization with mitochondrial stress and TDP43, and additional evidence has recently or last year been shown by Felix Kleefeld and his team. So arimoclomol could be incapable of fully reach all damaged muscles, and maybe this single heat shock protein that is targeted by arimoclomol could not be sufficient to really control the cell stress, and other cell stress molecules could be more relevant, and the mitochondrial pathway could be more relevant also for the pathology. And again, we don't know about autophagy or sirolimus. So coming to the clinical part with the trial design, we know for a number of years, and these are clinical presentations, clinical landmark papers published back-to-back 2011 that have clearly shown that in the real world we have 5% more or less yearly progression in the patients, which doesn't sound much, but if you really think about 1 year, 5%, after 5 years, 25. So this is very significant. And a very elegant meta-analysis of the co-chair of this session, Edi Nadav, really looked at all of these previous clinical trials and looked at the placebo group. Because it's different real-world evidence, and then in the clinical trial, what happens under control conditions with the placebo group. And he could, with this meta-analysis and comparing all of these studies, could show that yes, also in when all of these studies were taken together, there is a decline in these patients, which is comparable both for the MMT, QMT, and also for the IBM FRS, where there is much larger variation, but still a continuous progression rate with an estimate pooled at 12 months. So looking at all of the clinical trials that I've showed before, when you look at which duration was used, so the table will stay silent, and I will just highlight the time or the different outcome measures. So the early trials used crossover with 2 times 12 weeks or 12 weeks, 24 weeks. So from today's perspective, this was relatively short. More recent studies, and one of the early studies used 1 year more or less, 48 or 52 weeks, and the recent studies, as we saw, used 80 or 84 weeks. Half of the studies used the quantitative muscle testing or MMT as primary outcome measure. One study used the 6-minute walk test, and 2 studies used the IBM FRS. So far, the best outcome measure is unclear, and maybe we need a functional index or consider subpopulation testing or certain subtests, for example, for swallowing. And it is unclear if 1 year is enough or better 84 weeks, and possibly the IVIG studies that have been done are too short. So in summary, IBM pathology includes inflammation, degeneration, and cell stress. Mechanisms like aging, senescence, mitochondrial damage are important. Combination treatments may be promising, and the optimal study design is yet unresolved. So the duration of 1 year or longer is probably advisable. The best outcome measure is so far unresolved. A functional index may be needed, and functions like swallowing are important. And as we already saw and heard from our patient from Australia, there is a number of things that we can provide help with, and it's important that we don't leave the patients alone with this. Although there is no pharmacological treatment, physiotherapy, rehabilitation is important, and to let the patients have all these assistive devices and to help this care is really of utmost importance. So I would like to thank you for attention, and the team from Göttingen and Rüdersdorf who have helped to generate some of the data that I presented today. Thank you very much.
Thank you, Dr. Schmidt, and we have time to take a question, if anyone has a question.
If I may, since no one is asking, regarding the interferon gamma, what are your thoughts on anti-interferon gamma therapies?
Interferon gamma therapies. This has not been tested yet. So interferon gamma, the source of interferon gamma is the least from muscle. So the muscle can produce CXCL9, various chemokines, IL-1 beta, IL-6, but the source of interferon gamma is more the T cells. So it hasn't been done. Personally, I would be skeptical.
Yeah. Jens, any thoughts about using qMRI as a primary endpoint to shorten trial duration and maybe select those drugs with more potential that could then proceed to larger phase 3 clinical trials with a clinical primary endpoint?
Thank you, Pedro. Of course, it would be helpful if we would have a good biomarker that gives us early information if a drug is working or not, so that we could dissect and know if to proceed and how to proceed, or that we only proceed with those patients who respond. If it's possible to have such quantitative MRI techniques or certain sequences, I think this would be a valid way to go.
One last very quick question.
Hello, thank you very much. I was just wondering, since you show that INOS plays such an important role in the pathogenesis, is it also possible that other immune cells like monocytes, that they are the main mediators of INOS, play a crucial role in the pathogenesis?
Thank you. Yes, so the INOS that we looked at really derive from the muscle cells themselves, but of course there are certain isoforms of NOS, eNOS, INOS, nNOS, and inflammatory cells, of course, can also produce nitric oxide and add to this detrimental feature, but this, at least to my knowledge, has not been specifically looked at.
Yeah.
Thank you. Thank you, Dr. Schmidt. And we'll move on to the next talk.
So it's now my pleasure to introduce Dr. Jemima Albaida. Dr. Albaida is an associate professor of medicine in the division of dermatology in Johns Hopkins. And she is the director of musculoskeletal ultrasound and injection clinic at Johns Hopkins.
Okay, thank you to the organizers for the invitation to come and speak, and actually particularly speak on this topic, which I would say is quite novel in Gcom, right? So, and I love how we've set up this session where we had the patient really introduce this, and we've talked about possibilities of cure, and now I think let's talk about possibilities of care, especially expanded care. So I have some disclosures which have no relevance to this talk, but I have a disclaimer. I am not a palliative care specialist, but I do practice primary palliative care, and that's become a particular interest of mine in the last few years, especially as we've been taking care of patients with IBM that continue to progress and we don't have effective cure. So my objectives for this talk are quite simple. It's really to shed light on the spectrum of unmet needs in patients with IBM, which I think our patient did very well, and to identify the role of palliative care in addressing these needs. And then over the last part, just trying to see a practical approach that we can take in incorporating these strategies in our care of patients with IBM. So I would like to start with a case, and I think this is a case of a patient I had seen that really did open my eyes to how we could do better. So this is one of the earlier patients I had seen in my early career. And so this is the story of Mr. F. So he was a piano player, he was a chef, and by trade, he was a social worker. He began to have symptoms quite early in his mid-40s with both weakness and trouble swallowing. Like many patients, he saw several specialists without a clear diagnosis. And finally, a muscle biopsy was done, and he was diagnosed with polymyositis on the basis of endomysial infiltrates and an elevated CK, and he was started on high doses of steroids. But 5 years in, he's still on the steroids, and he basically has diabetes, he's gained weight, he's more weak, even more than ever. And so they decided to be seen in a specialty center, and he was seen in the Hopkins Maya Center, and I was fortunate enough to be the one to see him. And so when we actually saw him, he definitely had Cushingoid features, right, habitus all the way, but the diagnosis was not in question by that time. He was in his 50s. Definitely deep finger flexor weakness, forearm atrophy, quads atrophy. And we really did clarify the diagnosis that this is not refractory polymyositis, this is IBM, and our focus became trying to get him down on the steroids, got him to PT-OT and all that. But we don't often see patients who are local. So these patients lived, he lived several states away. And at that time, we would see patients quite infrequently, like once a year. And so we would, he started having trouble being seen or coming to our center, and he was getting most of his care locally. But we stayed in touch, obviously. And then as with this disease, you start seeing progressive dysphagia, right? So he had repeated aspirations, was hospitalized for pneumonia several times. And in fact, during one of those hospitalizations, a PEG tube was placed with very little discussion about pros and cons of it. But also with each hospitalization, he got more debilitated. And at one point, he was discharged in a wheelchair, and he never regained the function or ability to walk. He also lost the ability to do the things he loved, right? Play the piano, cook. And that also, together with social isolation, led to a lot of depression. His wife was his primary caregiver, and they had no children, and so she was literally solely focused on his care. They were a little more savvy because he was a social worker, so she knew how to get these resources. She figured he needed a fitted wheelchair. They got a mobility van, and by the end, she got him a Hoyer lift, and she had taught herself how to do it because he had really needed transfers and help with everything. And finally, during a similar hospitalization for respiratory failure where he had been intubated, not the first time, he passed away suddenly. And I remember the wife was saying, she just didn't know it was the end until the end. It seemed he had always been sick, but then he would get better. And then so she just never understood. In fact, the day before he passed away, she had asked their primary care about palliative care. And his response to her was, oh no, let's not give up on him yet. Right. So in that case, like just thinking about that patient, it really does highlight more of the complex needs in IBM, the multiple unmet needs. And this is the way I'd been thinking about it. Clearly, even with Kevin's description in the earlier part of this talk, there's way more symptoms than I've even listed here, right? Dysphonia, a lot of the distress that's emotional. But I also think there's a bit chunk that we are not doing as well in terms of conversations that needed to happen, right? Whether prognosis, it would help with complex decision-making, goals of care, advanced care directives. But a lot of support was also needed, right? For the caregiver who was dealing with caregiver burden, the economic burden of all the things our patients actually have to buy for themselves and the implements that sometimes are not covered by insurance. And social work would have been very helpful in this case. And so as I was thinking about that, and actually sometimes it can be overwhelming, right? As a provider, you're like, I'm not sure how I can get all of this instituted. And then you actually remember there is a specialty that deals with this, and that is actually palliative care. And so what is palliative care? So one definition is really that it is specialized medical care for people living with serious illness, focusing on relieving symptoms, pain, and stress to improve quality of life for patients and families, right? So the focus is really quality of life. And that's really an amazing thing to combine with the medical types of treatments we're doing. So it's appropriate at any age or stage of illness, and it's important to make this distinction. It can be provided alongside curative treatment. Another definition that's particularly almost striking is that palliative care can be seen as the identification of total suffering or total pain. And here you'll see we do quite well with the physical, right? But there's a ton of things like social, spiritual, psychological, unseen, and sometimes not spoken about by the patient if you don't specifically ask for it. I actually think it's important to talk about the myths of palliative care. And I think this still is pervasive, whether among providers, but also patients. I find myself actually, when I first started talking about it, being quite sort of defensive. Like when I introduce the word palliative care, I almost have to say immediately, it's not hospice care, right? Because it is scary for a patient to think about it. So that is one of the myths. Hospice is part of palliative care. It's end-of-life care towards 6 months. But there is a whole range of palliative care that can be integrated earlier. It is not giving up on the patient, as the doctor had mentioned, and it does not preclude IBM-directed care, right? So sometimes when I talk about with the patient, like introducing the concept of palliative care, they're like, oh, but I'd like to talk about the clinical trial. And actually, doesn't preclude us from talking about the clinical trial or talking about any other types of treatments that we could offer the patient at that point. I think there is this concept that I think was part of the what's been a pervasive sort of thought about palliative care that doesn't do it justice. So you see here in the past, we would think, since it's been mostly used in cancer, right? So you try for medical therapy, curative therapy, and at some point, when it starts becoming futile, then you say, we transition to palliative care, right? But that's not actually the model anymore. And a better model and way of thinking about it is that it can be instituted at any point, and early is better. And then you provide disease-modifying care. But as treatment becomes less and less effective, more and more symptoms happen, right? So palliative care, the gradient just becomes, it shifts a little bit more from focusing on treatment towards more of symptom relief and quality of life. And then hospice care comes towards the end. 6 months, again, is sort of the cutoff for that. But really, you see that care even continues after bereavement care, which seems to be an amazing concept to, in terms of supporting our patients. So I hope I don't really have to answer this question. I mean, we do understand why palliative care would be very, very important for IBM. So we know it's a progressive disability. It's a life-altering illness. We don't have effective medical therapy. And there's a high burden of symptoms and emotional distress. In fact, as we know it now, we know there's a high morbidity and mortality. And also, patients are dying of direct IBM complications, right, with pneumonia and respiratory failure. This is a concept I actually want to clarify, which I think is very helpful. So primary versus specialty palliative care. So primary palliative care is being provided by us, neurologists, rheumatologists, and we're doing that naturally as we offer recommendations for how to help the disease. When we say, oh, let's get you to a swallow evaluation, let's get you dilatations or botulinum toxin injections, that's all palliating their symptoms, right? And when we talk about the disease and how to think about the future and goals of care, those are all palliative care practices. And they really are skills that all physicians should have, right? So, but then when we now talk about specialty palliative care, we're referring out to our colleagues in palliative care. So it's a team of physicians and their basically, that's the one thing I like about referring out to specialty palliative care, that they work in teams because they realize complex care is needed. So you usually get access to a physician, nurse practitioner, social worker, nutrition, a chaplain, therapists, and so on. So they're particularly focused on symptom relief. And it's often offered when symptoms are complex, decision-making is challenging, or the level of distress is high. So when I was thinking about this, then it became very clear we need to refer out to specialty palliative care. And so when I had reached out to colleagues in our institution, part of the question that was almost shaping what would we be doing is trying to understand what's actually there. There's very little in the literature. And so we actually started by looking at what's the utilization in inpatient, because inpatient is a very good touch point, right? It's easy, you can refer out, you have access to palliative care in the hospital. And certainly before invasive procedures and things like that, goals of care should have been had. And so we used a national inpatient sample. It's an openly, publicly available database. And we just restricted to 2016 to 2020. And you found that a sizable number of our patients are getting admitted, right? So over 9,000 patients were hospitalized with IBM. When you added particular codes for palliative care consults and referrals, only about 7.1% received palliative care services, and 29% of those had required respiratory services. What's interesting is there's still a reasonable number of 9% who actually did get intubated but never received palliative care, similar to my patient, Mr. F. So, but we also found that we were trying to look at what led to referrals, whether it's being in a city versus like a more rural area. Those never were significant, but it seemed that higher comorbidity scores led people to think of palliative care more. But like many palliative care studies, it showed that people who were referred for palliative care had shorter length of hospital stays and lower hospital charges for the patient. So that's inpatient, which is probably the easiest way to implement it, because outpatient, it's a little harder. But overall, when you think about some of the barriers that could be coming into play with us using palliative care, Dr. DeViser had written a nice review on this in neurology, and I think you guys are far ahead of us in rheumatology. But I think there's a general barrier and there's IBM-specific barriers, right? So general, sometimes there's a lack of awareness of the helpfulness of this approach and how to integrate it into your plan and like when. But also there is truly a lack of, there's not enough palliative care specialists, and sometimes certain regions don't have any. And even if there are, they might not know our disease well. So it's hard for them to kind of discuss what to do with the patient if they don't know the disease. So some education also needs to happen for them. But also there are barriers from the patient, and I've experienced that, where even though you talk about it, they'll say, I'm not ready to think about that. Okay, and that's why there's this sort of stigma and a myth about palliative care. But at least from IBM, when you think about it, some of the problems are, versus ALS, it's kind of easy to say, we need this because it's so grave and they decompensate so quickly. IBM, it's sometimes slowly progressive, right? And you don't know when are we going to introduce this. And the trajectory of each patient is highly variable. We don't have any defined triggers for referring. And I think this one came to light where, when we thought about it, there's limited contact with the healthcare system. Many of our patients, at least seeing us who know the disease, might be very infrequent, like once a year and so on. They basically don't see the doctors anymore because they feel like there's no treatment anyway, so it's kind of futile. But at the same time, you can always, with every visit, determine what symptoms are emerging, what else can we do to sort of help with their disease. So as mentioned, I think a lot of what, when you think about what to do for IBM, a lot of the literature is, let's say, from ALS. So one of our colleagues, I had been working with a neuropalliative group, they were embedded in the ALS clinic, right? So it's normalizing the conversation. They get a diagnosis, they're seen by therapists, then they're also seen by palliative care. So it's not scary, it's just a normal part of care. Definitely, incorporation of palliative care in the area of ALS has really been shown to improve multiple outcomes. So in this one study that my colleague had done, looking at patients who had ALS and had received palliative care services, they just did a qualitative survey. And what they found was that patients were most commonly satisfied with the neurology ability to answer disease questions, counsel about medications, and manage their motor symptoms. That makes sense, right? But they were less satisfied with the ability to manage mood, address spiritual existential concerns, and find in-home care providers and coordinate care, right? That's also not surprising to us, like even as rheumatologists and other providers. So sort of the conclusion was that specialty palliative care provides emotional support and care coordination that is complementary. But patients did say the doctor needs to bring it up. They would not be the ones to think that it was actually good, and also the doctor endorsing it really, really helped their uptake of that referral. And they thought that it's not too early, never too early to actually discuss this, but they're often referred too late. I think I just wanted to comment on this because neurology, it's pretty, they're pretty good. Like I think they deal with so many diseases that don't really have a good cure. So it's easier for, I think, neurologists to understand palliative care. I will say in rheumatology, one of our barriers is that we're used to having treatments, right? So we're used to almost like being able to raise people from the dead. So it comes to your mind late to sort of think about palliative care and other adjunctive measures. I actually like this. I took a picture of this and then I was like saying, what are the reasons for referring to specialty palliative care? And you can use any of these reasons, advanced care planning and so on. And more often than not, more than one reason is used. But I found it was very helpful for me because I can tell you that sometimes I get a little like, not defensive, but sort of where you're like, is it okay to refer or will they judge me? Like I just want them to have a goals of care discussion and start talking about advanced directives. I feel out of my depth with that. But actually, completely appropriate to refer to specialty palliative care, and I've never been turned away for doing that. But I think one of the questions really, the truth is timing of the referral, right? So as I mentioned, sometimes it can be slowly progressive, but sometimes not. So the heterogeneous trajectories and the prognostic uncertainty tends to be there even in our own minds. If I've seen the patient for the first time, I can't really tell if they're going to be a fast progressor or a slow progressor, and when do I trigger. I show you a figure on the side of the proposed trajectories of dying, right, from the geriatric literature. So when someone has sudden, a sudden accident, sudden death, you can't really intervene with palliative care. The terminal illness is the cancer model, right? You know patients are going to die from that. It's easy for them to say when they'll actually trigger. I actually think that maybe IBM is somewhere in the frailty especially for our slow progressors, the older age group, and that's a little harder to determine when to actually say, oh, they need palliative care. But there are certainly a group of people who model after this organ failure, right? Like the patients who fall and then have decreased functional, the functional decline, and then suddenly they'll get back a little bit more with some PT, but then boom, they aspirate, and then further functional decline, and then you just realize they never get back to that baseline, and then they're further and further. So even at that, wondering when that sentinel event is, that opens your mind that, oh, we need to be doing something more. In terms of timing, it is certainly some things to think about is that those who were more affected by their disease had greater benefit from palliative care, it seems. They're more interested in it, obviously, the more severe their symptoms are. But certainly, disease stage or severity does not strictly correlate with perceived quality of life. So, in fact, your young patient with a new diagnosis, sometimes I do that in my head. I'm like, oh, you're kind of mild. Like, you're not as bad as 90% of, let's say, the patients I'm seeing. But actually, that's not fair, too, because actually those patients are the ones who usually have such distress and anxiety about the diagnosis and the loss of vigor, you know, in the early years. This is a question I actually thought was quite helpful from my palliative care colleagues, where they say, you know, sometimes when you're thinking, should I be thinking about palliative care? Should I be thinking about advanced care planning and so on? You just take a step back and say, would I be surprised if this patient died within the next 6 months or within the next year? And if reasonably that question can be answered by a yes, you should probably be doing something. And it's not wrong to actually think of referring or even start having those conversations about advanced directives. So this is just what I think in terms of possible triggers for referral, collating from the data from neurology and just thinking about IBM patients. So I think definitely significant disease progression. How you define that is an area of question, but I think sometimes when you see the IBM FRS drop several points, that's pretty significant. I also think about it when, let's say, someone is stable using a rollator or a cane, and then they now are in a wheelchair. That's a big transition that leads to a lot of changes and adjustments that you need to actually be addressing. But certainly frequent hospitalizations, dysphagia, that is no longer responding to your maneuvers with dilations or injections, weight loss, increasing falls, and I love this actually, distress of the patient, caregiver distress, and you can add physician distress that's actually there. And sometimes it's true, where when you feel, I'm out of my depth, like I'm really sort of, that is a reason to actually refer. And then obviously if patients request palliative care, and so the patients who are here, you can ask for this. And in countries, I did include it, where physician aid in dying is permissible, when they bring up that question, it really is sort of a reason to think about palliative care. So that specialty palliative care we've talked about and referring out, but obviously we still have to do some stuff and you have to do a lot of it. And I realize part of palliative care practices is really what we call as high quality communication, right? And most of that is actually listening and active listening. But I just collated some tips which I thought were helpful for me as I was doing this, right? So in the palliative care experience, they say you want to set realistic expectations, but it's also important to preserve hope. So really making sure that balance is good. You also really want to provide anticipatory guidance to a patient so they can plan ahead and really determine how they want to spend their time, you know. And then you want to explore patients' values and goals early on for shared decision making, and that's happening at multiple points, right? It's not just a peg tube, it's not just bigger interventions. I like this whole, because sometimes even breaking bad news, you're like, how do I do that? They actually suggest an ask, tell, ask approach, right? You ask the patient what they're ready and willing to hear. You actually ask for permission to talk about, do you want to talk about, let's say, prognosis or what I think your disease is like, whatever question, but you ask first. And if the patient gives you permission, then you tell what you, how you understand it. And then ultimately, you ask back again to the patient any questions and any follow-up. In terms of sometimes the question about prognosis and what can be, they have this thing called best case, worst case, most likely, right? So I like that, where it's like best case scenario is this, then worst case could be this, but most likely is you're probably going to be stable for a few years. It's kind of like the airplane, right, where they say case of a water landing, which is very unlikely, but they tell you sort of what to do. So I think that's a good maneuver. But ultimately, for me, I just use this where, we do a lot in our clinical visits. We do IBM FRS, we have to do their strength, all of that. But I always want to end with asking, like, what symptoms are most concerning to you at this time, so I can make sure I address that. Because in the end, it's really up to us to talk about prognosis and trajectory because we know the disease best. It's not really the specialty palliative care who have never seen your patient or know about IBM to do that. So at least in our experience, and if you wanted to think about how are you going to do this, I had reached out to our palliative care department in my institution, and they had referred me to one of the neuropalliative specialists who was working in ALS. And so we thought, great, we can do that model, embed them in our myositis center clinics and so on, until we came up against tons of administrative and financial barriers in finding support for this person as well. And so in the end, we've created a pathway to the outpatient palliative care clinic. And so in our, they distinguish oncology and non-oncology indications, right? So non-oncology are chronic diseases, heart failure, ILD, things like that. So appropriate was IBM. And then reaching out to them to explain our disease and almost how to think about muscle disorders and, in whichever way, so that they understand what the patient experience is like. And then I started referring patients when I would have these clinical triggers that were noted. But I will say that not everyone is receptive to a referral, and anecdotally patients, but I've actually told them I wanted to partner with them because we're trying to understand things. So please let me know your experience. And the people who have done it, qualitatively will say incredibly helpful. And it led to further referrals to other things like nutrition that I wasn't thinking of. And so in the last part, I just wanted to, let's almost, now that we know, I almost feel like, how do we revise the story of Mr. F? I think it only starts when we see them, but certainly even that change in diagnosis is something that is like a sentinel event and very confusing for the patient. So a longer conversation might be had, but sometimes you can't discuss everything. I might have gotten them back more frequently just to talk about different issues. When he could no longer be seen in our specialty center, use of telemedicine might have been helpful if available, or discussing with their local team about getting on the same page about goals. But certainly early palliative care referral, looking what the community and family resources are, patient navigators, and so on. In terms of dysphagia, not just the things we're doing, but really also goals of care, discussion about a PEG tube, pros and cons. It's not always, they don't see that as actually very, very helpful because it doesn't prevent aspirations. And then really screening for depression before it's very evident, exploring alternatives. They always think about this, like, oh, he liked to play the piano, he can't do it anymore, but can he listen to music and having that play in the background? This is a perfect time for community resources, right? Support groups like the TMA would have been very helpful. Respite services for the caregiver, hospice towards the end, right? And even bereavement services, lots of things that could have been done. And I think about this a lot, like let's say with that patient, right, with Maya Angelou who had said, do the best you can until you know better, then when you know better, do better. And so I think our challenge is, I think we can do better. And so this was just sort of how we had been framing it, and I realized education is needed even among our community in rheumatology, so we're trying to advocate for this. We do very well with the physical part, but there's like domains of psychosocial, spiritual, social, all of that, and there's ways that we could think about this. I just want to leave you with some helpful resources because it is true there's the Center for Advanced Palliative Care that you can actually go to. They have modules for communication. You can go through training of how to do goals of care and so on. But very helpful to find palliative care doctors, where I realized most of our patients don't live by us. And so the way I had set it up in Hopkins isn't applicable. But then they're in Florida, they have palliative care. I basically go online and find a provider, or I rely on the primary care, like, please, do you know any palliative care doctors in your area? And so I hope that as we conclude, you've really been able to get more of an understanding of the complex unmet needs in IBM and how we could potentially do better by referring early to palliative care. But certainly this is an area that I think should be an area of active research as to what are the needs and how we can best meet them. And with that, I thank you for your attention.
Thank you. Thank you, Dr. Obalda, for this wonderful presentation. I think we have time for quick questions. Yes.
Come to this one.
Oh, yeah, maybe the next one.
And if any of the patient would like to share thoughts or have a question, we could also bring the microphone to you.
Shout, or I'll repeat your question.
Thank you, Mima. Just 2 comments. It's a shame that this session is actually only in the IBM session. Because
it could be for everything, yes.
It works for everything, and this would be a topic probably for the next Gcom to have it in a patient workshop.
Absolutely.
A second comment is that I think the word, you mentioned about the difficulty in approaching a patient with this topic is really from the word palliative, because
I know.
of the whole, stigma of that. So maybe we can think about doing a terminology change.
Yeah. I'd have to ask the entire specialty to change the name. But it's true, it is true. That's part of the stigma, right? Yes.
Thank you very much for an excellent lecture. I'm so happy that this important topic is now...
Marion, you could have done this talk.
No,
I was like,
on the program,
why am I giving
the program of the Gcom. One comment, please. We know from the ALS literature that there are huge cultural differences.
Yes.
For instance, people from the eastern part of the world are less inclined to discuss this sensitive topic as regards, as compared to the patients from the Western world.
Absolutely.
Yeah.
As a comment, absolutely. In fact, and also there are some where spiritual practices are already inherent, so they don't need, but there's definitely some where, but I would say, and that's where your sensitivity as a physician comes into play, right? And you can actually sense who you can start talking to about it earlier than later. Yeah.
And I did not quite understand, and maybe it's the same question, but we have heard yesterday that ILD can be very severe.
Absolutely.
And dermatomyositis with MDA5 MDA5 positive antibody. Do you think that there is a place for palliative care in these patients
Absolutely.
as well?
In fact, well, because this is an IBM session, but we refer, so in our, we have a pulmonary palliative embedded in our clinic, and we have a breathlessness clinic. So in fact, when I couldn't make inroads with regular palliative, she was like, I'm happy to take your patients for IBM. So she'd do the goals of care and things like that. Yeah.
We have another session in this room. Christine, do you want to go quickly?
Yeah.
Sorry, that will be the last question. And
this is the patient with...
do you have
Oh, yeah.
Okay, so we have to stop. Let's continue.
My name is
We have 5 minutes before the next session.
My name is Marie-Christine Breveld. I'm an IBM patient.
Yes.
I have a very, very sensitive question.
Okay.
Thank you for your presentation. Palliative care in the Netherlands also includes assisted death.
I understand.
I know a lot of
That's why I put that.
And a lot of IBM patients, and we were talking about life expectancy. In the Netherlands, the life expectancy is shortened because of a lot of patients choose
to use
assisted death. I think it's important that this topic is also addressed in the future, because there's a lot of need of assistance psychologically, etc., etc.
Yeah, understood. So in fact, we don't have experience with that, but I think the next session, we should have someone from the Netherlands in particular talking
Thank you,
about
Dr.
assisted
Alwada.
dying.
Sorry, there's another session
Yes.
that's starting now. But
Okay, thank you.
Okay. Hello. Thank you. Good afternoon. Thank you. Thank you for being here. Enjoy your lunch. My name is Angela Ceribelli. I'm a rheumatologist. I come from Ospedale Civili in Brescia, which is located in northern Italy. And today I will co-chair this session with Eleni Tiniakou from Houston, Texas. First of all, I would like to thank Octapharma for the symposium. I would like to thank Gcom for the opportunity to talk. I will start with my presentation first, then Eleni will be presenting her presentation. We will have a question and answer session at the end of the Eleni presentation. So feel free to use the microphones in the rooms, but we will also have a QR code so that we have an iPad where we can see your questions. And at the end, Eleni will go on with concluding remarks. Okay, so this is the QR code, okay, to ask questions. So take your time to scan the QR code. Okay, so I think I can start with my presentation. I have nothing to declare. So as we've heard in these days, idiopathic inflammatory myopathies are really rare, heterogeneous conditions that can affect the muscles, but in many patients, other organs and systems can be affected. These are the pictures of one patient that we have diagnosed in our unit 2 weeks ago, basically. She has dermatomyositis with anti-TIF1 gamma. She has a history of breast cancer. We know patients have genetic predisposition to the disease, but there are also some environmental triggers that can lead to the immune tolerance breakdown. And it is very important to identify which autoantibodies are involved in inflammation and tissue damage. This shows the heterogeneity of the condition. In our clinical practice, I think we see every day different patients, even if we call them dermatomyositis. Some of them may have dysphagia. Some others may have pulmonary disease, some other, again, myocarditis. And this also depends on their specific autoantibody signature. This is true also for dermatomyositis. We know that patients with MJ, NXP2, or MJ5 may have severe skin manifestation of disease. And we all know what is the paraneoplastic dermatomyositis, in particular in patients with the T1 gamma and the XP2 antibodies. This is a very severe subset of the disease. And sometimes we still see difficulty in diagnosis because the target antigens of antibodies are in the cytoplasm. So we see reports sometimes that talk about cytoplasmic ANA, but no definition of autoantibody. Moving to the treatment of dermatomyositis patients, we all start from glucocorticoids. I know that we can do IV therapies, we can go from IV to oral therapies with different dosage, but then we move to immunosuppression. If this doesn't work, we have to think about IVIG, immunoglobulins, and in particular today the the attempt is for a more personalized choice of therapies, including IVIG, for example, for severe refractory myositis patients who have, for example, dysphagia. So the ProDerm study was fundamental for the approval of IVIG use in dermatomyositis patients. It was the demonstration of the safety and efficacy of this therapy. It enrolled 95 patients that were randomized 1-to-1 to receive IVIG 2 grams per kilo every 4 weeks up to week 16 versus placebo. After week 16, patients were entering the open-label extension period up to week 40. And these results show that there was a stable improvement of the mean total score from week 4, so early after the study, up to week 40. And the results show that the primary endpoint was met. There was a significant proportion of responders with at least minimal improvement at week 16. But also the secondary endpoints were met. In fact, there was a significant proportion of responders with at least moderate or major improvement at week 16. As for safety and tolerability, the reports, the results show that most patients were complaining about headache, 42%. Other symptoms were less frequent, such as fever, nausea, or vomiting. And the severity of these symptoms, however, was defined as mild in 74% of cases. When it comes to serious adverse events, there were 8 thromboembolic events in 6 patients treated with IVIG. 6 in 5 patients were considered possibly or probably related to IVIG. So what was analyzed was the baseline risk factors in these patients who had events, and in fact, they were significantly higher. So this led to the study protocol amendment. So there was a reduction in the infusion rate. So as you can see here, before and after this change, there was a reduction in the number of events. No hemolytic transfusion reactions were observed, no deaths occurred. So the results from the ProDerm studies demonstrated that IVIG has a favorable safety profile for the treatment in adult dermatomyositis patients. And this provides evidence to use this type of therapy in dermatomyositis patients. After the publication of the results, there were several post-hoc analyses. This first one wanted to analyze possible predictors of response to IVIG in patients with dermatomyositis. It was particularly focused on the serological subtypes of dermatomyositis. So this was the distribution of autoantibodies in the ProDerm patients. What was observed was that IVIG was effective in the dermatomyositis population analyzed, regardless of their demographic features and autoantibody status. However, they observed that patients with higher cutaneous disease activity and anti-Tif1 gamma responded better to IVIG. On the other side, pulmonary disease predicted a lower but still effective response to IVIG. This second post-hoc analysis was focused on the efficacy of IVIG beyond skin and muscle. So the authors wanted to focus in particular on arthritis, arthralgia, and fatigue. And they observed that these aspects were significantly improving under treatment by IVIG. Also, this post-hoc analysis confirmed the efficacy of the use of IVIG for active skin disease. So patients starting with moderate severe CDASI score were improving, going down to mild CDASI score. And also from a muscle point of view, there was a gain in muscle function, so an improvement in the MMT8 scores. So based on these results, we wanted to analyze also our patients. So I will show you what is our real-life experience in a single-center cohort. So basically, we evaluated retrospectively 15 adult dermatomyositis patients followed in our unit. They received, as in the ProDerm study, 2 grams per kilo of IVIG in a course of 2 to 5 days every 4 weeks for a total of 6 months. And now we are following these patients for a period of 48 months. So our patients have a mean age of 61 years. They're mainly female patients. In 2 cases, we define them as overlap with scleroderma, with mild skin features, no organ disease related to scleroderma. We had 2 cases that we defined amyopathic dermatomyositis and 5 paraneoplastic dermatomyositis patients. They had solid tumors. Unfortunately, 3 of them died during follow-up. This is the distribution of autoantibodies in our population. So as you can see, there is a very high prevalence of anti-Tif1 gamma antibodies, followed by lower prevalence of other myositis-specific or myositis-associated autoantibodies, which is a bit different from the ProDerm autoantibody status. So as you can see here, the comparison, for example, it is particularly evident for Tif1 gamma, which is much higher in our cohort, probably related to the paraneoplastic forms, which were Tif1 gamma positive. So our patients had dermatomyositis with active muscle impairment. In particular, the reason why we decided to go on with IVIG were muscle weakness and dysphagia from a muscle point of view. They were already taking corticosteroids, both IV and then followed by oral steroids, and they were all at least taking 1 immunosuppressive drug. In 10 out of 15, there was also active skin disease, as you can see here, mainly represented by Gotrans papules and heliotrope rash. 2 of them didn't respond to the adding of IVIG, so we decided to change therapy. Basically, as for the ProDerm trial, we had no safety issues. Only 1 patient had nausea and headache. We are observing clinical improvement of these patients, which is also reflected by the significant decrease in CPK levels before and after the last infusion. So even in our experience, we can say that IVIG is safe, is effective. We are treating adult dermatomyositis patients with active muscle and skin disease, but we are also treating paraneoplastic forms of dermatomyositis which are non-responsive to conventional treatment, such as corticosteroids and immunosuppressants. So again, I would like to thank Gcom for the opportunity to talk to this in this symposium. This is the group in Brescia I belong to. We are a big group. We are part also of the European Reference Network for Myositis. And I would like to thank you for your attention. Okay, so now we can move to Elena's presentation, please.
Good afternoon, everyone. Again, thank you for the introduction, and I want to thank Octapharma for the symposium. I'm really pleased to be here. So, for this part of the talk, I'm going to talk about the knowledge that we have about IVIG and other types of autoimmune myopathies. These are my disclosures. So IVIG is a plasma product. It derives from large donor pools. Like each batch contains antibodies from 10,000 of healthy donors. And they contain a broad and diverse repertoire of antibodies with different pathogen specificities. And this is where it relies its function. And that's why it was initially used as a means of artificial passive immunity. The idea is like very straightforward. Patients with primary immunodeficiencies that they don't produce any antibodies, they get IVIG to protect themselves from infections. But soon it was seen that it has some effect on autoimmune diseases as well, and it has an immunomodulatory effect. The first case was published in 1964, a case in Germany where a child with primary immunodeficiency was being treated with IVIG for that reason, but then later on developed autoimmune thrombocytopenia, and the physicians noticed that the IVIG was also improving their platelet count. So that was the first indication that IVIG maybe has some more effect, expanding from its antibodies against specific pathogens. So since then, IVIG has been approved by FDA in the US for many different indications, like CIDP, ITP, Kawasaki, and of course, Dermatomyositis. But it's also been used in many other diseases off-label, many other autoimmune diseases. So all of these diseases have a different pathogenetic mechanism. So based on this, we cannot infer a common mechanism for IVIG. And that is the strength of the medication, most likely. So it's a quite powerful immunomodulator because it acts at many different levels. So we have the Fab-dependent, which is the part of the antibodies that binds to pathogens. So the immunomodulatory effect depends on neutralizing pathogenic autoantibodies, cytokines, chemokines, receptors. There is the Fc-dependent portion that inhibits phagocytosis, blocking the access to Fc gamma receptor, inhibits antibody-dependent cellular toxicity, and modulates the half-life through FcRn. And then complement inhibition by neutralizing C3A, C5a, and reduce complement deposition. But besides that, it has a strong effect on the cellular immunity it's unappreciated. It can affect the T cells. It boosts regulatory T cells, suppresses autoreactive T cells, and affects the phenotype of CD4 T cells. It can have an effect on B cells. It seems that it does affect and suppress autoreactive B cells, and can also affect antigen-presenting cells, like dendritic cells, by regulating their maturation and function, and macrophages. But besides that, it seems that there are some other molecules inside the IVIG product that go through the process, like chemokines, cytokines, receptors, that could be also contributing to the immunomodulatory effect. So IVIG, it has multiple mechanisms across diverse immune pathways. It's effective for a wide range of autoimmune conditions. It's fast-acting. So we could say it's kind of similar to steroids. But compared to steroids, it's immunomodulating. It's not immunosuppressant. So therefore, it has limited systemic toxicity. So given the fact that it's so close to steroids, but it has a safer profile, what is the role of IVIG in the treatment of inflammatory myopathies? So Dr. Ceribelli already talked about dermatomyositis, where we have the most robust data, and we have the first successful clinical trial in myositis, where it showed that it was quite effective for patients with dermatomyositis defined by antibodies like TIF1 gamma, MI2, and XP2. But what about the other subtypes, like antisynthetase or immune-mediated necrotizing myopathy? And I'm not going through the IBM data where Dr. Schmidt before saw that it might have also some signal for dysphagia in IBM. So starting with antisynthetase syndrome. Starting with a case. This is a previously healthy 25-year-old female diagnosed with GWAS antisynthetase syndrome based on Raynaud's, fevers, weakness, lung disease. The patient was treated initially with steroids, as we do for all patients, at the same time starting a steroid-sparing agent, mycophenolate. However, this was not enough, so added rituximab. The patient continued to have active disease and added tacrolimus. So the patient was relatively stable, but a few months later developed worsening shortness of breath, fevers, and now a CPK of 15,000, worsening muscle weakness. So the patient has a flare of her disease. So at this point, what do? Like the patient is clearly like in a very impressive immunosuppressive regimen and still is not controlled. So the question is, would you add IVIG for this patient? And what is the evidence of adding IVIG in a case like this? So I'm going to reference two case series that are relevant to this. Both of them are retrospective studies, one from Hopkins, where 17 patients with antisynthetase-associated lung disease were treated with IVIG, and they showed improvement in their FVC DLCO and as well decrease in the prednisone dose they're on. So not only was effective, but also had a steroid-sparing effect, which is quite significant for our patients. And the other one is a recently published study, actually from Portugal, a much larger study of 69 patients. Now, this included all patients with myositis-related ILD, which half of them had antisynthetase. And they also saw improvement in FVC, DLCO, and they saw there was a very good effect when combined with mycophenolate or rituximab, so without increasing the safety signal. So from this, it seems, retrospective studies, that it might be some effect in the pulmonary function testing. And how about the ProDerm trial? Dr. Ceribelli also showed before that there were some patients with antisynthetase antibodies included in the study. Now, the goal of the study was not to study lung disease, but if you see the data, all of the patients, they had some lung function symptoms as indicated by MDAT. And when they did the post-hoc analysis for the pulmonary symptoms, they saw initially 36 patients out of the 95 had VAS, pulmonary VAS more than 0.5, which means they had pulmonary symptoms. But at the end of the 40 weeks of the trial, the percentage decreased to 23%. And at the same time, the VAS score decreased by 1.115 centimeters for patients on IVIG compared to 0.17 centimeters for patients on placebo, again showing a significant difference between the two groups. And this is not, the study again was not designed to study the effects of IVIG on the lung, and we don't have a specific number of PFTs, but again, it's a good indication that probably has some good effect on lungs as well. And that is also reflected on the recommendations published by the 23 American College of Rheumatology and CHEST guidelines on ILD in patients with rheumatic diseases, whereas you see IVIG is not reflected in the other diseases like scleroderma or rheumatoid arthritis, but it is included in the recommendations for ILD associated with myositis. And this is more evident in rapidly progressive lung disease, either MDA5 or not, where IVIG is one of the first medications that can be used for rapidly progressive ILD, especially in patients that there is risk for infection. And this is quite significant for our patients because these are quite sick, very immunosuppressed patients, and there's always the risk, we're always afraid of infection. So now, moving on to immune-mediated necrotizing myopathy, we start with a case. This is a 63-year-old male with hyperlipidemia, uncontrolled diabetes, hypertension, classic metabolic syndrome. He presents with proximal upper weakness of 4 out of 5, but significant weakness of the lower extremities, like hip flexors 2 out of 5, and CPK 9,000. The patient was diagnosed with statin-associated anti-HMG-CR. So we have to treat this patient. But this is the patient with high comorbidities, uncontrolled diabetes. The question is, would you add steroids to treat this patient? Or is there any other way that we can treat him without getting him worse? And that is what is the evidence behind IVIG monotherapy, and this is based both on published data and personal experience. The first report was this case series of 3 patients with Dr. Andy Mammon, and the first patient that you see is the case that I presented you before. So these were 3 patients with HMG-CR myositis, but had contraindications to corticosteroids. Most of them had diabetes, and it was very difficult to add steroids. So they were treated with just IVIG. And as you can see here, like the first patient, the case I presented before, within 3 months, the CPK decreased from 9,000 to 2,000. And similarly for the other patients, even within 1 month, you can see a significant decrease in the CPK. And also for the muscle exam, the patient before had 2 out of 5 muscle strength in the hips, which means that he was not able to lift the legs above the chair or the bed. But now it's 4-, which means not only he is able to lift them, but he is able to resist has some resistance to the examiner. And then the other patients also showed a significant improvement in the strength. But we also have a recent study published by Amato and Shu. These are 15 patients with HMG-CR myositis that were treated with IVIG monotherapy. All of them, they had a 96% decrease in CPK on IVIG monotherapy within 6 months. And then 67%, like 2/3 of them, they normalized their CPK within 7 to 15 months. And 77% had normal or near normal strength, which means 8 out of 10 patients went close to their baseline with just 6 months with only IVIG monotherapy, which is quite significant for this aggressive disease. And that is also reflected on the ENMC guidelines recommendations for treating of necrotizing myositis. IVIG is considered one of the first-line agents for treatment of HMG-CR myositis with steroids and methotrexate, but can also be used as monotherapy if there is some contraindication to steroids, and then second line for SRP. And then this brings me to something I'm very excited about. It's about our clinical trial, MIGHT. This is a phase 2 clinical trial based on the hypothesis that IVIG is effective in HMG-CR myositis. And our goal is to measure the clinical outcomes, but also immunologic markers of the disease pathogenesis. This is an NIH-sponsored trial that's supported by Octa Pharma, and it's co-led with my co-PI, James Andrews from University of Alabama. So this is like a randomized placebo-controlled trial. The patients are divided 1-to-1 to placebo control for 3 months, receiving IVIG versus nothing, and then 3 months open label for all of them receiving IVIG. And the IVIG will be given at home, and this is the first placebo-controlled trial conducted through home infusion delivery where everything is blinded. So by the end of 3 months, the outcome that we are measuring is change in CPK, which I showed you before in the case series. There is a significant decrease in CPK within just a few months. So we are able this way to keep the trial short and able to measure a change in CPK. But we're also going to measure immunologic markers of disease, like percent change in number and phenotype of HMG-CR specific CD4 T cells, hoping to shed some light into the pathogenesis of HMG-CR, but also how IVIG might work in this disease. This is a multi-center trial, and going from top right, it's with Rohit Agarwal from University of Pittsburgh, Dr. Lisa Christopher Stein from Johns Hopkins University, and Dr. Michael Weiss from University of Washington. So it's a multi-center study, and we're recruiting patients with HMG-CR myositis. So in conclusion, IVIG can be used as first-line treatment for other diseases besides dermatomyositis. It can be used for HMG-CR necrotizing myopathy and even as monotherapy because it's quite effective. Can be used for rapidly progressive lung disease in combination with steroids and other medications. Can be used as second line for other forms of immune-mediated necrotizing myopathy that have not responded to first line. It can be used also for lung disease, like in severe antisynthetase syndrome, because it does have a signal that it might improve the lung function. And then we have the specific indications. Since it's fast-acting, it can be used for situations where the patients are hospitalized and we need something to act fast, and the steroids are not always the answer. Patients with dysphagia, with respiratory failure. When there is concern for infection, again, our patients are quite usually sick, and there are many immunosuppressants. Or patients cannot tolerate corticosteroids. And I would like to acknowledge and thank all the partners for the MITE clinical trial, the MITE clinical trial investigators. NIH, this is funded by NR21, Octa Pharma, because it made our trial possible and feasible, and CSI Pharmacy will be doing the home infusions. And this is, and of course, all my collaborators and from the UT Houston Myositis program. So with this, thank you for your attention, and then we'll move to questions.
Thank you, Eleni. So I think we can open the session for questions. You can use both the microphones in the room or the QR code, so we can read your questions from the iPad.
Actually, I'm curious if there is time, like if people are using IVIG for lung disease, and this is something that you commonly do, or you're relying on something else.
So thank you very much for both your presentations. My question was more probably for Angela, but maybe you have some input also. So in your cases, you had some representation of paraneoplastic cases of dermatomyositis, and it's how, if I understood the protocol well, you did 6 months of IVIG monthly, and then how are you managing after those first 6 months the follow-up, especially in those patients where maybe you have some problems with immunosuppression? So do you prolong it? Do you reduce the dose? What are you doing with the IVIG after those 6 months?
Yeah, for paraneoplastic cases, we have to collaborate with the oncologist. So based on the therapies they do for the oncology part, we decide what to do. So as I told you, unfortunately, 3 out of 5 patients passed away. So in the other 2 patients, we are just observing them in the follow-up period. For now, there was no need to repeat a course of IVIG. So we are ready to repeat the entire 6 months course if needed, but the efficacy was okay for now, so we are just following up.
And you didn't have to do any maintenance kind of regimen afterwards?
No, not yet.
Okay, thank you.
So I'm reading one of the questions. It's for me. For the 2 of 5 that didn't respond, why they didn't respond? So basically, there was no improvement of the skin disease and of the dysphagia in particular. So because the situation was really becoming severe, we decided to stop treatment. For the patient who didn't respond for severe skin manifestations, we are currently asking for compassionate use of a biologic agent, which is anifrolumab, so we don't know yet. We have to ask for permission. While for the patient with the severe dysphagia, we are currently thinking of like palliative care.
There is another question about, yesterday we had presentations on JAK inhibitors. What is your perception for the future? For me, like, I have used JAK inhibitors off-label for very severe cases, and for all of the cases it has been in combination with IVIG because they were severe cases, and actually with the combination they did well. from my experience.
In our center, we used JAK inhibitors for 3 cases, again, for very severe skin disease, and they were working well. And again, it's a compassionate use of label, so we have to ask for each patient if the possibility to use them.
So I have a question about the IVIG trial in the IMNM patients. The first 12 weeks of placebo IVIG trial. So the patients who are on placebo, will they receive any other treatment at all or completely placebo?
Yeah, so that's a great question. So initially we wanted to do IVIG monotherapy versus placebo, but then that is very difficult to recruit. And the thing is, we know that IVIG is working. That's why we allow, there is a background therapy with 1 immunosuppressive agent and up to 20 milligrams of prednisone to cover for these 3 months. But ideally, we would like to study monotherapy versus placebo.
Okay, one question for me. Angela, what were the pre-treatment of your patients? Do you identify groups? Not really. So the pre-treatment was very heterogeneous because there were patients coming from different types of disease. So we were not able to identify groups based on clinical or serological status. Okay. So it's very difficult to understand if there's a subgroup which may be more responding to IVIG, maybe if we start early. So in our experience, we couldn't do that.
I have to say there have been some a trial in malignancy about using IVIG because there is a thought that maybe there are some these antibodies against cancer, cellular anti-malignant antibodies, and it was safe and showed some only mild improvement. That's why it's not been used, but it might be even beneficial for cancer in the end. So the question is, what is the optimal washout period for IVIG, especially for trials where there is a switch from IVIG to another treatment? That's a very good question. We had considered that actually if we would allow patients with previous IVIG treatment. I don't think there is a good answer. I would say 3 months to be safe. The question is, even if a few doses of IVIG have some permanent effect on the immune system, that we might then miss that. But I would say, like at 4 weeks, theoretically, it should be washed out.
Another question is, what is your typical treatment duration with IgIV in dermatomyositis and also in statin-induced IMNM? So in dermatomyositis, usually we do the 6 months course. Then we reevaluate the patient after 3 months, and every 3 months we evaluate the need for another course of IVIG or to a change in the treatment.
I think my practice is a bit different, but I think it's the difference between the US and Europe, like how much the access to IVIG. So I keep patients on IVIG forever, and actually I tend to taper first immunosuppressants, again because of the risk of immunosuppression, whereas IVIG is safer if the patient is okay with the infusions. But especially for statin myopathy, my experience is they remain on that for years. After they're stable for at least 6 months, maybe try to reduce the dose or increase the interval between. but there are cases that we cannot taper off the IVIG. But we have, I think, access to IVIG.
Yeah, sometimes we have problems with even like the space for the infusion. Yeah.
And I think the other benefit is that there are pharmacies that they do the infusions at home, so that is it's much easier for the patients than going to an infusion center.
Yeah, that's true. Yeah. Okay, so thank you for the questions. So Eleni will start with the closing remarks. Thank you.
Oh, sorry. So closing this, so we saw that the PRODERM trial demonstrated significant improvement in disease activity in patients with dermatomyositis versus placebo. And then such findings contributed to IVIG being approved for dermatomyositis in different countries. Retrospective real-world data from Ospedale Civili of Brescia with Dr. Ceribelli confirmed and showed that IVIG therapy can be safely and effectively administered to patients with dermatomyositis and active skin and muscle disease, and also can also be used in paraneoplastic dermatomyositis when they don't respond to other steroids and other immunosuppressive agents. Evidence from the PRODERM trial demonstrates that patients with lung symptoms can experience meaningful improvement. And retrospective data across different centers show that IVIG can be associated with improvement also in PFT markers. IVIG has been shown in real-world experience to be an effective first-line option and can be used as monotherapy in anti-HMCCR IMNM. And we have our ongoing phase 2 clinical trial is evaluating the role of IVIG in HMCCR. And we hope that we're able to uncover also the impact on immunologic disease markers, shedding some light into disease pathogenesis and also how IVIG works in this specific disease. So with that, we want to thank you for your attention, and the slides are available at the Octapharma Science Hub. Thank you.
Thank you. Oh my goodness, somebody put that under the table. I got it. I'll get it. You printed up all of them.
Just for us.
Terrific.
The others, I asked them if they want. They said no, but I think it's better to have it in paper rather than if something doesn't work on our laptops or so.
This is still our work.
Yeah. So that's the correct one. I should have printed these. Actually, now that you're saying that, is all correct except that Hannah is presenting on Jessica's behalf.
And so we'll present this, we'll do this together the way it was suggested. And then,
Which means you present Flores and I present her
yeah.
mom.
And then you want to do Hannah, I'll do Meredith, you do Angelo?
Okay.
Is that okay?
Is that okay? Yeah.
I think the weather's too nice and nobody wants to come back inside. I went down for lunch to the water.
Ah, nice.
And just sat out there. It was so beautiful.
So close to the river, it gets a really nice landscape view with the bridge, and also at night. At night is super nice.
Now, is that a river coming into the sea, or is
Yes.
it still an inlet from the sea?
No, so it enters in a part of the city and then it will land on the sea, but is on the other side of the bridge. Have you seen the Christ, the statue of the
Yeah.
Christ? The other side is the sea.
Right, I know
Yeah.
from the map.
Yeah,
but,
yeah.
But this is considered a river.
Yes,
And it's
yes.
fresh water. It's not salt water.
It's fresh water, yeah.
Yeah. Okay.
Although in some areas below the other bridge, Vasco da Gama, it's already a mix between sea and river. But in this side, it's just river. Shall we start to be on time?
Yeah. Make sure everyone has enough time.
Good afternoon, everyone. May I ask you to sit, please? So it is a real pleasure to me to be here and present this session, moderating this session on juvenile myositis with Brian Feldman. I think you all know him. He's a pediatric rheumatologist from Toronto in Canada. And we'll have brilliant speakers towards this afternoon and also 3 oral presentations at the end. And without losing more time, we're going to start presenting Flora's and Flora's mom. And please, Brian, can you start with Flora's story? Unfortunately, this is going to be recorded because they cannot be here, but they will be present for questions if there's any questions for them. But we'll just introduce them briefly.
Now, do we need to start the slides ourselves, or will you start them? They understood. Do I need to go do something?
No, just
okay. So the speakers today are going to be Flora and Suzanne, her mother. Flora's story is one of resilience, determination, and purpose. She was diagnosed with juvenile dermatomyositis as a child and faced years of physical challenges and uncertainty. Today, she's an adult and in remission and pursuing her master's degree in social work. And her idea is to transform her lived experience into a source of strength and advice. And so by sharing her journey with JDM, Flora hopes to offer understanding, encouragement, and solidarity to others navigating similar paths.
Beside her every step of the way has been her mother, Suzanne, an unwavering advocate, caregiver, and source of strength. Through years of navigating the challenges of caring for her daughter, Suzanne has gained invaluable insights. She now uses this experience to guide and empower other families facing similar diagnoses, ensuring no one feels alone in their journeys. Can you put the slides, please? It must be a recording, so I think, can you press play?
Hi, my name is Flora. I'm 25 years old and I've been in remission from juvenile dermatomyositis since November of 2014. Something important to say up front is that I honestly don't remember a lot from my JM experience. A lot of those years are blurry for me, partly because I was so young and partly because I think I blocked out a lot of those memories. So what I do remember comes in pieces. The first thing I remember is the amount of medications I was on. There were so many over the years that I couldn't even tell you all their names now, but I do remember how some of them made me feel, especially the steroids. I remember feeling angry and frustrated a lot and being irritated by tiny things. I didn't feel fully in control of my emotions, which was really hard as a kid. I also remember the hospital stays for my IVIG infusions. I was often admitted overnight. The hospital room noises really stuck with me. The beeping, the machines, and the nurses waking me up what felt like every 30 minutes to check my vitals. It made sleeping almost impossible. JM also affected school and social parts of my life. My mom would talk to my class about what was going on and how they could help me. On the playground, I couldn't participate in a lot of activities, especially anything in the sun. I had to wear a hat and constantly reapply sunscreen, which I hated because none of the other kids had to do that. The medications also affected my memory and ability to grasp concepts, especially as I got older. In high school and college, I relied on accommodations like extra time for tests, distraction-free rooms, extensions on assignments, and flexible attendance. Without those, school would have been a lot harder. Even though I don't remember everything, there were definitely positives that came out of the experience. One of the biggest ones is increased empathy. I connect with people easily who've had medical issues or medical trauma because I understand what it's like to deal with something scary and confusing when you're young. It has also shaped my future goals. Spending so much time in the hospital settings influenced my desire to pursue my master's degree in social work. I hope to work in a hospital environment supporting people who are navigating their own medical challenges. Growing up with JM definitely shaped how I speak up for myself. I think it gave me confidence in my voice and made me comfortable advocating for myself, whether that's in a medical setting, school, or just life in general. One thing I remember very clearly is feeling frustrated when doctors would talk about me to my parents while I was in the room, but wouldn't look at me or ask if I had questions. I understand that I was young and they probably assumed I wouldn't get what they were saying, but I still think it's important to include kids in the conversation at whatever level is age appropriate. As an adult, I still have medical anxiety because of everything I went through, but I also feel very capable of advocating for myself. I know what questions to ask and how to make sure I'm part of the discussion. I also still get anxious going into any kind of medical setting, and I have general anxiety as well. Therapy has been really helpful in managing all of that. It's given me tools like breathing techniques, grounding, meditation, and journaling prompts that I use both for medical anxiety and everyday stress. It's also helped me start working through some of the memories I blocked out from the medical trauma in a way that feels safe. Since going into remission in 2014, one of the most meaningful things I've done for myself was getting a tattoo of my remission date in Roman numerals. I had seen other people with remission tattoos and really loved the idea of marking something that used to define so much of my life. I chose the placement very intentionally. It's on my left arm, right above the spot where I used to get poked for blood draws, IVs, and infusions. For years, that area represented pain, fear, and feeling out of control. Now it represents strength, growth, and everything I overcame. Getting the tattoo felt like reclaiming that part of my body, transforming a place associated with trauma into something meaningful and beautiful. Remission has also given me space to reflect on how far I've come. Even though there's a lot I don't remember, I know that I'm not the same little girl who is constantly in and out of hospitals. There's a sense of closure that comes with remission, but also a sense of ongoing healing. The tattoo is a reminder that JM is still part of my story, just not the part that defines me anymore.
Hi, my name is Suzanne Edison. I live in Seattle, Washington. I am a former therapist and had been a longtime CureJM volunteer before I became the mental health coordinator. I am newly retired from that position. My daughter Flora, whom you just heard from, was sick for 1.5 years before being diagnosed with JDM at age 6. She was in and out of treatment for 8.5 years before going into a full remission that continues until this day. I am married and was working when she was diagnosed. I had to quit my job to care for her. My husband had health insurance with his job and kept working. One is never prepared for the changes and challenges that come with caring for a chronically ill child with a rare disease. Here are some of the things I have learned along the way the past 19 years and hope will be of some use to you as you care for those living with JDM and their caregivers. It is a journey with many ups and downs for everyone. Caregivers need a good support system, friends, family, therapists, a belief or spiritual support network. Educate them about the possible ups and downs and encourage them to join CureJM or another support network. Finding at least one other family going through this journey can be a comfort and a lifeline for all. Ask caregivers how they are doing. Caregivers are the vessels and containers that help their kids manage feelings and experiences. But they too experience a range of emotions, sometimes over and over again. Grief, for example, is a process, not a one-time thing. Caregivers feel overwhelmed. They might feel anxious or depressed, certainly stressed out. They might experience some guilt or anger, frustration, and fear. And there are also times of joy, laughter, and a new normal. Encourage caregivers to practice self-care. Remind them that if their well is empty, they can't adequately advocate and care for their child. It is okay to ask for help and to make time for their own care. This can be a simple thing from taking a bath, a walk with or without friends, even time to read or listen to music. It is important to remember that a rare disease diagnosis changes lives for everyone in the family. But their child is still a child and wants to be seen as more than their disease. Trust and listen to caregivers. They know their child best. They are part of the team. Believe them when they say their child seems more fatigued or in pain. If certain medications are having terrible side effects, like steroids, do try to find other options. In addition to the disease itself, the medicines have a huge impact on many things in a kid's life, as you just heard from Flora. It is not all in their heads. JDM involves a loss of control. Encourage caregivers to give their child choices to hand over some control. For example, when and where would you like your methotrexate injection on your body? Not, do you want it? As they get older, give kids a medicine chart to check off. Help them learn what meds and dosages they take as they begin to move toward adulthood. This helps them prepare for transition to adult care. Transition to adult care is important to discuss and often overlooked. We know, for example, there's less time in appointments. There will be no parents or caregivers to answer questions. Maybe the adult rheumatologist has little experience with the juvenile form of dermatomyositis. And you can help kids and families prepare for this through their teen years. It can happen gradually. Remember, mental, emotional, and physical health are not separate. Everyone needs and deserves attention to their whole person. Thank you.
Thank you, Flora and Suzanne. They are online, so if there's one question for them. If not, thank you for sharing your story. You're definitely courageous people, and we hope you, Flora and Suzanne, they are in a good moment now. We really hope so. Thank you for sharing. We are now moving for our first speaker, Hannah Kim. Hannah is a pediatric rheumatologist and physician scientist at the National Institute of Arthritis, Musculoskeletal and Skin Disease in Bethesda, and she's going to talk us about genomics in JDM. Hannah, the floor is yours.
Hello. I'd like to thank the organizers for the opportunity to give this talk. Here are my disclosures. As we know, JDM pathogenesis is complex, and it includes genetics, environment, and immunodysregulation, but I'll just be focusing on genetics today. Genetics in general can be described along two axes: effect size along the left, large and small, and then how common a gene change or variant is. Is it very rare or more common? The typical conditions that we think of as genetic and having like autosomal recessive or dominant inheritance are typically rare, very rare alleles with a very high effect size, so high on the effect axis and low on the allele frequency axis. And then for complex diseases where genetics is just a component of what's causing the disease, such as in JDM, the variants observed tend to be more common in the population, but less effect size per variant. And this is typically assessed by genome-wide association tests or studies. In idiopathic inflammatory myopathies, or IIM, including JDM, HLA risk factors have been what's most consistently and most significantly found. You can see on this Manhattan plot on the upper left, there's a high peak at the MHC locus in chromosome 6. And this HLA haplotype, including class 1 and class 2 MHC, is generally overlapping with the ancestral haplotype. And specifically in JDM, DRB1, O301, and C0202 have been validated. And this work has been done by many large studies, including the Myositis Genetics Consortium, initially by Fred Miller. And Claire Deacon and her colleagues had looked specifically in JDM and found in DRB1, O301, in position 37 to be very important in attributing this JDM risk. And then Rothwell et al. In 2019 also found, interestingly, for anti-TIF1, that juvenile in onset, this DQB1, O201 is significantly associated, but actually a different DQB1, O202 is associated with adult onset anti-TIF1. So within this MSA group, there's differences between adult and juvenile onset. Another important genetic risk factor that's been described is the gene copy number in complement 4, or C4. So these graphics show the gene copy number distribution in controls and then in JDM. So each bar represents a different gene copy number. And C4a is an acidic subtype, and C4L is a long subtype. And you can see for the JDM distribution, it's overall shifted to the left, so that there's a overall lower gene copy number, and you can see this particularly for the red bars, which are a low copy number, that the bar is higher, like in JDM versus control, so they have more frequently a low gene copy number. So overall, there's a decreased gene copy number of C4 or C4 deficiency that's associated with JDM. And this same group, Zhao et al., had done a multivariate logistic regression looking at HLA class 2 DRB1-03 and C4 deficiency together to see if they are both independently contributing to risk for JDM and other IIM, particularly because we know that they're close to each other physically on the chromosome 6. And with doing this logistic regression in JDM, they found actually only the complement deficiency, their gene copy numbers remain significant, not the DRB1-03. And this was true for both DM and JDM, but it was different for PM and IBM, where the HLA was still associated independently. And then, of course, there are many other genetic risk factors that have been described beyond the HLA region. You can still see that high peak around chromosome 6, but there are many other genes that have been associated. This list is just meant to show that these genetic single nucleotide polymorphisms, or SNPs, range the chromosomes from 1 to 22, and the vast majority of them are not exonic. Only these ones highlighted in red, these 3 are exonic, but most are intronic or outside of the genes or in between genes. And these associations have been seen either in IIM overall or in JDM alone or in a different subgroup, and many of them also overlap with other autoimmune diseases like rheumatoid arthritis or SLE. And for some of these genes that have been associated with these SNPs, they have functions that relate to biological plausibility for what's been described with pathogenesis in JDM, such as TCR and BCR responsiveness, T lymphocyte differentiation, and interferon signaling. So as you may know, there are systemic autoinflammatory interferonopathies that have some phenotypic overlaps as well as their interferon score overlaps. But this led to this next project looking at the genetics between these conditions. So we used a novel approach to looking at genetics in JDM sequencing. We looked at trios, so the JDM patient and both of their parents, as well as siblings in JDM by MSA group, and then also in undifferentiated systemic autoinflammatory disease. We were interested in comparing their HLA haplotypes, as well as looking at the non-MHE SNPs that have already been described in IIM, as well as using the typical rare disease model approach of looking for high-impact rare variants, which hasn't previously been done. And we also were interested in assessing for any combined burden of any rare variants we found with HLA and JDM. For the undifferentiated systemic autoinflammatory cohort, they were defined by having features of autoinflammatory disease such as unexplained recurrent or persistent fever, elevated inflammatory markers, rash, or other unexplained organ-based inflammation. And excluded were any suspected malignant process patients, those who had a genetically defined SAID. So for example, people with proteasome mutations and Kendall syndrome were excluded, and any documented myositis or elevated CKR aldolase patients. So an overview of our methods were that we did whole exome or whole genome sequencing in anti-TIF1 JDM, anti-NXP2 JDM, and some other MSA status JDM patients, as well as SAID patients, and then the parents of all of those patients and some siblings of JDM patients. We did HLA frequency analysis in an expanded JDM cohort, and also transmission disequilibrium testing in an expanded cohort of JDM patients. And then we did a burden analysis on the same cohorts as the whole exome and whole genome sequencing patients. So overall, as far as demographics, there were no significant differences. As far as myositis-specific antibodies, we were able to do testing for this in a subgroup of the SAID patients, 20 of them, and they were negative for any antibodies, and that was really the only difference that we found between these cohorts. As far as JDM disease, as is typical for a rare disease genetic model approach, A more extreme phenotype is more likely to have a high yield in finding a rare high impact variant. So we tried to select for a more extreme phenotype with a lower age of onset and increased signs of severe disease with increased number of hospitalizations, use of devices, and skin ulcers for the JDM patients that were included for the whole exome and whole genome sequencing versus the other patients that were just used for HLA studies. So first looking at the HLA class 2 haplotypes, we were comparing their frequency in JDM patients, specifically looking at anti-TIF1 patients and anti-NXT2 patients versus SIAD patients. So the haplotypes are listed on the left, and then the blue dotted line is the anti-TIF1 odds ratio and 95% confidence interval, and the purple dotted line is anti-NXP2 odds ratio and confidence interval, both versus SIAD. This first haplotype is the ancestral haplotype, including the DQB1-0201 that was previously described to be higher in juvenile anti-TIF1, different from adult onset anti-TIF1. So this first haplotype, H2 and H3, both had increased odds in anti-TIF1 versus SIAD, as well as this H7 listed at the bottom. This H7 was overall just a very rare haplotype, but it was significantly increased in anti-TIF1 versus SID with a high odds ratio, but the range was very large because of its rare incidence overall. And then these bottom two haplotypes, H4 and H5, were increased in anti-NXP2 in purple versus SIAD. So each of these haplotypes is completely different, and it's just the class 2 haplotype. Then we did transmission disequilibrium testing, taking advantage of having the sequencing information, or this HLA haplotype information, for both the parents and the JDM patient, as well as siblings. And this answers, or asks the question, does an affected child, JDM in this case, inherit a particular genetic variant, in this case the HLA haplotype, more often than expected by chance, or 50%? And how does that compare to unaffected siblings? So when we did this, we were looking at what are the odds of inheriting this particular haplotype from a parent for each JDM patient, separated by anti-NXP2 in this case in purple and anti-TIF1 in red, versus by chance. So we saw for H2 and H3, which we previously saw were more common in anti-TIF1 versus SIAD, to be increased odds in JDM patients versus by chance, and the bottom two haplotypes, H4 and H5, to be increased in anti-NXP2 patients versus by chance. And these odds ratios became more significant when they were combined, H2 and H3 together for anti-TIF1, and H4 and H5 together for anti-NXP2. And this information was supported by what we saw from this sibling TDT as well. Next, we looked at all the SNPs that were not in the MHC region that were previously reported, and generally found that they were not differentially present between JDM and SID. We limited this to just a whole genome sequencing, since so many of the variants were outside of the exons, but greater than 95% were not significantly different between these two groups. Next, I'll walk through the algorithm or typical pipeline that we used for finding candidate variants, candidate rare variants. So we started from all variants in all genes, and next we excluded any HLA variants or any unknown or uncharacterized genes. And then we focused on missense changes, which change the amino acid, because they're more likely to be damaging, and loss of function variants, which could be within or outside exonic regions, but likely to change function. And then to focus on variants that are more likely to have a high impact, NOMAD is a database of healthy individual sequencing, and you can check for any variant you find to see if it's there and how many individuals it's found in. And in general, for this type of analysis, if there are less than 20 homozygotes in that large database, then it's more likely to be of high yield or high impact. Allele frequency, or MAF, of less than 1%. And this is different from genome-wide association studies, where you're typically focusing on variants that are greater than 1% or greater than 5%. So this resulted in 905 variants in 413 genes for JDM versus 1333 variants in 573 genes for SAID. And then, yes, we also noted that there were no variants associated with known SAIDs, such as proteasome mutations associated with Kendall or TMEM173 mutations associated with SAVI in the JDM patients. Then we were interested to look at, were there any genes that had at least 2 patients with variants that had zero variants, in the SAID patients. So this led to a list of just 9 genes, and these fell into 2 categories you can see on the right: costimere structural proteins and stem cell renewals. And when we did an enrichment analysis of all the candidate genes that we found, one theme was structural muscle, costimere, myofibril, and sarcolemma in the purple box. We know that the sarcomere is like a critical subunit in the muscle. It has like titin, myosin, and actin, and it's very important in contractile function of the muscle. And then the costimere is what connects that sarcomere to the muscle cell surface, or sarcolemma. So it's very important in lateral force transmission and overall function of the muscle. So seeing this theme, we came up based on literature, a structural muscle gene list. And then the other things that came up in enrichment analysis are these MP categories, which stand for mouse phenotype. So that means like when those genes are altered or mutated, that's the phenotype that's seen in a mouse model. And it happened that all of the mouse phenotypes that came up were muscle-related. So we created a second list in this muscular phenotype from all of those mouse phenotypes, enriched categories that came up, as well as the human equivalent, all of the genes that can cause human muscular dystrophy. And those are all put into this muscular phenotype list. And we saw both for the structural muscle gene list that they were more frequent in JDM patients, on the left in red versus SID in gray, and similarly for muscular phenotype genes. And because of this muscle theme, we were interested in further modeling some of the muscle variants. And just to review kind of the function, we know that titin, which is this long purple left to right protein with coils at the ends, is the largest human gene. It has multiple types of domains. It's very critical in the structure, elasticity, and recoil of muscle, and important both in longitudinal and lateral force transmission, and interacts with many other proteins, including nebulin and obscurin. Nebulin is closely aligned with actin in the thin filament and maintains its length and stability, while obscurin in the middle is a structural bridge for the sarcomere and is also connected to the costimere and transmits lateral force. So this model looks at 2 particular variants in obscurin, which is modeled in the tan ribbons at the top, near its interface or interaction with titin, which is represented by solid white spheres on the bottom. And for one of these changes, phenylalanine to tyrosine, it changes from hydrophobic to hydrophilic. For another change, it changes from cysteine to glycine, which results in a broken disulfide bond. And both of these changes would be expected to change the secondary structure. And that could have effects both intramolecularly or within obscurin and intermolecularly between obscurin and titin. And we would hypothesize or believe that it's likely that these could affect their ability to function in maintaining structure and transmitting force. So these types of changes were found not only in obscurin, there are other changes seen here, but in nebulin and titin at the bottom. And because all 3 of these proteins are so integral and interconnected in the sarcomere, and there might be 1 or more variants present at once, we believe that there is potential for disrupted mechanical function because their function together is also important in the contractile function of the sarcomere longitudinally, and also how it transmits force through the costimere to the surface of the cell. And together, those things might impair muscle function and increase potentially the risk for injury. So these pie graphs show the distribution of each of these 3 categories of gene changes: structural muscle, muscular phenotype, and stem cell renewal. They're each in different colors, and there's also colors for the overlaps between these lists. And you can see overall that the JDM pie is larger because it has more of these genes. Additionally, when comparing JDM and SIID, you can see that the curve for number of genes per patient with JDM is shifted to the right. So most patients with JDM had 2 or more burden genes. And burden gene is like a change in any of these 3 categories: structural muscle, muscular phenotype, or stem cell renewal. Whereas the SIID patients most likely had 0 or 1 burden gene change. And then finally, we did a burden analysis, which looked at each of the gene changes by these categories and combined the haplotype information that we had. So when looking at what are the odds of all JDM patients having any muscular phenotype gene change, there are increased odds, and that was significant. And also for anti-NXP2, adjusted by the haplotype 4 and haplotype 5, there was an increased odds, and that was significant. And then looking by stem cell gene changes, this was significantly increased odds for all JDM patients, and also for anti-TIF1, adjusted for those haplotypes that are associated, 2, 3, and 7. And then overall, for any of the burden genes from the 3 categories, this was significant for all JDM and each MSA group adjusted by haplotype. So in conclusion, HLA haplotypes, including DRB 0301 and C0202, have been associated with JDM. And in our research, we confirmed that DRB 0301 is associated with anti-TIF1 JDM, with distinct class 2 haplotypes associated with anti-TIF1 and anti-NXP2 JDM. There have been multiple common variants or SNPs associated with IIM, including JDM, by GWAS studies and immunochip, but we generally found that these are not different between JDM and SIID. And by whole exome and whole genome sequencing, comparing JDM and SIID, JDM has an increased burden or increased frequency of these categories of gene variants: structural muscle, muscular phenotype, and stem cell renewal genes. And the structural 3D modeling we've done in sarcomere muscle gene variants indicates they may be destabilizing, affecting their ability to transmit force laterally and longitudinally, increasing the risk for injury. So I'd like to acknowledge that this was led by Rafael Gobak-Monsky and Adriana Almeida de Jesus Rashid and her group in NIAID, and bioinformatics there, and then in NIEHS, Dr. Lisa Ryder and her group, including biostatistics and computational modeling, as well as the Oklahoma Medical Research Foundation, who did MSA testing. And thank you for your attention.
Thank you so much, Hannah, for being on time. Questions for Hannah?
Hannah, I might just start. One of the genes that you showed us that has variants in JDM is the Titin gene. When we think about this as an immunological disease, as opposed to the structural homology that you're talking about there, when we think about it as an immunological disease, is there any homology with any viral agents or other things that might indicate why variants there might lead to JDM?
No, it's a good question. I think we haven't looked at specific homology with exposures such as viruses.
I think there's a question from...
Hannah, I was just wondering if you wanted to conjecture about whether you think these muscle variants are modifying disease risk by like increasing susceptibility to like muscle injury at all and like autoantigen exposure, or like what do you think is the pathomechanism connecting them to like an autoimmune process?
Yeah, it's a good question. We haven't done any follow-up studies to definitively prove anything or even really discuss it in detail, but I think personally, my hypothesis aligns with what you're thinking about. Like, we think there might be an increased risk of injury, and that might increase autoantigen exposure, and somehow maybe that increases the risk, but we don't know for sure.
Another question from Lucy.
Hi, Hannah. Lucy Wedderburn from London. It's really interesting new data. I think for the GWAS replication, you wouldn't have seen it in 50, because we didn't see it until we got to 400 or 500. But the other way around, have you thought of ways that you could validate those new findings? Either have you looked in a very large control cohort? Are those at similar frequency in healthy population? Or perhaps another way, other data available in whole genome, or even if there are tagged SNPs for that titin and the other genes in the GWAS? Don't know. It's a sort of broad question. How could you validate this, really?
No, it's a good question. I mean, yeah, I think this is kind of an exploratory project, and
sure.
Yeah, there's probably many ways to validate it, as you explained, but we haven't done that yet, but that's a good point.
I think there's lots out there you could use already publicly available, maybe. Yeah.
Can I also ask a question? It's more a curiosity. Did you study other antibodies, other specific antibodies other than NXP2 and Tifon gamma, like Me2 or others?
I think there was one or two that had anti-MDA5, but the most common were anti-TIF1 and anti-XP2, so that's why we focused on analyzing those.
Okay, wonderful. Hannah, thank you very much.
Thank you.
Our next talk is being given by Dr. Meredith Wilkinson, is an Arthritis Research UK Career Development Fellow at UCL, Great Ormond Street Institute of Child Health. Her research focus is on immunometabolism in dermatomyositis. Mary, take it away.
Thank you so much for inviting me to speak today. Hopefully, I will be giving you an overview of RNA sequencing and that it will become clear as to why it's being used and the advances into sort of spatial declarations. So as I said, I'm going to go through bulk RNA sequencing, single cell, spatial, and then sort of conclude with some data that we've generated in our lab. So what's the differences? There's a lot of different images you could use or analogies, but essentially bulk sequencing is everything in your tissue, and it's difficult to differentiate between the different cell types. Single cell is obviously you're taking that tissue and sorting it into the different cells prior to sequencing. And then spatial is being able to see each cell and their transcript in situ in the tissue. So for bulk RNA sequencing, you take your tissue sample, whether that's blood, skin, muscle in 4JDM, and then you isolate your RNA, generate your libraries, sequence, and do your quality control and then data analysis. And this is generally, we're looking at differentially expressed genes and pathways. So there's been quite a lot of studies that have used RNA sequencing as a technology, and we're getting more and more data. This is just some examples that have been in both blood, skin, and muscle. To highlight Jessica Turnier's paper, which was looking at tape strips from the skin and bulk sequencing. And this identified differences between lesional and non-lesional skin, highlighting these different pathways. But also what's interesting that sort of confers with my data, and there's been a lot of discussion about mitochondria at this meeting, is the fact that even in non-lesional skin, you're seeing a mitochondrial signature, not for everything, but it's interesting that potentially it's more of a global tissue response. So from sort of the studies at home, we can see that bulk sequencing can be done in the peripheral blood, muscle, and skin. One of the sort of main findings that we see across a lot of different techniques is this upregulation of type 1 and type 2, but also a spectrum for an individual patient and the different tissues. And again, we've got this mitochondrial dysfunction that's evident across the different tissue types. So moving into single-cell sequencing. So as I said, this is taking your whole tissue and sorting it into droplets for the different cell types, then continuing for the cell lysis, isolation, cDNA amplification. And the difference here is that you're able to define the individual cell types and cluster them based on the comparators, so between sort of the control and the patients, or over the maybe naive to on treatment. So there's been more and more papers in this area, both in adult and juvenile. To focus in was this particular paper by Chen et al., which looked at comparing muscle and the peripheral blood. So for this, they had 1 healthy control and 2 JDM muscle biopsy samples, which they sequenced. And this shows sort of the different clustering of the different cell types. What their sort of main findings were was the CD74 positive smooth muscle cells, and this was associated with an inflammatory state. The CCL19 positive fibroblasts, and again, these have this upregulation of the type 1 and type 2 gene expression. And then one thing, obviously, that single cell helps us with is looking at ligand receptor interactions, and this showed this interaction between the immune cells, the CD74 positive, and fibroblasts. And this was sort of the first example where this could be used as more sort of a reference to support further studies. Then we have the immune cell single cell paper that came from Jessica Neely and the San Francisco group. So they took patients naive of treatment and on treatment, looking at disease activity and looking at the sort of networks between the cells. They used CytSeq, so that's also looking at protein expression. So they were able to map the immune dysregulation in JDM. They identified transcriptional changes in B cells, which have also been seen in other studies by flow cytometry and other techniques. CD4 positive T cells, and these persisting with treatment, these changes. Again, the interferon type 1 response, and this was parallel to impaired cell death in cytotoxic immune cells. And also the proposing new therapeutic strategies to restore these balances in immune cells. And then I've been very privileged to share with you some of this very new data that's currently under review, so please don't take any pictures. So this is led by Jessica Turney and Jessica Neely. This is a big collaborative project that's funded by the CZI in collaboration with CureJM. And so this is a pilot study from going to be a much bigger study looking at single nuclei transcriptomics of muscle. So this identifies that across the different sort of cell types that's seen in muscle, we have an upregulation of the interferon score, and specifically highlighting endothelial cells, macrophages, and satellite cells. And then this second plot identifies that the top interactions in muscle was between endothelial cells and macrophages. And all of this is sort of showing that we've got impaired vascular remodeling and muscle regeneration in JDM. So we're now moving on to spatial transcriptomics. And we've seen examples of this across the conference, mainly in adult, but this is obviously going to be in juvenile. So there's sort of 3 different approaches to spatial. So the first is sequencing-based, and generally this platform uses 10x Visium. So this is actually sequencing-based. It takes the tissue on the slide and uses library construction to be able to read the sequence within sort of this in situ. The original version is not quite single cell. The new, more advanced version now is based on a grid rather than a circle, so is much higher resolution and could be perceived as more single cell. And in the case of muscle, probably a bit broader. Then we have the probe-based methods. So this is using probes and fluorescent markers. And this is a technology called geomics, which I'm going to give you a bit more detail on. So again, this is more looking equivocal to bulk sequencing. It uses regions of interest and then looks at the RNA probe count from each of those regions. And then we have imaging-based. So we have two competing technologies, 10x Xenium, and also nanostring Cosmex, which are quite similar and just have different numbers of genes that you can look at, but essentially it's using imaging in specific fields of view from the tissue. So this is an example of some of the papers that have been published recently. So there's Visium technology is being used by two different groups, one in Hong Kong from the adults, Myositis looking at MDA5, and then there's the group in France led by Rodero who used again Visium in JDM and sort of showed that they had biopsy sample from a patient who'd gone into remission and looked at the sort of differences in muscle biology. We've recently published, and I'll show you some more data on this, looking at geomics, and so the sort of more bulk sequencing within spatial. And then we've actually heard and seen quite a lot from Andy Mammon's group, and looking at a specific gene set in adult myositis using the Xenium platform and specifics of gene set that they've derived from bulk sequencing, which, just to state, transcriptomics works very well when you're validating a gene set that you've already defined from bulk sequencing. So moving into the data that we've been generating in our lab. So this is from the published data, and this is all very much led by Antisynthetase, who's a PhD student in my lab. So we took 3 JDM biopsy samples. 2 were TIF1 gamma, 1 was negative MSA, and 3 controls, age-matched controls. So this was using the geomics platform, where you hybridize with the probes, and then you're sort of using the spatial profiler to count and collect those oligos. So the pipeline was developed by Aris to be able to extract the counts, and then through a series of pipelines, we were then able to look at differential gene expression analysis and downstream beyond that. So as I've sort of alluded to, we're looking at regions of interest. So if you take the geomics, we had 28 areas selected from 3 muscle biopsies of the treatment-naive JDM patients and 3 controls. And this is looking at the whole transcriptome analysis. This isn't selected genes. These examples of some of the regions of interest. So we have control, which majority is myofibers. This is a JDM area where it was very much selected to be majority myofibers. Then we have areas where we've got CD45 staining, so we're looking at lymphocyte infiltrate. And then we were specifically interested in CD68 macrophages because we'd seen mitochondrial signatures within monocytes in circulation. So when you do principal component analysis to look for sort of separation, it's quite clear that there's differences between patients and the controls, but also between macrophage areas and the muscle-specific tissue areas. The macrophage areas are just in the JDM, as I'm sure you're aware. The immune infiltrate is very minimal in control healthy tissue. So concentrating on the specific regions of interest where it is muscle cells specifically, when you do differential gene expression we had 448 differentially expressed genes when you compared the JDM to the controls. And then when Aris did pathway analysis, the main pathways that were upregulated, to no surprise, was the interferon pathways, and the ones that were downregulated were mitochondrial related. Something that gets used quite a lot in myositis is an interferon gene score. So Aris calculated a score based on 15 published genes for interferon. And what was striking was the upregulation of these genes across all the different regions of interest and thus tissue sort of types compared to controls. But interestingly, we had 2 patients that had this upregulation across the different tissues, but 1 patient that was above controls, but quite different from the other 2 patients. And when we looked into that, the patient that has the less interferon also has a negative MSA. So this is something we need to explore a bit further, looking at MSA subtype and how this sort of profile looks. When Aris did this, calculated a score for mitochondria, and this was 41 genes that were differentially expressed. Again, control versus JDM, we have this downregulation of mitochondrial function. Across the different tissue types, markedly lower in CD68. But then when you look across all 3 patients, we've now got a fairly universal downregulation of mitochondrial function compared to controls. So this seems to be independent of interferon. Aris validated this against a previously published bulk RNA sequencing from JDM muscle. And this was looking specifically at the mitochondrial encoded genes, which were unfortunately not in our genomics data set. Mitochondrial genes are a little bit tricky and people don't like them, but anyhow. Validation from this sequencing identified that there was a downregulation, and quite strikingly, of the mitochondrial encoded genes compared to controls. And as I said, with the fact that we have this 1 patient in the genomics data set that had lower interferon, but still striking mitochondrial dysfunction, when we did validation in immunohistochemistry, staining for MXA for type 1 interferon and COX SDH to look at mitochondrial complexes, there was no correlation between the 2. So mitochondrial function could be independent of interferon. So this work is published. I'm now going to talk about parallel work that isn't published, but is using the technology of the CosMx platform. So rather than having the full transcriptome, CosMx first developed their platform using 1,000 genes, mainly for tissue typing. And this is image-based using antibodies, so you can segment at a single cell level. I thought it would be useful to highlight that it's not easy. Aris can tell you a lot more about this than me, but when you do these technologies, you don't start potentially knowing how to do the analysis. And one of the things that's important, especially for rarer diseases, is having a reference data set. And there isn't really a single cell data set that can be used at the moment to sort of for a reference set for this analysis. So when Aris did unsupervised clustering, the cell labeling maybe isn't as useful as we want, and thus being able to interpret it is a little bit harder. So Aris looked at the literature, and he identified a single cell data set in IBM, and used that to infer how to do supervised clustering within our data set, which came up with cell types that were more interpretable. And so we actually were seeing sort of reactive muscle fibers, which are very much predominant in JDM compared to controls, and that the immune cells are very much apparent in our patients and less so in controls, which then enables us to consistently sort of go forward and take these cell types and look at them in more detail. So you can do lots of different things with these sort of large data sets, but to be able to validate that COSMEX was reflective of geomics and other findings within the literature, Aris did pseudo-bulk, so he took the full gene list and so comparing the different fields of view from JDM to controls, was able to identify 491 of the 1,000 genes were differentially expressed, and that, again, the pathways were associated with an upregulation of interferon and a downregulation of mitochondrial genes. So this is good validation. This analysis was done last week, and it's very pretty. I hope you agree. So this is looking at the controls. So this is a field of view from the control tissue and mapping those cell types that have been defined by the clustering, and showing that we have the green being sort of the true muscle fibers, and sort of, just for an example, the light blue are lymphocytes, which there are far less of than if you look at the JDM, where we have quite a different landscape. We have sort of obviously a vessel here, where we've got the parasites sort of surrounding it, and we've got sort of a lot more sort of lymphocyte infiltrate. Equally, the sort of muscle fibers are a lot more sporadic in terms of their shape and size. So just by looking at the images, we're sort of gaining quite a lot of information from that. We have a long way to go in terms of the sort of analysis and cell types we want to look at. But for example, taking the macrophages that we were specifically interested in from that 1,000 gene list, and considering there are very few macrophages within the controls, we were still able to see 32 differentially expressed genes, comparing the macrophages from JDM to control. So why should we do spatial sequencing? Well, we've taken the muscle biopsy. We want to retain that spatial context because there's so much happening in that biopsy that you can't tell by doing sort of bulk sequencing. We want to be able to understand the cell-to-cell interactions, which will enable us to understand the sort of the pathways and mechanisms, identify functional tissue areas, and reveal where active biological processes are rather than just knowing what they are. So we've got a big sort of progression in the field to go into the single cell and spatial sequencing, but there are a lot of limitations. You need to sort of understand why you're doing it, what you're doing with it. I think the sort of very much single cell resolution, so we're looking, if you're looking at Xenium or the COSMEX, they are more validation platforms. You do need to probably have a reasonable understanding of what's being seen in bulk sequencing and being able to answer specific questions, whether you're looking at MSA subtypes, clinical variation, specific pathways, because these technologies are very expensive. So I think the growing field will help us sort of to talk to each other and sort of build that picture. So none of this work could be done without the juvenile dermatomyositis cohort and biomarker study in the UK. So I'd like to thank everyone who's involved with that. My group, so Aris and Nia. Unfortunately, I have to leave this afternoon, so if you have any further questions, please find them. And thanking everyone that's involved in my work, but mainly the patients and families. Thank you very much.
Okay, let's open this up for questions. And Mary, I'm going to start while people get their thoughts together. So what you've shown us is a very cool tool, but when we're thinking about causal structures and causal timing, what can we learn from looking at the, for example, muscle and spatial transcriptomics at one point in time? Like, how do we determine what's chicken and what's egg?
Very good. This is a question that I get all the time with the mitochondria. Yeah, we're looking at a very specific piece of tissue that sometimes might not even, unless it's sort of guided by ultrasound or other radiology, it may not be the most focal point of that sort of lesion. So I think it gives us a lot of information, but we can't rely on that as everything to be able to then sort of trajectorize what's happening with that patient or patients. So I think it's all a sort of jigsaw puzzle of being able to use the information we learn from the muscle at that time point and working out what markers from that we can then look at in blood and sort of see if there's a prediction marker from that. But ultimately, we do need to know what's going on at tissue level to be able to define targets and to be able to use the identified mechanisms to be able to improve some therapies, which, yeah, I guess if it is a specific time point, but usually the muscle is being taken before they're receiving treatment at the beginning of disease. So hopefully that does give us the best sort of information we can. I mean, ideally, we'd want to see what happens 10 years down the line, but that's not feasible at the moment.
So I have a follow-up question, which is, so we have these previous studies of looking at bulk sequencing largely in the periphery, and now you've got some preliminary information about what's happening in the muscle. To what degree can differences in the sequences expressed help us understand why it's so important to look in the muscle? Like, why can't we just look at the blood? Why do we need to know what's in the muscle?
So for an example, what we've looked at in terms of the blood versus the muscle, we're getting quite different gene expressions for the mitochondria. And what we've seen is that with the mitochondrial encoded genes, there is a fundamental problem happening within the mitochondrial genome in muscle. That's not the case in blood. We're having a sort of associated gene signature, and they're very different cells. I mean, mitochondria, muscle require a lot of energy. You have very densely packed mitochondria. So thus, we need to see definitely at a tissue level what the sort of the signatures are and what the sort of mechanisms are. I think also this reflects on how heterogeneous the disease is, and thus one signature is not going to be the same for every patient. So that's also going to be reflected across the different tissue types. I hope that helps.
Are there any other questions? Well, if not, a big hand of applause for Mary.
We are now moving for our next speaker, Angelo Ravelli, Professor of Pediatrics at the University of Genoa and Scientific Director of the Gosling Institute. Angelo was also President of the Pediatric Rheumatology European Society and co-led with Brian Feldman the task force that developed treatment recommendations for juvenile myositis. And he's going to present us a talk about treat to target and treatment advances in juvenile myositis. Please, Angelo, the floor is yours.
Thank you very much, Rachel, for the kind introduction, but particularly for the invitation to Gcom 2026. Great pleasure to be here. So my task today is to present you the multinational effort which has led to develop the recommendations for the treat-to-target strategy in juvenile dermatomyositis, and I will also provide you with some information on the most recent therapeutic advances in the management of JDM. So here are my disclosures. So I don't know how many of you are familiar with the treat-to-target strategy. So I will just remind you how it works. So let me first emphasize that it is only concerned with the overall therapeutic approach and has nothing to do with the choice of specific medications. So what you essentially should do to implement this approach is before starting the treatment in your patients to predefine first the therapeutic goals, the targets that you want to reach. Let's say ideally the state of remission, or in the most difficult or most long-standing patients, you might be happy with reaching only the minimal state of disease activity. But then you also need to pre-establish the timing when you want to reach the targets that you have decided. And third, you need to decide to choose the outcome measure, ideally a composite disease activity score that you wish to use to assess the level of disease activity and therefore to evaluate whether the target has been reached. And then you start your treatment. For instance, you might want to achieve a 50% improvement in the overall level of disease activity at 3 months and the state of inactive disease remission at 6 months. Then you start your treatment. At 3 months, you measure the level of disease activity. If you have reached your target, you continue by continue on the scheme, therapeutic scheme that you have decided initially, with the aim of maintaining the target achieved, sustained for ideally forever. If the target has not been reached at the first visit, then you should adapt the treatment. So increase the dosage of medication, add additional medication with the aim of reaching the target at the subsequent visit. Then you measure again, target reached, continue, trying to have it sustained over time. Target not reached, again, you increase, implement, adapt the treatment with the aim of reaching the target. So you continue with that way until you have achieved the target that you have predefined, and that should be maintained ideally forever. So as probably many of you are aware, that the recommendation for the treat-to-target strategy has already been published through multinational collaborative efforts for juvenile idiopathic arthritis in 2018 and also for childhood onset lupus a few years later. So we thought that the time had come to set up this strategy also for JDM. And which were the reasons that led us to start with this project? First of all, as you all know, JDM is like GIA, and particularly lupus is a multi-system chronic autoimmune inflammatory disease. Although there has been a remarkable prognostic improvement over the last decades with reduction in mortality and amelioration of functional outcomes, still many children with JDM experience a decrease in their quality of life, and many of them also still accrue a sizable amount of permanent organ damage. And also, we all know that despite the recent therapeutic advance, still a sizable proportion of JDM patients still do not respond satisfactorily to the contemporary therapies. On the other hand, as it has been reminded yesterday by Brian, only a few randomized controlled trials have been performed thus far in JDM, essentially 1.5, let's say, and despite the promulgation of several therapeutic recommendations, still no standardized, harmonized, widely agreed therapeutic protocols exist. And we knew that several studies, particularly in the adult rheumatology field, the adult rheumatology have started much earlier than us to apply this strategy. These studies have shown in several adult rheumatic diseases that this approach may improve disease outcome. And in the end, there were not yet recommendations available for the T2 tag in JDM, so we decided to start this multinational collaborative effort, which was conducted following strictly the standard operating procedure that were promulgated by the EULER. So we first created a steering committee. Brian and I were the PI of the project, and the steering committee first formulated 14 questions which were aimed to explore the available evidence that could support the creation of these recommendations. Then we asked for very smart fellows to perform a systematic literature review to look at the existing evidence that could be used to answer these 14 questions. Then we gathered an international task force of experts in JDM. First, we developed provisional recommendations. Then these were submitted to a Delphi survey. The entire task force answered this survey. Based on their responses, their suggestions, their criticisms, we set up the preliminary overarching principles and recommendations. And these were discussed in a consensus conference, and we ended up, after discussion and voting, with the final set of overarching principles and recommendations. But in parallel with this effort, the 4 fellows were kind enough to make a retrospective review of the patient series of patients with JDM followed at Sick Kids in Toronto and at the Caslini Hospital in Genoa, Italy, to seek for data that could be useful to set up the targets and the timelines for their achievement. And this was important because these data came from the real world of clinical practice, and they supported the discussion that was essentially made up after the review of the systematic literature review. So combined literature data with real world clinical data. And this is the outline of the systematic literature review. The fellows scrutinized as many as 2,714 articles, and they ended up with 205 articles, which showed sufficient data that could serve to provide information useful to answer the 14 PICO questions that were formulated by the steering committee. And this is just to show you a few examples of the review of patient data that was made on patients followed at the Cassini Hospital in Genoa. We draw Kaplan-Meier curves to examine over time the timing of achievement of the most important, potentially important targets, and also the proportion of patients who were able to reach these targets. For instance, for the time for skin remission is shown here. We found that more than 80% of our 44 patients that were evaluated were able to achieve a complete healing of skin disease, and this occurred after a median time of 12 months. The time to achievement of complete recovery of muscle strength, so muscle remission, this was achieved by around 95% of the patients within the median time of 6 months. Muscle enzyme normalized earlier, more quickly. They reached the normal level again in 94-95% of the patients, and this occurred within a median of 3 months. Achievement of inactive disease using an established definition that are the modified Printo criteria, and the inactive disease was reached by 80% of the patients within 13 months on average. And finally, all patients had been able to have the doses of steroids reduced below 0.3 milligram per kilo per day, and this reduction in the dosage was achieved after around 12 months since the onset of the start of the therapy. And this is just to show you how the newly developed composite disease activity score, the JDMI, Juvenile Dermatomyositis Activity Index, which was developed and validated by our group, how it perform well, so it depicts well the progressive improvement in the level of the progressive decrease in the level of disease activity over time. And this is how the task force that we gathered to make this exercise it was composed. There were 40 participants from 20 countries that I highlighted in this graph. They came from 5 continents. We included not only pediatric rheumatologists, there were 28, so the greater proportion involved pediatric rheumatologists, but there were also 2 neuromuscular specialists, 1 dermatologist, 2 health professionals, 1 physiotherapist and 1 research nurse, 2 patients, 1 parent, which was supported by Cure JM, that I thank for their help, and there were 4 research fellows who were those who made essentially the work. And this is the photo of the task force group during the break of the consensus conference. The consensus conference was held in this beautiful ancient villa, which belongs to the Gazzini Hospital, and is the site of the Gazzini Academy, which is the branch of the hospital which is dedicated to scientific and educational activities. So we stayed together for 2 days, discussed quite heavily, and voted for each of the potential recommendations. So I will show you some, the most relevant. So for sake of time, I can't show you all of them. You can find in the published paper. Anyway, just these are the overarching principles. The most important principle, the first one, is that we acknowledge that any therapeutic choice, but not only the choice of the medication, the choice of the targets, the timing, the decision of the time of the targets should be always discussed and shared with the parents, and whenever appropriate also with the patient, with all components of the multidisciplinary healthcare team, not only, of course, pediatric hematology, but all the specialists which participate in the care of the child. And point C is the multidimensional goals of the treatment. So we know that we should not just aim to control the signs and symptoms, but we also need to try to prevent the development of organ damage, disease complication, particularly carcinosis and lipodystrophy. We also need to prevent flare. But this objective should be always coupled with avoidance of drug toxicity. We should not overtreat our patient. We should prevent treatment complications, particularly those related to steroids or to major immunosuppressants. And of course, we care for children and adolescents, so we need to take particular care to optimize physical function, mental well-being, growth and development, particularly pubertal development. Quality of life should be preserved, and we should take care about the child has have a good education and participate in social activities. Point is very important because it regards steroids. We recognized that glucocorticoids remain a major essential therapeutic intervention that is needed, fundamental to achieve early disease control, but we also want to acknowledge that the doses of steroids should be progressively reduced whenever possible by keeping control of the disease, and to the lowest level needed to achieve, to maintain disease control, and should be whenever possible, as early as possible, I would say, discontinued through the optimization of concomitant immunomodulatory therapy. And the point F is the main essence of the treat to target. So the treat to target requires the regular assessment of the state, the level of disease activity, and the adjustment of the treatment until the desired target is reached. Going to the recommendation, this is the first set. So it was on the left, on the right, you see the percentage of agreement about this single individual recommendation that was achieved during the consensus conference. So 90% of the participants, 91% of the participants agreed that the primary target should be in all patients the achievement of inactive disease, that is complete absence of any signs and symptoms in muscles and skin, but also in other potentially involved organs and systems. We prescribe very clearly that disease activity should be always assessed using a standardized and validated in children and adolescents assessment tools, ideally a composite score. And then we defined the, let's say, interim targets that might be, that should be pursued while trying to reach the final, most qualified target of inactive disease. So initial target, already within 6 weeks, the minimal level of clinical improvements should be obtained, and this should be measured using an available definition of improvement, a composite disease activity measure, or if these measures are not available, by simply through simply the physician global assessment. Of course, patients should be free from life-threatening conditions at this time. Then, after 3 months, a moderate level of clinical improvement together with normalization of muscle enzymes should be achieved, and the life-threatening conditions should be completely resolved at this time. Third interim target, after 6 months. At 6 months, the complete normalization of muscle strength need to be reached. And the primary target of inactive disease, defined as I said previously, should be achieved within 12 months. At 1 year, the patient should be in complete remission. Glucocorticoids should be aggressively tapered, of course, by keeping an eye on the course of the disease in parallel, and so as long as interim targets are reached, of course, so we should make sure that the interim targets are reached while decreasing steroids, but once the child is doing well, we should be able to discontinue completely these glucocorticoids within 1 year, within 1 month, or sooner whenever possible. As I said, another main principle of the treat to target is that once the primary target has been achieved, it should be sustained, and we need to continue to monitor our patients by applying the outcome measures that we have chosen to make sure that the target is maintained over time. Switching briefly to the second part, emerging therapies for the GM. So this is a list of the therapies that have been proposed recently. We have heard yesterday that we are now living an exciting time in the area of new treatments for inflammatory myositis, either in adults, both in adults and in children. And we also have a bright future ahead of us. And you see here the newly proposed medication belong to 7 different categories. There are the JAK inhibitors. There will be 2 oral presentations after my lecture on baricitinib and tofacitinib. So they are largely used so far, which had to inhibit also the interferon signals, which is of course helpful. Then there are the medications which are aimed to deplete B cells, which are the main inductor of the pathophysiology of the disease. There are monoclonal antibodies, but there is also a bifunctional chimeric monoclonal antibody which downregulate B cells. So B cells are one of the main specific targets of the future therapies. Then there are the agents which address the survival, reduce the survival, or inhibit the differentiation of B cells, those that are directed towards BAFF, such as belimumab and blisbimod, or those who are directed against TACI, which still is part of the BAST and APRIL system, such as atacicept or telitacicept. Then there is daratumumab, which targets plasma cells, is an anti-CD38 compound. And then there is CAR-T cell therapy, which is perhaps at the moment the most exciting intervention. This has been largely discussed in the previous days of the meeting. And there are then specific medications that are directed against reactive oxygen species scavengers that have an antioxidant effect, and metformin inhibit the function of mitochondria, which we have heard previously are very much involved in the pathogenesis of the disease. And finally, the medications, the agents which inhibit type 1 interferon, which is one of the main determinants of the pathophysiology of the disease, such as anifrolumab and adazucibart. So some of these agents have already been tested in patients with JDM, but most of the data are published as isolated case reports or small patient series. So I have chosen, for sake of time, two of the most recent experiences. The first regards anifrolumab, the interferon, type 1 interferon inhibitor. So this 8-year-old girl, very young patient with JDM, she had prominent skin disease. She had not responded to S flare despite the use of all available medications, which include IVIG, methotrexate, MMF, calcineurin inhibitors, tacrolimus of cyclosporine A, rituximab also, and JAK inhibitors. But once they gave to these patients the first shot of anifrolumab, which was given 300 milligrams once a month, you see after 3 months there was a remarkable, impressive improvement in skin disease. There were some remaining lesions which were, however, expression of residual damage, but all the signs of active disease disappeared completely and remained disappeared until the last follow-up. And then the last case, we have here Rebecca, who was mostly involved in this experience. So this is a child with JDM, which was again refractory to all contemporary medications, essentially, was treated at Bambino Gesù Hospital with CAR T-cell anti-CD19 CAR T-cell therapy. And the results were again striking, because you see the skin lesion before and after, and 34 weeks of treatment. So the complete healing of inflammation in the facial area. On the right, the reduction in the signaling hyperintensity in the muscles disclosed by repeated MRI. In the upper part, the decrease in the physician global assessment after the administration of CAR T cells, the increase in muscle strength measured with CMAS, and also the improvement in skin disease demonstrated by the reduction in the score of the cut. But on the right side, in the upper part, you see the course of the cell depletion. B cells disappear completely, and in parallel there was an increase in the CAR T cells, very shown also in the lower part by this. Anyway, you see in the how you say this analysis. But anyway, so you see on the right side that the CAR T cell populate the peripheral blood. But what was most important in this experience is the assessment of the proportion of cells of the various immune cells before CAR T cell therapy and after, and then after CAR T cell therapy. On the left side, you see that the proportion of plasmablast and of autoreactive cells, the blue and the red part of the first column on the left, is quite sizable. But after the therapy, there was a reconstitution, of course, of the immune cell sets, but the pathogenic cells, the plasmablast and the autoreactive cells, were almost disappeared. So this gives the hope that the immune system could be in some way reset and might have lost the initial error which has led to the development of the autoimmune disease. So with this, I am done. Just let me return for a while to the treat to target efforts. I wish to thank the Casini Hospital, my hospital, which because it supported entirely the effort by investing the funding donated by the Italian taxpayers. So, part of these taxes are given to charities, and we get quite a bit of money. So through this money, the project was supported. And finally, I also wish to thank the components of the steering committee. There is Lisa Ryder here. I don't know if there is anyone else. Lisa McCann is there, and Brian Feldman, who was the co-PI of the project, but I wish to thank in particular the 4 fellows who made most of the work, who were Sylvia, who is here in the audience, on the left Anna, then Tali, on the right side Jane. And then I thank you very much for your attention.
Thank you, Angelo, for your amazing talk. Any questions for Angelo? While you're thinking, oh, sorry. Go ahead, Mark.
Thank you, Angelo, for the great overview. And I was always, with this question, you showed in your later sheet that we had this lack of clinical trials, 1.5 RCT. So what do you do if you don't reach your endpoint, if you don't have RCT-proven therapeutical options? How do you choose then for your next treatment?
You mean when you don't have any therapeutic option left, what they choose? You mean?
I mean, without having evidence-based therapeutic options, how do you choose your next step after you had your evidence-based medical options and you don't reach your target?
Sorry, do you understand that?
There's a lot of echo. Maybe speak. Yeah, there's a lot of echo.
Okay, shall I do it again? Yeah. Is it better now? So my question is, if you don't make your endpoint at 6 months or 12 months, according to the treat-to-target strategy, what is then your next step if you are through your evidence-based options qua medicine?
Brian, can you help me?
So how do you make the choice if we don't have evidence-based clinical trials to choose the next agent?
There's a lot of echo.
This is what I thought I had understood. So, therapeutic choice. You know, therapeutic choice is based on essentially on experience, on reading the papers, on participating at congresses and talking with colleagues. So I think that nowadays, if I can, let's say, outline what I think is the ideal therapy for JDM, I would say, first, of course, high-dose steroids remain the mainstay of the treatment, so pulse steroid therapy, together with methotrexate, we still remain the second-line basic therapy. And then, if this doesn't work, this is a good part. So it's essentially the treat-to-target mandates, as I've said, the quick control of the disease. You should control as quickly as possible the disease because we know that this helps to avoid complications, to prevent calcinosis and other disease complications. So what I would say, what I would say, what would do historically would be in a patient who does not improve satisfactorily, combine methotrexate plus cyclosporine A plus IVIG. If this doesn't work, you might either replace methotrexate with MMF or add or replace methotrexate with the JAK inhibitors. If the patient still does not do well enough, I would switch more quite rapidly to rituximab nowadays. This is the current therapy that I would. With this approach, our experience is quite positive, I should say.
Question from Adam?
Yeah. For the treat-to-target follow-up periods, I saw that they were straight, not dependent on what medication was being used, but we have different medications now with very different response times that we would expect. What was the reasoning behind picking a set time instead of setting a different time for follow-up or goal based on the medication?
I find it difficult to understand.
So depending on which medication you treat, you might expect a different time to response. Why did we set fixed times rather than basing it on the different medications used?
You know, it's, of course, this is an important question. So this are the main principles of the treat-to-target. So you need to set fixed time points to make the therapeutic change in case the target is not reached. Of course, this is, they are not written on the stone. This is just a kind of example of examples of reference points that should be helpful to make the best possible therapeutic approach. Of course, we know as clinicians that such tight therapeutic approach might not be feasible, but still, with the new therapeutic interventions, we thought, or at least the participants in the consensus conference thought that these let's say, ambitious targets and timeline should be feasible, should be achievable.
Thanks, Angelo, for a nice talk. So my question is regarding that you have portrayed treat to target as a blanket therapy for GDM as a whole. But as the understanding of the disease is improving, there are a set of patients, I mean, it's not a very homogeneous disease. There would be patients with MDF5 or autoantibody positive or more aggressive. So do we need to, what is your take on that? Do we need to, escalate the treatment upfront rather than waiting for the timelines that you have set?
Let me ask the question.
Narendra, that was really hard to hear back here. So are you talking about different MSA?
So my question is...
Different MSA groups?
Yeah, different subgroups. JDM is not a homogeneous disease.
Should we expect different treatment response times?
Exactly.
With, yes. The antibodies, yeah.
Okay, so it's the influence or the impact of specific myositis-specific antibodies was acknowledged in the recommendations, but we thought that so far the evidence is not sufficiently strong to make a different recommendation by the presence of a particular myositis-specific antibody. So it's because there is not sufficiently strong evidence that they impact the prognosis, even though, of course, it should be taken into account in deciding the specific therapy, what is the specific risk that is associated with a particular myositis-specific antibody.
Belina? Hi, I'm Belina from the US. I just wanted to get a clarification on the steroids, because I think in the beginning it said very gradually wean down on the steroids, but at the end I thought once you reach like remission, then you try to aggressively take it off. So I was hoping to get clarification on that, and if in your experience, what's like the interval how you wean down the steroids in your practice?
Brian.
So with the prednisone taper, what's the process that you're using for prednisone taper?
So the prednisone taper. So it's as I said in my introduction. So the treat to target has nothing to do with the choice of a specific medication, but also to the way a specific medication is tapered or increased. This is up to the clinician. It's we just recommended, not mandated, recommended that steroid therapy should be discontinued ideally, of course, within 12 months. But the way you reduce steroids, we tried to propose, Silvia, you remember well, so we propose a specific recommendation about the timelines for reduction of the steroid dose below a certain threshold, but this was not accepted by the participants, by the expert participants, who thought that the reduction in the steroid dose should be decided by the clinician, taking into account the endpoint of 12 months when it depends how the patient responds, how the disease improves or worsens, so it's not part of the treat to target strategy.
Angelo, thank you very much.
Okay.
We're now going to move on to our selected oral presentations. We have three of them. The first is Mark Janssen from Utrecht, coming to talk about validation of galectin-9 and CXCL10 as predictive biomarkers for disease flares in juvenile dermatomyositis.
So indeed, thank you very much. And first, obrigado Raquel por este congresso incrível, este maravilhoso. Desculpa para meu sotaque brasileiro. Eu vou continuar em inglês agora.
It's great, thank you, Mark.
So, these are my disclosures, and I'm sorry for my rusty Portuguese. I want to start with a case which is on the treatment at this moment. I called her Sophia for here. She presented in 2020 at the age of 11 with severe muscle weakness, strongly positive GWAS. She could not lift her head, could not roll over, had livid eyelids, cotron papules and MCPs, dysphagia and achalasia. And on her MRI, she had diffuse inflammation of the muscles. We didn't do a muscle biopsy. And her CT scan of the lung showed interstitial lung disease with a clear impaired lung function. So her diagnosis was JDM, and we found anti-PL7 antibodies, which made it an antisynthetase syndrome. She was treated with methylprednisolone, methotrexate, prednisolone, and we started CellCept because of the ILD. And she recovered quite good. Her muscles became normal, the skin normalized, and she also didn't have complaints of her respiratory condition. The ILD was not completely in remission on radiology. But then in 2023, she experienced a flare with muscle pain, myopathy, and also coughing and skin disease. And you see again edema on the MRI and a worsening of the ILD on CT scan. And you can see an elevated CK in the lab and elevated interferon-induced biomarkers, galectin-9 and CXCL10. But when we look back, we actually saw that galectin-9 and CXCL10 were already increased more than a year before. And this was something strange, and we thought, hey, could we have seen this coming? There is a clear need for predictive biomarkers in JDM. We have a lot of biomarkers which correlate with disease activity, but not predicting flares. We know that dermatomyositis is heterogeneous and it has an unpredictable disease course. 1/3 is refractory, and after another 1/3 experience a flare. So at the right side, you see the Dutch cohort where 1 in 3 of the patients had a flare, mostly in muscle and skin, sometimes skin only or in other organs. So we need biomarkers to predict these flares. Galectin-9 and CXCL10 are interferon-induced biomarkers, and they've been validated nationally and internationally as robust biomarkers of disease activity, but we wanted to know, can these biomarkers also predict an upcoming flare? So for that, Saskia Veldkamp, our PhD student, who's now finishing but could not be here, has done a lot of work. We analyzed more than 1,100 longitudinal serum and plasma samples in 158 patients in 4 countries. We measured galectin-9 and CXCL10 by a Luminex assay, and we prospectively collected clinical data. We had the definition of a flare of increased disease activity acquiring an intensification of systemic therapy to really have the clinically relevant flares. And we used only the steady state time points because we know that these biomarkers need some time to normalize. So we didn't use the first 6 months after diagnosis and not the 3 months after flare because they're still elevated then and not during refractory disease. And that left us with 765 measurements from 141 patients and 66 flares. And then we used loose curves to look at biomarker levels in the 2 years before a flare and to see the trends. We then looked at trajectories with joint latent class mixed effects models, LCMM, and then we calculated the risk by doing Cox proportional hazard methods. So these are the loose curves, and what you can see here, that is a line in red. And this line in red are the patients who experienced a flare at time point 0, while the blue line are the patients who did not experience a flare. And what you can see is actually the same as we saw in the individual patient, that for the whole cohort, the galectin-9 and CXCL10 already started to rise around 10 months before the flare, while CK was only elevated at the flare or maybe just before. So now we have an indication that these biomarkers start to elevate before a flare, but could we also prove that these trajectories are associated with a higher risk of flare? So for that we did joint latent class mixed effect models, and these models combined these longitudinal outcomes with a time event, in this case a time to flare. So time point 0 here, that's not the time of diagnosis, but the first biomarker measurement. And then we could distinct 3 different trajectories, of which 2 have elevated galectin-9 levels, the green and the red line. So in the green class, 100% of the patients had a flare, and in the red class, 89% of the patients had a flare, while in the low galectin-9 class, the black class, only 15% had a flare, of which half of the patients only had a skin-only flare. So no muscle, skin only. And for CXCL10, we saw exactly the same results. So next we wanted to see if we could quantify this associated risk with elevated biomarkers. So we did multivariate Cox proportional hazards, and indeed, we could see that a doubling of galectin-9 doubled the risk of flare during time. And that did not apply for skin-only flares, so that's important to know. And that was exactly the same for CXCL10. A doubling of CXCL10 doubles the risk of flare, but not for skin-only. Then we made Kaplan-Meier curves. So they show the probability of remaining flare-free after a low versus a high biomarker measurement. And we defined high as the 75th percentile as a cutoff. And the blue line are the patients with low galectin-9 and the red with high galectin-9, and the same applies to CXCL10. And what you can see here, that if you have a high galectin-9, your risk to flare in the first 6 months is 20%, comparing to a low galectin-9, it's only 3%. And that was the same to CXCL10. And when you calculate then on these binary biomarker levels, you have a hazard ratio of 6. So 6 times more risk of a flare. So then we thought, what happens if you combine these biomarkers? And that's what we did. So we made Kaplan-Meiers again, and we looked at the combination of biomarkers. So low galectin-9 and low CXCL10, they are displayed in the blue line. And the purple line are the combined high biomarkers. And then the flare rate is even higher, 30% in the first 6 months. And if you then calculate binary, you see that the hazard ratio is 12. So you have a 12-time higher risk of flare if you have both elevated biomarkers. So to conclude, we saw that galectin-9 and CXCL10 start to rise about a year before the flare. And these elevated trajectories of galectin-9 and CXCL10 are associated with a higher risk of flare. Galectin-9 and CXCL10 are strong predictors of flare, with the exception of skin-only flares. And when these biomarkers are both high, there is a 12-fold higher risk of a flare. So what has this for implications? We think that these biomarkers can guide decisions of tapering or intensification of treatment. And then we can reduce subclinical chronic inflammation, and we hope to improve the patient outcomes by that way. We implemented these biomarkers now in the Netherlands and in our national guidelines, so we can do it as routine diagnostics. And I think that's really the step we have to make as an international community, not just to do these things in research setting, but just like you do a muscle examination and a CK, you should measure it every time, and then you can start using it in treatment decisions. I have to acknowledge my team, Femke van Wijk, Annette van Rooij, and especially Saskia Veldkamp and all the international collaborators we have. Thank you so much.
Okay, so this abstract is now open for questions.
Thank you so much, Mark, for an excellent presentation. Can I ask you, in routine clinical practice, if they go up up to a year, before a flare. So you have a patient who is completely fine clinically, and the galectin-9 goes up. What are you going to do?
Yeah, so I had a patient this year, but now these data are unbiased, so we didn't do anything because we were not sure. We want to publish it and then... but this patient was completely fine. Always repeat your measurement because galectin-9 and CXCL10 are type 1 interferon-induced proteins. So if you have a flu or rhinovirus, then they will go up. So recheck them to see if this is something serious and if they keep on rising. What I won't do at some point is stop the medication, because this was like a patient where we were doubting to maybe reduce the dose or start stopping. Don't do that when you see rising biomarkers. Yeah, and if it really keeps on rising, the question is whether you should already intensify your treatment. And that the fair question is that's what we don't know. We know your risk of flare. You can explain to the patient, but we don't know if you should really intensify your treatment. I would not stop.
Okay, Kaveh first at the back.
Thank you. This is a wonderful presentation. My question is kind of about the final slide, when this might be available. In the United States, neither CXCL10 nor galectin-9 are, as you know, commercially available. And it seems to me like the time frame with which to make such tests commercially available in the United States context would be quite long, unless somebody is already well along the way that we don't know about, or that I don't. So I guess my question is, we can get commercially Siglec-1 via flow cytometry in the US, and we can also get CXCL9, other biomarkers like GDF15. These are all different, but maybe in some sense related. Do we need to go through the whole process of commercializing CXCL10 galectin-9, or is there a sufficiency of any existing biomarkers, and what do you think?
Yeah, so this is a problem. We were just talking about it. I don't know why it's so difficult, but yeah, this is a commercial thing. Siglec-1 is a really reliable disease monitoring biomarker. We validated that, and also in adults. So this is very promising. The problem is we measure Siglec only in research setting, and now you see the problem. We don't have enough samples to do these analyses, because you need you need every time samples to see those trends. And in research setting, we take 3 times per year, which is not enough. So we will try the following years to include Siglec-1 for this analysis to see if it has the same effect. I think it has the same effect, but I cannot be sure. Yeah. So that's the fair question, the fair answer to your question. Yeah.
Thank you.
Okay, one last question.
Hi, I'm Angela Chun from Chicago. I find it curious that the skin-only flares didn't seem to be as strongly associated with the rises in galectin-9 and CXCL10. Do you feel like that is because there's something inherently different with those kids who have only skin-only flares, or do you feel like it's because of how, depending on how you collect your data, how people were kind of calling skin-only flares, right? Was it just erythema versus actual papules or nail fold capillary changes? Yeah. Or do you feel like if you kept watching those kids and you didn't adjust treatment despite that skin-only flare, do you feel like over time that you would see the same kind of rise with muscle involvement?
Yeah, this is the intriguing question, which also surprised us. It can have something to do that mild skin disease doesn't lead to interventions, or that we act differently on it. And so I'm not sure what the reason is. That's the answer. And I don't know what would have happened if you didn't get muscle disease with it, and you would just have waited a couple of months more. So this is definitely, we have to dive into why it's really different for this group. I have to say that all the patients who had a muscle flare almost always had a skin flare as well. So this were combined flares, but the skin only, you're right. And it's intriguing, but I don't know exactly why. Yeah.
Thank you.
Thank you so much, Mark, for your great presentation, but also for your nice introduction in Portuguese. Well done. May I call to the stage now Narendra Bagri from India, that is going to present us his work on tofacitinib for calcinosis cutis associated with JDM, an open-label single-arm study.
Good evening, one and all. First of all, thank you, organizers, for giving me a platform for presenting our work. So we are presenting this work, tofacitinib for calcinosis cutis associated with juvenile dermatomyositis, or what we call this as a TOFCAL study, which has been undertaken by one of my fellow as a part of a thesis. So no personal conflict of interest, but for this particular study, we got a small intramural funding from our institute for the biomarker study, and the drug was provided by an Indian pharmaceutical company. However, the company had no role in the study design or conduct, or no access to the data of the study. So let's move on to the background why we thought of this particular study is, as we have been listening since yesterday, that there is probably interferon-mediated inflammation in juvenile dermatomyositis, and probably a misconduct by the mitochondria which lead to efflux of the calcium and therefore the dystrophic calcification. And this again is driven by probably interferon signals. And having learned this, probably there is a need of a JAK inhibitor might be of therapeutic implications in this. And as we understand, the calcinosis cutis is a debilitating complication affecting about 30 to 70% of children with JDM. And often the treatment is refractory to a host of agents which have been tried for many, many decades. So this is the slide that I was talking about, the probable role of interferons in the pathomechanistic pathway of calcinosis cutis. And taking a lead from this, some of colleagues here have published case reports where there has been some improvement in the calcinosis cutis with use of JAK inhibitors. Although the numbers were small, but probably it gives some value to this. So coming with this background, we formulated a research question that can the administration of tofacitinib for a duration of around 6 months reduce the burden of calcinosis cutis, and which was assessed using Agatston score on ultra-low-dose whole-body CT scan in children with juvenile dermatomyositis. Is Adam here? He was talking about the ultra-low-dose whole-body CT scan, and we chose this because there are clinical challenges in ascertaining the exact burden of calcinosis cutis in clinical practice. So this was chosen as a primary outcome variable. So using this ultra-low-dose CT scan, we chose to go forward for this study, which was essentially an open-label, single-arm study in a single center at the All India Institute of Medical Sciences, New Delhi, which is the largest public-funded tertiary care apex referral hospital of India, where I work. And the study was taken during the last 2 years, and the study population was children 2 to 18 years of age diagnosed with JDM and calcinosis cutis. With this research question, the primary objective of the study was to study the effect of tofacitinib as an adjunct to the standard therapy, which is defined as steroids less than 1 mg per kg per day and methotrexate or MMF or IVIG in children 2 to 18 years of age with calcinosis cutis, which was either diagnosed clinically or with an imaging study. And the outcome variable was change in the Agatston score from baseline at 6 months. And the secondary objectives were to see the change in the CMAS, C-DASI scores, adverse event of the drug during the study follow-up, and we also did interchange in the interferon levels, alpha and beta, during the study period. The sample size calculation was done based on the data from Adams' study, where they have done a probable mean of around 4,600 of the Agatston CT score, and few of the case reports which have shown 50% reduction in the clinical, though it's not the absolute way, but yeah, this is what was the available information. And with this, using a pre-post analysis we took 20 patients would probably be needed to at least demonstrate 50% reduction. So the inclusion criteria were children diagnosed with JDM or JDM overlap, along with calcinosis cutis on physical examination or on previous imaging, while the children who were on immunosuppression, such as cyclophosphamide during the preceding 1 month or rituximab in the last 6 months, were excluded. The other exclusion criteria included the concomitant topical tacrolimus, active tuberculosis, or the hematological parameters, which forms a contraindication for starting tofacitinib. So the intervention was administration of the tofacitinib tablets or syrup as per the age-appropriate bands. And the background therapy with stable doses of steroids, that is less than 1 mg per kg per day, was permitted along with the methotrexate or MMF or IVIG. And the safety measures were taken using constitution of a data safety and monitoring board, and the CTCAE grading was used for defining any of the adverse event during the study period. So this was the Agatston scoring, which was done. The ultra-low dose CT scan was done in the Department of Radio Diagnosis, and the 2 independent pediatric radiologists reported the CT scan of these patients, and they were blinded to the clinical details till the final results of the study. The statistical analysis plan was defined for the tests which was needed, depending on as and when available for the patients, for the patient population, and the effect size was also calculated using D-robust test. So, coming to the results of the study, during the study period, we screened around 119 patients of JDM, and 2 of these patients were cis-overlap patients, and 25 had calcinosis cutis who were enrolled in the study. However, because of the protocol deviation, there were some technical flaws in the CT scan capturing of the images, and we lost the data of 5 patients who were excluded from the analysis, and finally 20 patients who completed the follow-up were used for this per protocol analysis of our study. The baseline characteristics, if we look at around 18 patients were juvenile dermatomyositis and 2 patients had an overlap. The duration of the disease was around 57 months, and the duration of calcinosis cutis at enrollment was around 31 months in these patients. The calcinosis cutis was spread practically all over the body in most of the patients, as can be seen. And most of these patients does not have a severe muscle disease as reflected by the CMAS score. Now, coming to the primary objective, after 6 months of therapy of tofacitinib, what we can see is there is a remarkable reduction in the whole body CT scan scores calculated by Agatston scoring, with almost 42% reduction in the score from baseline in these patients with a significant p-value. And 80% of the patients, that is 16 out of 20 patients, showed reduction in the Agatston scoring from baseline, and none of the patients experienced any further calcification, ulcerative lesion, or worsening of the disease, overall disease, during the study period. And here are the clinical implications of the drug, where we can see that there is an improvement in the ulcerative lesions, including the size of the calcinosis cutis, which is in parallel with the reduction in the size of the reduction demonstrated on the CT scan findings. So, though the drug, the intervention seemed to show a decline in the trend of the CDCI as well as improvement in the CMAS, though it was not statistically significant, probably because the disease was not muscle predominant in the enrollment as well. The steroid doses, if we can look, there are 5 patients who were on steroids. 2 of them continued steroid till the last follow-up, and in both of them, there was a significant reduction in the mean dose of the steroids. The other concomitant therapy which was allowed for the study was the mycophenolate, methotrexate, and IVIG in the numbers which have been shown. In parallel with the reduction in the Agatston score, we could also demonstrate that there was a decline in the interferon levels in patients from baseline to the follow-up of the study, and both levels showed a significant decline in the study population. 3 patients experienced adverse event, but they were CTCAE grade 2, and which were just pus from the calcinosis site and an episode of URI requiring temporary cessation of the drug for a week or so. So, though we could find that the tofacitinib on top of the standard therapy could show a reduction in the Agatston scoring, but of course there are limitations of the study. And of course, the first one is a single-arm open-label study, which limits a direct comparison with the standard arm. The relatively short follow-up of the study, probably we speculate that the overall regression longer time. So 6 months probably has shown a CI score, which we have used in study for the clinical outcome, probably is not powered enough or designed enough to take into account the overall burden of the calcinosis cutis, because it just says yes or no. So to conclude, This is, for our knowledge, was open-label study which has used the tofacitinib for calcinosis cutis in children with juvenile dermatomyositis using a validated objective criteria, that's Agatston scoring. And what we found is that tofacitinib, in addition to the standard therapy that I have shown, has shown to reduce the burden of calcinosis cutis as measured by the Agatston scoring using low-dose whole-body CT scan. And there were no significant major side effects, adverse events noted during the study follow-up. So thank you so very much, and I would like to acknowledge our study team, Dr. Victoria Pirvet for allowing us the CDCI score, Myositis Clinical MCTC group for giving and supporting an opportunity for presenting the data here, and of course, one of my fellow Pavdeet who has helped me in formatting the slides. Thank you so very much.
Any questions? Belina?
Hi, thank you so much. I'm curious if any of your patients had almost like inactive muscle and skin disease, but just calcinosis, and if they showed any improvement?
Okay, so you are saying that only calcinosis, no muscle disease. Yes, I have not presented the data like that in terms, but there were a couple of patients who just had calcinosis cutis, and they showed an improvement during the follow-up period.
Question in the back.
Thank you for your talk. Do you think that tofacitinib will be effective long-term to treat calcinosis, or just in the initial instance that you've shown from this therapy?
What I can gather is you are saying that using tofacitinib, will that be of help in management of calcinosis cutis?
For long-term treatment.
As a long-term treatment. So our reflection of the study is that it actually matters when do you start the treatment, because if I have not shown that data, but 4 patients who have not shown response or not shown a reduction probably had a longer disease duration compared to those who have shown a response. So it's the time when you start the treatment. If the damage or the calcific stone has already been set in, probably it will not melt by whatever therapy we use. But sooner we start the treatment, more likely the chances of success are there.
Thank you.
Last question.
Hello. Thank you for a great talk. That was really lovely. I was wondering if you're seeing an 80% of the patients had a partial reduction in calcinosis after they already had calcinosis, and therefore it seems like this is very much part of the mechanism of calcinosis. Do you think it would be worthwhile to check if giving 6 months of a JAK inhibitor right after diagnosis might prevent calcinosis?
That is stretching too much, but I would say that the moment you pick it up, start hitting it earlier. That's what my take on this. But preemptively starting it would be, probably I won't agree with this given the present evidence.
Sorry, have no more time for questions. Really sorry. Thank you so much for your talk.
Okay, and our last abstract for this session is the longitudinal single-cell transcriptomic analysis of type 1 and 2 interferon response genes in refractory juvenile dermatomyositis patients with baricitinib treatment, being presented by Hannah Kim.
Hi, I'd like to thank the organizers for the opportunity to give this talk. It's really being given on behalf of Jessica Simpson, who's a third-year pediatric rheumatology fellow, but not able to be here to present it herself. So these are the disclosures of the authors on this project. So as we know, the role of interferon in JDM pathogenesis is pretty clear. There have been multiple studies showing an increased interferon-regulated gene and interferon-regulated protein expression in JDM, not only in peripheral blood, but also in muscle and skin, and this generally correlates with disease activity. And we also have seen increased interferon alpha itself in JDM, peripheral blood, as well as increased interferon gamma in JDM muscle. So looking at how type 1 and type 2 interferons signal, we know that interferons can be divided by type, so that they share a receptor if they're the same type. So interferon alpha and interferon beta are type 1, and interferon gamma is type 2. So when an interferon binds its receptor, that activates the downstream signaling, which utilizes Janus kinases, or JAKs, and that leads to STAT phosphorylation, activation, and downstream transcription of interferon-regulated genes and translation of interferon-regulated proteins. And then when a Janus kinase inhibitor or JAK inhibitor, such as baricitinib, is introduced, this inhibits this activation step at the JAK level and decreases the downstream signaling. So we had hypothesized this would help with juvenile dermatomyositis and previously published on 4 refractory patients having clinically significant improvement. There were 4 patients, 2 anti-TIF1 and 2 anti-NXP2. 3 were female, and they had ages between about 6 and 21 years at the start of the study, though they all were juvenile in onset. They had a mean disease duration of about 6 years, and we had used the interferonopathy dosing of baricitinib, so they were starting on 4 to 8 milligrams per day. And they were all very refractory and on other treatments. All biologics had been washed out other than IVIG, but they were allowed to continue steroids and other oral DMARDs. So at 24 weeks, we had published that there was significant improvement in multiple validated clinical disease activity measures, including physician global, patient global, and the CDASI, and these are just some example pictures showing improvement in rash and improvement in nail fold capillaries. We also looked at some interferon-related markers, which included the whole blood 28-gene interferon-regulated gene score, which had been developed and validated comparing interferonopathies to healthy controls on a nanostring platform, and then also shown to be elevated in JDM in general, and an increased IP10 or CXCL10 protein at baseline that decreased with baricitinib. So even though we have that data, the objectives of this study were to answer some remaining questions. Is there a difference in how the individual cell types respond to baricitinib? Is there a difference in type 1 versus type 2 or specific interferons and how they respond? And is there a difference in how that change looks looking further out to 72 weeks? So to answer these questions, we used single-cell RNA-seq, which was performed on peripheral blood mononuclear cells on these 4 patients, looking at week 0 or before starting baricitinib therapy, at week 4, week 24, and week 72. So the data was processed using Seurat, and we used Azimuth to do cell type annotation. And cell types with less than 30 cells were excluded from analysis, and lowly variable genes and genes that were not highly expressed were removed from analysis. So how did we look at this interferon expression? We looked at 4 different interferon-regulated gene scores. 2 were hallmark from the molecular signature database, or MSIG database, 1 for interferon alpha and 1 for interferon gamma. We also looked at the same 28 interferon-regulated gene score that had been established in interferon 2, patient 3, patient 4, the rows. And then for each cell type for a patient, we showed the data as violin plots. So on the left is time point 1, or starting baricitinib, and then in sequence, time point 2, 4 weeks, time point 3, 24 weeks, and time point 4, 72 weeks. So in this example for patient 1, CD14 monocytes, when comparing baseline to time point 2, it was significantly decreased, which is signified by the black star. Also for time point 3, but actually at time point 4, there was a significant increase in this interferon alpha score. So when looking across overall all these 6 cell types and 4 patients, most of them had significant decreases, which is what we expect in general with a JAK inhibitor for the interferon scores, or each of these interferon scores, because it blocks both type 1 and type 2 interferons. There were some cell types that had insufficient cells for analysis, and there were some comparisons that were not significant to baseline, and then there was just this one where there was a significant increase. And to help kind of summarize these results when we were looking at it, we looked, for example, by specific cell type, so looking just at the CD4 central memory T cells. For each patient, there are 3 comparisons, baseline versus time point 2, time point 3, time point 4. And for patient 1, all 3 of those were significant. And then we looked at that for all 4 patients, so 4 times 3, there's 12 comparisons, and tried to look at how many of the comparisons were significantly decreased. So in this case, looking at interferon alpha scores and CD4 central memory T cells, 12 out of 12, or 100% of these comparisons were significantly decreased. So we did this type of analysis for all 4 of these scores: interferon alpha, interferon gamma, the 28 gene and 21 gene scores. And to just summarize what we saw, overall, similar to that large violin plot summary, generally most of the scores in most cells had decreases at most time points. We did see that first patient have an increase in interferon score at the last time point, 72 weeks, for CD14 monocytes for both the interferon gamma alpha, interferon alpha and interferon gamma scores. Looking at cell type over all patients and all scores, CD4 central memory T cells had the most consistent significant decreases, or were most sensitive to baricitinib, and having only one that didn't significantly decrease. And then the MAI T cells were the least sensitive, with 59% of the comparisons having significant decreases. And then by score, the interferon gamma score was most sensitive to baricitinib, with 88% of those comparisons being decreased, versus the interferon 21 being the least sensitive to baricitinib, with 80% decreases. But there is a much wider range, 59% to 98% for cell types, versus a smaller range, 80 to 88% for interferon score. Then we tried to looking at both by interferon score and cell type, what are the differences? And when we looked that way, we saw that there's some similarities between interferon 21 gene and interferon 28 gene scores, and then the alpha and gamma scores seem to behave more similarly in these patterns, so we could kind of group them together. B cells were consistently decreased in the interferon 21 and 28 gene scores. 100% of the comparisons were decreased, versus in the interferon alpha and gamma scores, 78%, so they were less sensitive with those types of scores. And then the CD14 monocytes were more responsive with the interferon alpha and gamma scores, with 89% of the comparisons significantly decreased, versus only 56% in the interferon 21 and 28 gene scores. So in conclusion, refractory JDM patients who had all clinically improved with baricitinib treatment added had decreases in 4 different interferon-regulated gene scores in most cell types through 72 weeks on baricitinib. The interferon gamma score and the CD4 central memory T cells were the most sensitive to change, though there was some variation by score and cell type. And we found that both type 1 and type 2 interferon-regulated gene scores decreased in multiple cell types on baricitinib, correlating with clinical response. I'd like to acknowledge particularly Jessica Simpson, as this was her project, her fellowship project, the others in our group, including bioinformatics, the sequencing core, and others who collaborated on this project. And I'm happy to take any questions.
Okay, so we have a question from Aris.
Hi, thank you for the great talk. I was wondering, because as you said with your Venn diagram, a lot of the genes in all the different interferon databases or the different scores that exist are kind of overlapping. If you've tested for genes that are specific to type 1 or type 2 interferon, rather than including all the overlapping genes, and if there's any differences between them. Thanks.
Yeah, that's a great question. We're actually looking at that now, but we don't have the results yet.
And the CD4 T cell memory cells appear to be more sensitive. We think baricitinib is effective clinically in this disease. Does that give us any information, do you think, about pathogenesis or at least mechanism of effect of baricitinib? Or is it just too small a series of patients to really tell?
I mean, I think it's interesting. I don't know if it's true. And I think we're also interested, as what was asked about, if there really are differences between the 2 types of interferons. But yeah, I think it's hard to know if this is a difference in how baricitinib works versus a difference in what was there from the JDM to begin with. And like, that's the difference. So it's hard to tease out that diff.
Jessica?
Thanks for the great talk. Kind of along those lines, with any of your work, have you, I guess, had a sense of which among the JAKs, since they're blocking different specific types of JAKs and ticks, if you have any sense of which you think is more effective in JDM?
Yeah, that's a great question. I mean, I'm curious if anyone else has any direct experience, but usually what I say when people advise is that we don't have enough information to say one is better than another. So whatever you can get access to first is is the best one for that patient, as far as like insurance and restrictions like that. We know that the most accessible JAK inhibitors, tofacitinib, ruxolitinib, baricitinib, the first generation ones, are pretty nonspecific. I have less experience with the later generation ones that are supposed to be more specific, so I guess time may tell.
Well, if there's no more questions, that brings us to the end of this talk. Rachel, you had an announcement?
Yeah, just before you leave, I just would like to ask you if you could join together on the stairs. So the plan is to try to take a group picture, okay? Let's try, at least, okay? Thank you so much for this table, and thank you for all the speakers. You are great.
patient will be she's live now.
Ah, okay.
She just joined the Zoom. So
okay, okay. Very good, okay.
Is real. I also traveled a lot before, but now find that difficult and don't ever travel alone. I still use a wheelchair at the airport and bring a walker with me. Even with so much functional improvement, I find it difficult at times to live alone. So I recently moved in with my daughter and her family. I feel much safer knowing I don't have to try and do everything myself, and I get to spend every day with my granddaughter. At this point, my CK is back to normal, although I continue to have some pain. In a few months, I'll try again to taper off prednisone more slowly. If I flare, my doctor plans to try a different immunosuppressant that's better for long-term use. All in all, if I must have myositis, I feel very lucky to be doing so well. Life is good. Thank you for listening. I'm happy to answer any questions.
Thank you very much, Debbie. Thank you very much. We had Debbie live before the beginning of the session. Unfortunately, we are experiencing some connection problems now, so we won't be able to have Debbie live at this moment. We hope we can solve it and still bring her on during the session a bit later on. So now we're going to move to our second talk, and it is now my pleasure to introduce Professor Lisa Ryder, a pediatric rheumatologist and head of the Environmental Autoimmunity Group at the National Institute of Environmental Health Sciences, part of the National Institute of Health in Bethesda, Maryland. She has made major contributions to our understanding of myositis, including the role of environmental exposures in disease development and phenotype expression, and today she will talk to us about environmental risk factors for myositis and its phenotypes. Professor Ryder, the floor is yours. Thank you for being here.
Thank you so much. I'd like to particularly thank the organizers for inviting me to speak here today with you. I'm going to speak on environmental risk factors for myositis, and these are my disclosures. The environment is important. The incidence of our autoimmune diseases is rapidly increasing in time. And these are numbers that Fred Miller has put together pre-pandemic. We're going to see a rapid rise, particularly in inflammatory bowel disease, rheumatoid arthritis, type 1 diabetes, psoriasis, and a little bit in myositis. And this is not just related to our better recognition of these diseases. These diseases have a low to moderate disease concordance in monozygotic twins. In fact, the heritability of these diseases in adults is less than 25%. The major genetic risk factors are environmental response genes, and there's biologic plausibility for the environmental factors from in vitro and animal studies. There are strong temporal associations with some exposure with disease onset, and seasonal and geographic clusterings in time and space, as you will see. We have evidence of de-challenge and re-challenge, especially with drugs and biologic therapies, and epidemiologic association or case control studies between exposures and diseases. In thinking about the individual exposome, we need to look across a variety of factors, both internal factors and external factors, and look at these in relation to susceptibility, including genetics, sex, and the developmental stage from conception through late adulthood. And these interact together to create immune dysregulation, inflammation, and the initiation of autoimmunity in susceptible individuals, and later clinical features of disease, and then we diagnose patients. In terms of what we have learned from epidemiologic studies in recent decades, we already know that physical exertion from a case-controlled study is a risk factor for dermato and polymyositis. And then when we looked in our case cohort study, we could see this as a risk factor for MSA-negative children compared to TIF1 gamma positive. In a case-controlled study from Sweden and case-sibling study, we could see psychosocial stressors as a risk factor for autoimmune disease and less so for polymyositis. And then we have confirmed this also in our case cohort study as well. And then smoking has been seen as a risk factor in several cohorts, particularly for Jo1 positive patients with the HLA-DR3 risk allele. This slide of drug risk factors put together by Latika Gupta through her recent review of literature shows a strong evidence for statins with immune-mediated necrotizing myopathy, as you heard from our patient today. In addition, a strong signal for immune checkpoint inhibitors. And then there are selected drugs, hydroxyurea, nonsteroidals, anti-TNFs with dermatomyositis, and protein pump inhibitors with polymyositis, with a variety of mechanisms proposed. We've tried to tackle the environmental factors through several different approaches. First, we partnered with the Myositis Association and Cincinnati Children's to create a national myositis patient registry of about 2,000 patients who enrolled with a 35-patient questionnaire with a lot of questions about the environment. 1,000 patients who enrolled with a 35-patient questionnaire with a lot of questions about the environment. We also created a twin sibling study. These are twins and siblings within 5 years of diagnosis with one of the 4 systemic autoimmune rheumatic diseases in adults and children. They had same-gender siblings within 5 years of age, and when possible, their parents enrolled. So we had 265 such pairs. And then we have the MyoRisk study led by Adam Schiffenbauer of Antisynthetase syndrome, and he presented data today showing infection and antimicrobial therapies to be a risk factor, and he'll have more to say on that in the future about the environment. We also have our natural history study data, and so if we look at season of onset, we can see that the antibodies have different seasonal patterns, with Jo1 a peak in the spring, SRP in the fall, and MI2 in the winter months. If we look at birth seasonality, meaning these are perinatal factors that may be important, we see a stronger signal in children and hardly any signal in adult patients. So we see that Hispanic patients have a fall signal, a fall peak, that P155 more in the winter compared to P155 antibody negative or TIF1 gamma. And then we have HLA risk alleles more in the fall. And these are, again, stronger in the children. From the MyoVision registry, in terms of geographic clustering, we see more prevalence in the northeast for myositis overall, but some hotspots of myositis, with DM a hotspot in the California region and less in the Pacific Northwest, and patients with lung disease, a hotspot in Texas region, and again, less in the Northwest. So we had done several studies looking at exposure to UV radiation based on satellite data. These studies were started by Fred Miller in the adults, looking worldwide and seeing more signal for UV radiation in the equatorial regions compared to northern regions. And this was confirmed in US patients and also found to be true for patients with MI2 autoantibodies, and stronger in women and white patients. In children, we looked at shorter-term UV radiation and looked at exposure based on residential location, looking at surface monitors, and we also saw a stronger signal for patients with TIF1 gamma, MDA5 antibodies, but not NXP2. So I was particularly intrigued that TIF1 gamma has a lot of photosensitive rashes, and XP2 really hardly any rash. And I think there may be some relationship there. We also saw from the MyoVision registry that patients with DM are more likely to have sunburn prior to diagnosis, particularly increased in women, and increased compared to healthy CDC data. And UV radiation we saw is associated with disease flares in adults and children. So then we're interested in trying to dissect what are the exposures based on lifespan. And so looking in children, we are seeing some different exposures than in adult patients. The first study looking at exposures from pregnancy was from Brazil, Adriana Ali's study, where maternal smoking during pregnancy and secondhand smoke were risk factors, as well as maternal occupational exposures to chalk dust and gasoline vapor, and atmospheric carbon monoxide exposure. We recently confirmed this in a case-controlled study from the twin SIP study in which 80% of the patients had JDM. And again, we see prenatal smoking, particularly paternal smoking during pregnancy, to be a risk factor, as well as early life in the first 3 years, household secondhand smoke exposure. We also saw the number of pregnancy complications to be a risk factor. Continuing to look in the children, we collaborated with Ben Larman and looked at a FIP-seq study where he expressed bacterial peptides in a library and looked at antibody reactivities to these. And he saw a high frequency of reactivities to different flagellin peptides compared to healthy controls. Flagellin is found in bacteria and is responsible for motility, but it's also in the gut and increased in patients with Crohn's disease antibodies to this peptide. But these patients with this reactivity were not patients with GI symptoms. And these patients react against a number of different bacteria, but they share a consensus sequence in this flagellin reactivity, suggesting that they are all reacting to flagellin. And this was confirmed in another assay, mesoscale diagnostics, looking at the antibody binding to flagellin. And here, almost half the patients with JDM were flagellin positive, all younger patients and majority patients with TIF1 gamma antibodies. And the only controls that were positive were siblings. And I might add that we also looked at these type of reactivities in adult patients and did not see them in adult dermatomyositis, antisynthetase syndrome, or IBM patients. So we followed this study up with a stool microbiome study with Neil Day and Ann Stevens from University of Washington. We took our cohort and the Seattle Children's cohort. They also enrolled unaffected siblings and parents and looked at both stool and oral microbiomes. Here, within a family, looking at the phylogenetic similarity of the microbiome, patients are more like their unaffected siblings than even other JDM patients to each other. But patients are more similar to their unaffected siblings than parents are to their parents, or siblings are to their parents. But between families, JDM patients are more similar in their stool microbiome than siblings are to other siblings or parents to other parents. In terms of oral microbiome with the saliva, there's a lot of shared phylogenetic similarity in the microbiome between all the different family members, perhaps because of shared food and shared utensils. But still, there are more similarity between families for JDM probands compared to each other, as opposed to siblings to each other or parents. So then when we look in more detail at the specific microbiome, there's still 4% amplicon sequence variance in the stool, differentially abundant in patients with JDM compared to their unaffected healthy siblings. And these bacteria, the ones in red, many of them have been associated with other autoimmune diseases, and some of them associated with disease activity and lupus, autoantibody responses in type 1 diabetes, pro-inflammatory cytokines in rheumatoid arthritis. Some of these bacteria, circled in blue, also contain flagellin. In the oral microbiome, 8% amplicon sequence variants are differentially expressed in JDM probands compared to their unaffected siblings. And again, some of these bacteria are ones seen in patients with adult and juvenile idiopathic arthritis and associated with periodontal disease and also inflammatory cytokines. Now, turning to adult exposures, we worked with Christine Parks, an occupational epidemiologist, on the MyoVision data to look at 1,400 patients who had worked at least a year and at least 10 hours a week, evaluating 12 different types of jobs, 4 different types of hobbies, and 8 different exposures that they may have had, evaluating particularly respiratory exposures with these jobs and hobbies for silica, solvents, and metals, and associating these with myositis phenotypes, because this wasn't a registry that had controls, so we could look at whether the relative risk is based on these exposures for these different phenotypes. Clinical subgroups and severe subgroups. Create a subgroup called lung disease plus fever and joint swelling that represented perhaps antisynthetase syndrome or other autoantibody-associated lung disease phenotype. We also had overlap myositis, where patients had myositis and at least one other systemic autoimmune rheumatic disease. And these exposures were evaluated through a job-hobby matrix scored by 2 occupational epidemiologists, and they were masked to the clinical data. And we assumed that the highest exposure intensity was most relevant because short, high-intensity exposures may relate more to higher internal dose and cumulative exposure. So if we look at these forest plots, and if you could see that a risk would be something that's clearly residing above 1, and protection would be below 1, that patients' silica exposure, patients with greater silica exposure have a greater odds of having dermatomyositis compared to inclusion body myositis in this registry. And there were stronger associations with high certainty exposures and also in females. And this was also stronger in patients who were smokers. We did not see a relationship to solvents or metals with dermatomyositis, and we didn't see a relationship to any of these with polymyositis. We then looked at relationship to overlap myositis, and again, silica exposure through these occupation and hobbies had greater odds of having overlap myositis, and this was true at all levels of intensity, but certainly stronger with higher certainty exposures and in females. And solvents had no association, and heavy metals only association in the highest exposure group. And then in terms of the lung disease plus phenotype, again, silica exposure had higher odds of having lung disease plus phenotype, but was stronger with higher certainty exposures and also in smokers. And then solvents had borderline association with high exposure, and so did metals. And then if we look at all these exposures together, there are increasing risk of this lung disease phenotype if we also add in UV and smoking. So patients with 3 to 5 of these exposures have even higher odds of having the lung disease plus phenotype. So what about mechanisms of disease? There's been some work emerging on this in terms of how these environmental factors may be contributing to disease mechanisms, perhaps best understood for UV radiation. So UVB exposure can induce apoptosis of keratinocytes and increase the expression of autoantigens coming to the cell surface. Certainly, this is seen for Mi2 autoantigen. It can also increase the cellular infiltrate, including T cells, neutrophils, dendritic cells, and macrophages that are recruited to the skin. And this then activates the type 1 interferon response, interferon beta, in the skin, as well as production of other pro-inflammatory chemokines and cytokines. There are emerging mechanisms for other exposures and other pathways of immune activation, and this is best worked out in rheumatoid arthritis. But silica, smoking, and asbestos can induce inflammation and activation of inflammatory pathways and cytokines. Also, there can be citrullination of target tissue antigens or even carbonylation, particularly with silica and smoking exposure, as well as solvents. We talked about UV radiation, and then there can be infiltration of immune cells with different pollutants, and induction of cell death and net formation, not just with UV radiation, but also silica, heavy metals, and smoking, and oxidative stress with smoke, wildfires, asbestos, and silica, and changes in epigenetic changes with several different exposures. There's much more work to be done in this area, particularly in myositis. And we're just beginning to understand then what are the gene-environment interactions. We understand very few of these, but we see that UV light exposure is associated with certain antibodies like Mi2 or TIF1 gamma, and has HLA allele association with Mi2, for example. Smoking with DR beta 10301 is associated with interstitial lung disease and JO1, and statin exposure with DR beta 111 with the IMNM phenotype. Now, many of these exposures are not unique to myositis patients. They're seen in other systemic autoimmune rheumatic diseases as well when they've been studied. So things like UV radiation, smoking, inhaled exposures, microbiome, and drug exposures. Not all of these have been studied in myositis, so we don't know the full degree of overlap. How about future directions of environmental research? Well, we've had to do everything by questionnaire data up till now to see if people have exposures. Going forward, we're trying to look at actual chemical exposures through blood and other body tissues. And this study was done by Christina Lanata and Lindsay Cresswell, looking in patients with lupus at a GC mass spec analysis compared to healthy controls. And they saw increased reactivity, increased evidence of exposures to acidic pesticides as well as phthalates in the blood. Now, whether these are disease-causing, we don't know. There's also a lot of analysis to look at GIS, linking residential exposures to satellite and ground sensor data, and then the possibility to wear sensors or have them in the home to look at things like UV radiation, activity levels, monitor particles in the home, carbon monoxide, or volatile organic chemicals, and others. We are also very interested now in a new paradigm called the exposome, looking at the total number of exposures that happen throughout the life course, from conception through the lifespan, and their implication on developing chronic diseases. So trying to take in more of the exposures that people have and see when in their life course they occurred. And we call this type of work GWAS to assess these many exposures simultaneously and look at their impact on different omics. Also, there's emerging data that there's association of some exposures with disease activity and flares, or disease severity, or even certain manifestations of disease. And I would say this is emerging particularly in rheumatoid arthritis, particularly with smoking and with silica exposure and air pollutants. So today we talked about a few of the non-infectious exposures. We see that psychosocial stress and physical exertion are risk factors for myositis. Commensal bacteria in the gut and oral microbiome may have implications in juvenile dermatomyositis. Ultraviolet radiation, particularly for adult and juvenile dermatomyositis, and some of the autoantibodies, and smoking and silica and heavy metals for patients with lung disease in particular, who have interstitial lung disease, overlap myositis, or antisynthetase-like disease. There are also drug exposures as well, and that these can activate different relevant pathways in the immune system. So the rapid rise in autoimmune diseases and other data implicate a strong role for environmental factors in these diseases. These are risks with other systemic autoimmune rheumatic diseases, and these exposures vary across the lifespan. They contribute to these immune pathogenic mechanisms, and they're risk factors for developing myositis with certain phenotypes, but they also may influence disease activity, flares, or even the disease course. So I'd like to say that we're ready for a paradigm shift, that modifying these exposures and lifestyle factors may not only prevent disease, but it may decrease its severity and aid us in controlling disease activity. We're going to embark on these type of studies for our next phase. We have a new study opening very soon called the Fast for DM study, where we'll be modifying dietary factors and looking at effects on disease activity. And then later this year, we'll open our image study, Investigation of Myositis and the Global Environment, which is an online questionnaire study to look at the exposome in patients with myositis and their relationship to phenotype and disease severity. So I'd like to particularly thank the members of our group, particularly Fred Miller, who I consider the father of this field, and long-standing mentor, many fellows and students who worked with us, collaborators from NIEHS and elsewhere, and most of all, our patients and supporters. Thank you very much.
Thank you very much, Lisa. And now I open this talk for discussion.
Lisa McCann from the UK. Thank you, Lisa. Fantastic talk. I'm just wondering, have you looked at the effects of vaping as well as smoking? It's clearly more common in the young population that we look after these days.
Mm-hmm. We added that recently to our questionnaires, and I've heard about patients with vaping, particularly teenagers who have developed MDA5 disease after that. So I think there could be something there, but we need to have more data.
Another question?
That was such a wonderful talk. Thank you so much. I was wondering if you're also looking at protective factors, things that if people are doing different things to stay healthy in particular ways, they might be at less risk of developing myositis.
Well, I hope that our next study will help us look into that a little bit with the, we're not only going to be giving high-dose omega-3 fatty acid in a randomized controlled trial, but also a general healthy eating pattern diet through a diet coaching program, and I hope these type of interventions may be protective. So.
Thank you. I just had one last question. You touched this on at the end, so probably some of these factors also influence disease activity. And from what we know now, from a practical point of view, what should we advise to our patients on the environment exposure?
Sure. So I think it's, first of all, important to take a good environmental history in the clinic and see what factors seem to be triggering disease onset or disease flare. And then for a lot of our patients with JDM, for example, we really go over photoprotective measures in great detail. We even use this app, UV Lens, that shows us the UV radiation at all the different hours of the day. We encourage patients to look at that as well to stay protected from high dose radiation from the sun. Yeah. So those type of things could be helpful.
Thank you. Thank you very much, Dr. Lisa. And before we move on, I think we can now have Debbie on screen too. So if you have any comment or any question, Debbie, good afternoon. Thank you very much for joining us. And we are sorry that we had some connection issues, but now we are hearing you, and I know that you are also hearing us. Thank you for sharing your patient history with us, and it was very touching. And if someone wants to make a comment to Debbie or some question, I think she's happy to take it. Karen.
So, Debbie, thank you very much for joining us. What would be your best advice to a patient, that you can relate to because of a similar journey?
My best advice, I think, would be to try and certainly find a doctor and take it seriously. When you have systemic weakness going on, I should have known better. I feel like that I should have taken it more seriously and not thought it was just a lot of other factors, getting older and GLP-1s and so forth. And be patient. It may take a while to find the right treatment. I was lucky, and mine worked pretty quick. But, I think there's a lot of hope out there and that you can get better. And so I would just hang in there and And I would also, as a physical therapist, I think that don't be shy about using assistive devices. You will have more independence and be better off if, don't deny your need for it. If you need something to assist you, get the right devices and right modifications.
Thank you very much. Any other questions? If no, thank you very much, Debbie, for joining us. Thank you.
Thank you. Thank you.
It's now my pleasure to introduce our final speaker, Professor Pedro Machado. Is a professor of rheumatology in neuromuscular disease and consultant at University College London Hospital and Northwick Hospital. Professor Pedro Machado is widely recognized expert with major contribution in the field of myositis and neuromuscular disease, which will give you a talk in how infection and vaccine as trigger of myositis. Thank you very much.
It's okay. It's okay. I'll use the keyboard. Yeah. Okay. Thanks for the introduction, and it's always a pleasure to participate in Gcom. So I'll cover infections and vaccines as potential triggers of myositis. These are my disclosures. So I'll start with infections, and I'd like to make here a difference between infective myositis and the potential of infection triggering a self-perpetuating autoimmune phenomenon that will lead to an idiopathic inflammatory myopathy. And my understanding from the title of this talk is that we're talking more about the later. But of course, there's been infectious myositis described in the literature, and this can be caused by a broad range of agents, including bacteria, fungus, parasites, and viral agents. In this scenario, they are usually uncommon because the muscle is quite resistant to infection. There's usually another concomitant factor promoting the infective myositis, and this could be a previous trauma, disuse, malnutrition, dehydration, critical illness, or the presence of an immunosuppressive state. Often there is direct invasion of the tissue by the infective agent, or there is a systemic, excessive, uncontrolled inflammatory response associated with infection, sometimes in the context of a cytokine storm-like syndrome. And a wide range of infective agents have been described as potentially associated with infective myositis. Probably the most common ones are viral agents. Influenza A and B and also enterovirus are the most commonly reported ones, but also other respiratory, enterovirus, arboviruses, herpes virus, hepatotropic virus, and retrovirus has been described as potentially associated with infective myositis. But I think the main topic of this talk is the autoimmune myositis. And I guess I'll start with a disclosure. And the disclosure is it's extremely difficult to prove causality between infection and the actual development of an idiopathic inflammatory myopathy above the expected incidence of that same myopathy in the general population. So that's my first disclosure. This is extremely difficult to do. And why? Well, one, it's particularly more difficult in the myositis field because these are relatively rare conditions, and therefore our statistical power is always lower. There's often recall bias from patients in terms of what they report as an infection. And in fact, if you look at the literature, many studies are relying on self-report of infection before the diagnosis, which can be inaccurate. There's also a lot of temporal ambiguity. Because infections may precede the symptoms, but these symptoms themselves can be manifestations of the idiopathic inflammatory myopathy. and they could be erroneously classified as symptoms of infections. Okay, so all this is really blurring the evidence. And then there's multiple variable latency times between an infection and the onset of the infectious disease, and then the potential onset of the idiopathic inflammatory myopathy. And this, of course, really makes it complicated in terms of causal inference. More, well, there are multiple overlapping mechanisms that have been described as potentially associated with this phenomenon, namely molecular mimicry, cryptic epitope exposure, bystander activation, or a dysregulated innate immune response, and they can coexist. So it's very difficult to disentangle sometimes between all these mechanisms. There is confounding by immunosuppression or an underlying immune dysregulation, and then other potential contributing factors are often not considered or not adjusted for in the analysis, and this relates very closely to the concept of exposome that Dr. Ryder already mentioned. I really think that we need that integrated view if you are going to be able to disentangle all these potential factors that are often correlated between them. And this could be things like smoking, obesity, changes in diet, social deprivation status, personal lifestyles, air pollution, and so on, and many of them have been mentioned today. So, despite that, there is some evidence, so let's look what the literature tells us. And I'll start with a study that was already introduced by Dr. Ryder, led by her team, so that's the MyoVision registry. You already heard a little bit about it. A cohort of almost adult patients with immune-mediated myositis enrolled between 2010 and 2012, although the cohort started early. This was to try to attempt a little bit to control for recall bias. There's the distribution of the patient population. They also considered a lung disease positive population. This was the group that had also had fever or arthritis that Dr. Ryder mentioned, and there was also a group with overlap myositis. So what they showed in this study is that febrile illness and respiratory infections and GI symptoms like nausea and diarrhea, they were all more common in dermatomyositis and polymyositis as opposed to inclusion body myositis, and that's what you can see there in the graph on the right side of the screen. With all the confidence intervals for the various factors beyond 1, therefore all statistically significant. They've also showed that upper respiratory tract infection was associated with lung disease, and as I said before, this is febrile or arthritis-associated lung disease, with the strongest signal being observed for pneumonia, where you can see there that there was an odds ratio of around 5. These associations were also observed to some extent in the overlap myositis group. Well, but then a question that could arise, well, is it infection or is it the antibiotics that you're giving for the infection, right? So they did an analysis that was adjusted for antibiotic usage, and in fact, the signal increased when the analyses were adjusted for antibiotic usage, supporting that it is indeed the infections that were associated with this signal that was observed. What does the data tell us ultimately? Well, it tells us that respiratory and GI infections may contribute to some IM phenotypes. It does not show causality, it shows associations. Infection could potentially act as triggers, but they could also be early features of the disease. ILD is frequent at the onset of antisynthetase syndrome and overlap myositis. Pneumonia could have been misinterpreted as interstitial lung disease. So there are several question marks here, but it does suggest that infection could at least have a modulating effect and influence the phenotype of the patients. It's still to be answered if there is a true causal relationship between the two. There's multiple pieces of evidence that have been shown in the literature for various agents, mainly viral agents, I've included here the ones that have more commonly been reported: influenza, Epstein-Barr virus, cocci virus, adenovirus, SARS-CoV-2, obviously, and we'll speak a little bit more about that, but also retrovirus like HIV and HTLV. Influenza, Epstein-Barr virus, and adenovirus have been associated with the presence of anti-Jo and anti-PL12 antibodies, potentially related to a molecular mimicry mechanism with host tRNA synthetases. Anti-MDA5 antibodies and anti-PL7 antibodies have been associated with SARS-CoV-2 infection, potentially with mimicry with the spike protein and toll-like receptor activation being mechanisms underlying this association. Cocci virus have been associated with increase with various myositis-specific antibodies. HIV and HTLV have been associated with both polymyositis and inclusion body myositis. Colleagues from Spain, they did this large study. It was a multi-center collaborative study that involved 11 referral centers, almost 800 adult patients with IIM recruited post-2022, all assessed with a wide range of antibodies. And they've shown that there was a serological shift over time. So they observed a rise of anti-PL7 and anti-MDA5. So the classical dominance of anti-Jo antisynthetase, anti-Jo antibody antisynthetase syndrome that we often describe. It's usually around 30–40% of the population with antisynthetase syndrome. There seems to be a shift with more predominance of an anti-PL7 and anti-MDA5 population, possibly related to that mimicry mechanism that I mentioned in one of my previous slides. Moreover, they also describe that patients with prior SARS-CoV-2 infection or heterologous COVID-19 vaccination showed a significantly higher frequency of multiple autoantibody positivity, and that's that group that you see in the middle, the overlapping group. So two potential contributing factors here. Although the presence of antibodies will not necessarily mean the development of the disease, so it's still to be completely elucidated if this will lead to a true increase in the disease prevalence. So let's now look at population studies. There are now many. I had to make a selection, so apologies to those that I have not selected. This is a study that included almost 7 million participants, and more and more 50-50 divided into people with infected by SARS-CoV-2, so that develop COVID-19, and the other half the controls. The observation period was more than 6 months, more than 180 days. So this is a large populational study. And they looked at multiple autoimmune and autoinflammatory disorders. I will focus on the idiopathic inflammatory myopathies only. And in this large nationwide study, there was no increased risk of idiopathic inflammatory myopathy after SARS-CoV-2 infection. There's obviously conflicting data. So I'll show you the other side of the coin. So this is a study done in the TriNext collaborative network, smaller study, less number of cases, but still a large study with almost 4 million participants and with a similar observation period of around 6 months. What did they show? Well, in this study, there was an increase in everything that they studied, which I always find a bit strange when everything goes in the same direction. Having said that, the signal was probably lower for the idiopathic inflammatory myopathies as compared to the other autoimmune diseases with an odds ratio of close to 2. And obviously, I could not cover all the large nationwide studies that have been published since COVID-19. So I looked at the systematic literature review. And there was one, quite recent, published, I think, in the end of late 2025. And this, taking all studies in combination, this included 97 million individuals. Okay, so I thought this could be a very good source of information. Now, again, they looked at various autoimmune diseases. It's a very informative study, but again, let's focus on the idiopathic inflammatory myopathies. If you look at the total, the risk for all autoimmune diseases combined, there is an increased risk, which is very much in line with all the previous studies that had been described overall, but then in each study you could clearly see differences. Not all autoimmune diseases are the same. Now, in this study, which is a meta-analysis of everything that had been published, there was no signal for idiopathic inflammatory myopathies. So apparently, SARS-CoV-2 infection, COVID-19, has not led to an increase in the prevalence of idiopathic inflammatory myopathies. Okay, it's time to move to SARS-CoV-2 vaccination and the risk of myositis. So this has been a controversial topic for many, many years, and those who are neurologists, and especially if you work with patients with Guillain-Barré syndrome, are very familiar with these discussions. Various theories have been proposed to justify this potential link. I've already mentioned some of these theories, including molecular mimicry, bystander activation, but it could also be adjuvant-induced effects. There's been several case reports in the literature associating a certain autoimmune disease with vaccination. However, if you look at the epidemiological data, actually that data is usually against such an association, with a couple of exceptions, one of them being the association between GWAS and H1N1 influenza vaccination. And importantly, overall, I would say that in the large majority of cases, causality has not been demonstrated. Where some associations have been described, I think the risks can vary depending on the type of vaccine, even for the same agent, and also they can be disease-specific. And overall, and I'd like to clearly say that, I think the benefits of vaccination have vastly outweighed the small potential risk of triggering an autoimmune disease, and vaccination is one of the best things that has happened in the history of medicine, and they've saved millions and millions and millions of people. Okay, so what is the clinical concern? The clinical concern is raised by several reports of new onset of autoimmune disorders after vaccination. This could potentially be the case with myositis. What is the challenge, the analytical challenge? Well, I mentioned that at the beginning in my disclosure, let's say. It's very difficult to distinguish between true vaccine-induced risk and the baseline background incidence of a certain autoimmune disease, especially when you vaccinate almost everybody, right? If you vaccinate almost everybody, some of those people will eventually have myositis anyway. But if by chance you happen to have the vaccine 2 weeks before you develop the symptoms of myositis, yes, of course, you'll say that it's because of the vaccine, but, it could just be chance. So I kind of combined two studies, so to allow me to look at a large number of participants. And I combined these Korean nationwide study that included around 9 million individuals, and the Nordic Scandinavian study where the Norwegian and the Swedish combined their data, and they look at more than 13 million individuals. In the Korean study, the focus was a bit more broad, so they look at all the autoimmune disorders, and they only looked at mRNA vaccines. In the Scandinavian study, they focused on myositis, and they looked at both mRNA and adenoviral vector vaccines. Duration of the follow-up in the Korean study, it was minimum 1 year, so a long follow-up, but pretty good follow-up duration also in the Nordic study with 6 months duration. Now, the studies, they differ a little bit in terms of the control population. The Korean study used an historical cohort, so they went back 2 years, and they looked at that as their control population, while in the Nordic study, they looked at the contemporaneous cohort that had been unvaccinated. So slightly different in terms of the control population, study design, let's say, but both equally valid, I think. And in both studies, the outcome was defined by ICD-10 codes. So in terms of the Nordic study, as you can see there was no association between vaccination and the development of idiopathic inflammatory myopathy, neither for mRNA vaccines nor for the adenovirus vaccines. And corroborating the myositis null hypothesis that was described in the Scandinavian cohort, also in the Korean cohort, the odds ratio was 1, so therefore no increased risk, allowing us to conclude that across both an European and Asian genetic background population, the evidence appears to reject the hypothesis that SARS-CoV-2 vaccines are triggers for idiopathic inflammatory myopathies. And now, interesting, I think it's interesting to look at the positive findings. That's something that you'd expect to find to validate the study. And we know that, and this has been very well described, that mRNA vaccines can cause myocarditis or pericarditis, right? So therefore, and I think they were really conscious by doing this. So they looked at an association with myocarditis and pericarditis in that population. And as expected, and this kind of validates and reinforces the validity of the study, there was an association observed with the development of myocarditis and pericarditis, which has been very well described. Interestingly, there was an exception in terms of the autoimmune diseases with an increase in SLE, although as you can see there, the risk is small, with the lower bound of the confidence interval being 1.02. Okay, just a few final remarks. I think overall there is still an evidence gap here. For the vaccines, I think the evidence is actually pretty good. I don't think it's the same for infections, but we still need those robust epidemiological studies that can prove causality. There are still some conflicting results, and it's still really important that we establish multinational longitudinal studies with reliable linked data where we can interrogate these questions, exposures. It will inform in the future if there are additional pandemics, if there are mass vaccination programs, ecological shifts when introduce new medication. And this is important because this will build readiness for faster targeted response to global health events, and it will advance our understanding of these complex diseases, the idiopathic inflammatory myopathy. So thank you for your attention, and I'm happy to take questions.
Thank you very much, Professor Machado, for this excellent and such important talk. I would turn to the audience for question, please.
Dinner time?
Since no one wants to ask a question, I'm really keen to understand. Recently with the SLE Europe Congress, they were looking at EBV reactivation, and we know this is a thing in multiple sclerosis and SLE. Do we have anybody working on this in myositis?
I do not know. Is anyone working in that? Yeah.
When John Harley discovered that EBV is associated with lupus, he used our myositis patients as controls and didn't see any signal. So,
Thank you, Lisa.
Hi, Pedro. Great talk. So we all have these patients that seem to temporarily have onset, for example, of MDA5 right after COVID vaccines. Knowing what you know and the data that you presented from a clinical aspect, do you feel comfortable revaccinating these individuals or not with the COVID vaccine after they've had that phenotype?
Yeah, so that's a good question. I think in clinical practice we need to be a bit pragmatic, and although I always try to inform my practice with evidence, real life is slightly different. I think the MDA5 case, it is different, and because it's such a small number of individuals, it will be very difficult to capture even in very large epidemiological studies. So yes, I would feel reluctant in revaccinating that scenario.
Pedro, so based on your talk and Lisa's talk, it seems overall there is much less link between IBM and the environmental or infectious exposures?
Well, there is less data at least. And as you and I know very well, IBM is a very enigmatic disease. And I think maybe that's something that we need to work a little bit more on. Yeah, that's true as well. That's a very relevant factor in terms of late diagnosis. Yeah.
Okay, thank you. Thank you very much, Debbie, Lisa, and Pedro. I think we had a wonderful session, but it's not the end of the day. So please don't forget, we have the networking event, our dinner at 8 p.m. at Patio da Galé. You are more than welcome, and it will be a wonderful night for sure. Thank you very much.